Halo, halo, tes 1 2 3 1 1. Oh, the judges are not here. Please. Oh, please sit over here, judges. One here and one here. Everybody's gonna be able to see the screen anyway. You're gonna. Oh, wow. Okay, that's a big screen. All right. All right, sure. Okay. All right. Okay then. Right. Okay. All right. Okay. Thank you very much. Let's begin. This is Arena 3. So we will begin by introductions from each of the group. Just introduce your group number, your name, and where you're from. And then we'll do the coin toss to decide whoever gets to decide who present first. And then we move along as usual. Please.
Okay. Good morning, everyone. My name is Sandy Octaviana, and this is my colleague, Mega Verina Warsito, and we are came from Brin. So that's all.
All right. Very good.
Hello, good morning everyone. My name is Sunuta Froji and I have with my college actually Miftahuddin Majid Khawairi, but he couldn't manage to come. And we are from Kire Imeri and we are from D33. Thank you.
Fantastic. All right. Heads or numbers? Heads or numbers? Heads?
Okay, number.
Number. All right. Okay. It's heads. So you can decide whether you want to present first or they can present first.
I think ladies always start first.
All right. Okay. So he decided these 7 will present first. All right. Just a short repeat. The first presentation is 2 minutes. Judges are able to score up to 3 points based on that presentation. And then each question or answer session will be limited to 1 minute. If it goes past 1 minute, you will have a minus 0.2 and you will be stopped. It will not continue like during our practice. During our practice session, if it's over, you can still continue and then you get a minus per minute, right? Per 10 seconds. But in this one, you will be stopped abruptly.
And who will operate the PowerPoint?
Right, the PowerPoint will be from here, so please feel free to use it. Okay, go ahead. All right, thank you very much, the audience, for coming, and I hope this will be a productive session. When you're ready, I will start the timer. When you start speaking, I will start the timer. All right.
Okay. Good morning, everyone. Welcome to our project. As you can see here in the picture, that antibiotic resistance increase every year. That's why we need new drug discovery. In our previous study, we found the microbial natural product, namely Plasmidis, based on the 16S RNA. And the aim of our project, there are 3. First is to identify antimicrobial activity, and the second one to identify the biosynthetic gene cluster, and the third one is to identify the biosynthetic pathway. And our method, starting with cultivation and extraction, and the second one is the antimicrobial assay, and the third and fourth one is the analysis of the metabolomic and genomic data. So our result highlight that our strain have anti-candidiase activity, and then the metabolomic and genomic approach unveil the biosynthetic mechanism of the strain to produce antibiotic compound. Thank you.
Thank you.
Okay.
Okay, all right. Thank you very much. That was 1 minute, 30 seconds, but I minus the time when we had the technical problem, so.
Okay.
All right, please.
Can I
begin.
start? Okay, thank you very much. I think it's very interesting topics because I have also background the bacteria and exploring the antimicrobial resistance. One thing that I want to comment is I think your approach is not very specific, because if you want to find the new drug for antimicrobial testing, or that you want to use as antimicrobial, you have to discover the outcome, not the metabolites. And I want to comment with your technique, the method. The first one is your rationale is not good, and your approach is not really good. And the next one is for the method. You use the methanol extract. You know the methanol is very toxic. How you ensure that the antimicrobial activity is coming from your extract or from the methanol? Okay, I think that's for the first session.
Thank you for the questions. For the approaches that we use, we ensure that the genomic and metabolomic approaches is the latest, the advances technology that can be used to reveal the novel and compounds according to each biosynthetic gene cluster, which can be analyzed using bioinformatic analysis. While we use methanol extract, for the information here, we want to discover new compound, and for the antimicrobial activity, we also tested the methanol, which is the negative control. So we ensure that the activity is solely based on the activity of the compound or the extract.
Okay. Okay. If you are thinking that your genomic profile, are you targeting the antimicrobial protein instead of the substance or the bioactive compound? The second one, how do you ensure that there's no any cluster between methanolic extract and the biocompound itself from the bacteria? And you need to ensure there's no any side product when you use the metabolic agents as your target, because in the future you will make this metabolic by reacting substrate and your enzyme probably then you gather from this, and there will be a side that you need to do the purification.
For the information here, we use the extract, which could contain like secondary metabolites and also primary one. But why we use metabolomic, which contain a lot of compound? Because we want to analyze what kind of compound that will be expressed by the strains itself. So we can integrate both the genomic analysis, the genome data, which we want to know the biosynthetic gene cluster, and which pathway that is responsible for the production of specific metabolite that have the activity against the pathogenic bacteria itself, which is we use Candida albicans here.
Okay, you don't mention about my methanolic side products. Is there any cross product from the reactions methanol with the compound? And also the last one, if you are thinking about the reactions, you need like another optimization. You need a living organism to have an optimization of the product, right? I think that will be your homeworks,
Okay.
and that's why I consider it's not a practice approach.
In terms of can we make sure that there is no cross reaction between the methanol and also the compound that we extracted? When we do the chromatographic analysis, we ensure that we have the the methanol is identified. Like cross analysis between the methanol and also the extract, and we knew from that and compare it to the database from the website what kind of compound that can be expressed by the myxobacteria itself. So I'm sure that why we use methanol, which is like the base, the basic extraction solvent that was used worldwide here. So if you,
Another question?
1 minute, please.
Another question?
Yes.
No, I mean you did not reply my another question about the the metabolic activity that you need to design with the living organism to optimize your productions. I think that's your homework. And the next one, don't you think that methanol is only non-polar solutions? Meanwhile, we have the polar solutions. We have polar biocompound that you should take. Probably they have the potential as the antimicrobial. That's why I'm considering why you only use this methanol extract instead of it's toxic. It's very toxic, and you cannot get the non-polar the metabolites that can be soluble in non-polar solutions. And the next one, I think from your result, the MICs you measure here is not MIC, it's not the inhibitor concentration. You should treat the CLS, right? Right? You should treat the CLS. It's MBC, not MIC. Okay, thank you.
The CLSE is also one of the methods that was described in the GLSI is the minimum inhibitory concentration, which we determine whether the activity is consistent, the bacteria is consistent susceptible or not, right?
Yeah.
Because there's a lot of them. And also for the polarity itself, for the methanol, why we use methanol? Because this is the preliminary study, and we use first methanol as the baseline of our study, but maybe for the next one, we will try to optimize the extraction methodology. And for the, for the metabolism, why we use it, this is the preliminary study, and the RSM analysis will be conducted next.
Okay. So
Very good. Last section. 'Cause
Last
it's last minute.
section.
Yeah.
okay, I have like two for the MIC and MBC. I think you measure with the I don't know the yes, you have to differentiate the MIC, its minimum concentration to inhibit the growth, not to kill the bacteria. If your methods measure the minimum concentration to kill the bacteria by detecting the substrate, right? The substrate appear after the bacteria metabolize that. That's why the substrate change from blue to pink, right? So I think that's the misunderstanding in your research. And also for the genomic profile, do you want the first question actually? Do you want to target the antimicrobial peptide by doing all the genomic profile? Right.
Okay.
Last answer.
Okay. There's a lot of questions here. And for why we use like MIC, not MBC, like this is solely, currently we only use MIC to determine the activity. And the second question is about for the resazurin test assay. Yes. It is like we use the extract from the bacteria to analyze the activity. And for the growth itself, we do according to the guidelines. So it has already been tested. So I'm sure that the methodology is validated. And for the last questions, is about
The genome.
Oh.
What do you mean by doing
by doing the genome analysis is we want to analyze the gene that was responsible for the expression of the metabolites.
Sorry, stop. Thank you very much. That was an amazing first round, ladies and gentlemen. Thank you very much. All right, so now we have I think we have a 5-minute break for you to consolidate your question and answers, and then we continue to the round 2, which begins with.
Yeah, okay.
Please feel free to read up on your presentation. Please go there to prepare your presentation also. That was really good, guys. That was really good. That was really good. That was really good. I think you.
So I have a problem with the resazurin. I have also experience with the resazurin because the resazurin will provide a substrate that can be metabolized but with the bacteria. causing
Interact with the bacteria itself, but
So you measure the minimum bactericidal concentration instead of minimum inhibitory concentration.
yes, that's I get what you asked.
Yeah. So just change the MBC, the MIC to MBC. It will be good.
Yeah.
It's really good. And that is the current score we have right now. D33 at 11 and D7 at 6.8. It will be fine. You can catch up because as you probably see, it is kind of easier to attack rather than defend. So you have time to maybe see his poster outside to compose some questions already from it. Bisa. Udah. Kenapa tadi ya? Baterai ya? Oh ya. Tes tes. Ya oke.
Thank you, Dr. Darwin.
Okay, 2 minutes left. After 2 minutes, I will tell that the 2 minutes over, and then when you start, I will start the timer.
Right.
Judges could hear well? Alright. Okay, 1 minute.
Ya, di sini aja atau di sini aja? Sini aja. Saya. Oh iya benar. Ntar ada operatornya nggak ada soal. Oh iya. Oke, langsung next slide aja ya. Sudah.
Okay, whenever you start, I will start the timer. Ready for round 2? Alright.
Okay, let's start. So I will deliver why we need to understand that the PCV13 is very impactful to Streptococcus pneumoniae, or how far this impact will be for Streptococcus pneumoniae, because we are now implementing this PCV13 in Indonesia since October 2022. So I think it will be good. It will be good for us to understand this one. So we try to use the secondary data that are uploaded in Pathogen Watch for all this Streptococcus pneumoniae, and we have collected data for some countries, and we have screened this data, and we only use 5,000 data of whole genome sequencing for the analysis. Next. So from this whole genome data analysis, we found that there is significantly different from the non-vaccine serotype and vaccine serotypes. And we also found that they are shifting the antimicrobial resistance from the more resistant to less susceptible after all the PCV13 introduction in 9 countries. And we also do the comparison with the non-vaccine serotype, the 22F and 33F of serotypes increase after the PCV13, so it will be our evaluations. And if we see the antimicrobial resistance, we have found decreasing of multi-drug resistance strain after the PCV13 introductions. Thank you.
All right, very good. 1 minute and a half.
Maybe I would like to ask why, after the usage of the vaccination, there's the decreasing number of the AMR, EMR susceptibility to the patients.
Okay, so why they decrease? Because in the PCV13 they have like vaccine serotype. We call it vaccine serotypes, and this vaccine serotype has higher resistance to the antimicrobials. So by implementing these vaccines, we can reduce their prevalence and also reduce the resistance of this Streptococcus pneumoniae. And then we can find that in all the data we found, the resistant gene and not the resistant characteristic will shift to the more susceptible because we eradicate all the vaccine serotype which are resistant to many antibiotics. That's why the antimicrobial resistance shift to less resistance.
Do the serotype affecting the resistibility of the bacteria itself?
The what? Sorry? Can
The
you?
serotype of the vaccination affecting the
The resistance?
the resistance.
Okay, it's not the serotype affecting the resistance, but the vaccine serotypes, the most invasive serotypes, including in this PCV13, bring their resistance to antimicrobials. So can you imagine, there are 10 people with 8 people resistant to antibiotics, and we eradicate the 8 people, and it's only less. It only remain 2 people, so we have less antimicrobial resistance instead of we have 8 people with resistant antibiotics. That's how the vaccination works to reduce the antimicrobial resistance after the vaccine introductions.
And about how about the vaccination itself? Do it work for the patients that previously had the pneumonia or the other kind of disease that was affected by the bacteria that was used for the vaccination? I
Okay, thank you. So one is your misunderstanding. It's not the immunization vaccine. It's not antibody-based vaccine. It is antigen vaccine. So it's not to be delivered or administered in the patients. It should be in our community by vaccinating. It's like COVID. By vaccinating our community, we will reduce the IPD, the invasive pneumococcal disease, or the cases. So that's how the vaccine works. It's not antibody-based. So it's not given in patients.
Can you make a clarified why you use pneumococcal vaccine in your study? Please make it clear.
Yeah, the PCV13 is most widely used recent vaccinations. We have now PCV15, we have now PCV20, but it's not officially administered in world. So in the world now, they are using PCV13 worldwide. So we can use this PCV13 as the evaluation how it will impact to the serotype distribution and also the antimicrobial resistant profile.
If there is any mutation in the bacteria itself, too, the activity of the vaccination, or do we need to change the serotype in the vaccination?
It's very rare to find this mutation in bacteria, including in PCV13. To consider the emergence, that's why we highlight the emergence of non-vaccine serotype among IPD or invasive pneumococcal disease patients. So they will replace the previous non-vaccine serotype to be more invasive after the PCV13 introduction. So there is no any mutations happen in the bacteria.
You need to elaborate more on your questions.
In figure 4, can you explain more
The what? The figure 4?
the antimicrobial resistance here?
Yeah, okay.
What do you want him to explain more about figure 4?
This one. Antimicrobial resistance in this graphic.
Okay, so I provide the figure A for the antimicrobial profiles. So you can see that there is reductions percentage before and after PCV13. And we see here, there is also reduction in the multi-drug resistant strain of Streptococcus pneumoniae, which is very relevant with this graph. So by reducing this MDRSP prevalence, we're reducing the antimicrobial resistant profile of Streptococcus pneumoniae by implementing this PCV13.
I want to know about the rationale how the antimicrobial resistance was shifting after the implementation of vaccinations. How do it works?
Two things. First one, by reducing the prevalence of invasive serotypes, we're reducing the use of antibiotics. The second one, by reducing the prevalence of the vaccine serotypes, which are resistant to many antibiotics, like I mentioned before in your first questions, we're reducing the antimicrobial resistant profile of Streptococcus pneumoniae. Like 8 out of 10 people with the resistance, like that, how the vaccination works.
Last session. Last question. Guess we're at 5 minutes. Finish? Nothing? All right, then we.
Thank you very much.
Thank you very much. That was again really good. I hope all of your sessions are like that. So now we have come to the second part of it, to the second second round. We finished the second round already, and so now we can see. Oh, is there a way to display the scores? Yes. Please stand. Please come here. Yeah, no, we have to shake and then we take a picture and all that. Thank you, congratulations.
Congratulations.
Congratulations. Congratulations also, you did good. Thank you. Sorry, we should take a picture. Yeah. We should. A better one. Good game, good game. Thank you very much. Thank you.
Okay, so for this
Don't worry. You still have a second round,
Yeah,
right?
yeah.
Waiting for the bracket.
uh,
Good job.
I have to bracket.
Oh yeah, okay.
So for the 33, we'll move on to the match 10, which moves to arena 2. Okay. For the 07 is moving to match 17.
Which arena?
Which is arena 1.
Arena 1. Alright. D7.
So next up for this arena is D49 versus D38.
D49 versus D38. Alright.
Should I move now or? If you want to move now, it's okay. But if you want to use the practice room, it's also okay.
It's not started yet, right?
You can check the time. It should start on the table.
They will finish all the first round, right?
We're not insane. So it's in the.
Alright. D38 versus D49. Okay. I expect the same zeal and the same spirit from the previous match. Udah, belum. That was really good, right? I was really impressed by the back and forth.
I feel pressure.
Oh, no. You should be a good example. You should be hyped up.
Please, no.
You should be high-spirited. Okay, we're gonna start by introduction. Just your name and your background. A bit of your background, like where you're from, and then we will do the coin toss, and then we prepare the presentation, and then we continue just like before. All right. Please, D49, start by introducing yourself.
Good morning, everybody.
Morning.
My name is Fikri Hanum Humaira, and my friend here is Yani Haidina. We came from National Research and Innovation Agency, especially in the Department of Applied Microbiology. Thank you.
Oh, do you don't want to introduce yourself? It's okay. All right. Okay. Please, D38.
Hi, good morning, everyone. So we are team D38. I'm Fabi, and my friend is Miftah. We are from Summit Institute for Development from Lombok. Hi, thank you.
Fantastic. Fantastic. Okay, thank you very much. Now for the second part, we're gonna do the coin toss, numbers or heads. Call it number. All right, she called first number, so you got heads. It's a number, so you decide whether you want to present first or let them present first. Wait, they present first? Oh, you go first. All right, please prepare. Oh, okay. All right. Some concerted effort here. Very good. Okay. Just a reminder, your presentation can be scored up to 3 points by the judges. So do it well, and then after that we continue with the question and answer session, which lasts for 6 minutes. I will start the timer when you start speaking.
Okay. Good day, my lovely audience. We want to tell you about our story journey to the east. We went to Sulawesi, the eastern part of Indonesia, to study about the most expensive food in the world. Now, maybe some of you are asking, do we really have that? Or why do you have to go thousands of kilometers to study that?
Well, actually, yes. We have ebony, an endemic of Sulawesi. Ebony is one of the most expensive wood in the world because of its strength and durability. However, based on IUCN, ebony is listed as a vulnerable species, one of them because of the plant disease.
So we came up with this idea. Actually, if we can understand the microbial diversity, in this case bacteria, from the asymptomatic and symptomatic leaves of ebony, we can formulate the natural biocontrol against this pathogen causing the disease.
At first, we thought that symptomatic leaves will have higher bacterial pathogen. However, we have a very interesting data where the symptomatic one actually have more beneficial bacteria. For example, in Spingomonas methylobacteria and bacillus actually abundant in asymptomatic leaves, where those bacteria are highly reported to have antipathogen activity. So what it can be happen? So this is maybe one strategy of the plant for survive of the pathogen and stress condition by recruiting some beneficial bacteria to help them to cope with the situation. So this is very interesting to isolate those bacteria and then formulating them as a biocontrol agent for the survival of the ebony. So we are team D49.
Thank you, and we are open for discussion.
All right, 1 minute per question. Total of 6 minutes per session. I will start the timer when you speak. Yeah, of course.
Okay, thank you so much. My first question is about your, in the background you say that, what is the cause, I think the research question is, what is the cause of disease in the celebica? But it seems to me in the conclusion you said that your conclusion is about the potential bacterial to prevent the plant pathogens. So I see it's not explain what's your research question.
Okay, thank you. So this is the problem of our study. So the ebony has the symptoms or disease, but we don't know yet what is the cause. So the first thing that we can do is we want to know how is the microbial diversity in the leaf. So we will know about what is the cause of the pathogen. And the first step, we use the bacteria. But the bacteria, as we see here, that they doesn't contain any pathogen. So this is not the culprit of the cause of the disease. However, we found a very interesting data where disease have more beneficial than that. So it is very important to isolate that, and then we can formulate it as a biocontrol agent in the future.
Okay, so you found that the bacteria is not the answer of your question. So what's your next move then? to answer your real question.
So yes, actually, we already have more metagenomic analysis, which is from fungi, because of the symptoms of the leaf is actually also similar to fungi. So we already isolate the fungi, and then now we try to postulate coach to see how the symptoms of those fungi that we isolated from the symptomatic leaves into the plant.
Okay, next question. You said that bacterial diversity in symptomatic leaves was higher than in asymptomatic leaves. But from the graph in your poster, it seems like the composition of the bacteria are the same between the symptomatic leaves and the asymptomatic leaves. The one that different is the intensity.
You mean the alpha diversity or
Yes,
beta diversity?
you said that in your conclusion results. I think you mentioned bacterial diversity.
Okay, so the alpha diversity, it is already confirmed that the symptomatic leaves has higher bacterial diversity. Maybe it's because of the specific condition, because of the symptom attack maybe, can also influence the symptomatic leaves. While in the beta diversity, it is because it is unweighted unifractal distance. So it is just reflect what is the type of the bacteria that present. So it's not include the abundance.
So you to show that there is bacterial diversity higher symptomatic leaves, you use the relative abundance. Is that correct?
Actually, in the poster, we show you the alpha diversity, the CHAO1 index, Shannon-1 index, and etc. And it showed that the bar is higher in the symptomatic leaves than asymptomatic. So
Okay.
in significancy, we can see from that.
Okay,
chart.
but in statistical analysis, we cannot say that based on the graph only. It's a descriptive analysis, but you have to show it in statistical analysis, in statistical inference. So what method that you use to say that it's significant?
Yes, we actually agree.
Yeah, also you can say that this one is more diverse, but you don't really have what the variables make you sure that this one is diverse than the other one, because the graph cannot tell the story.
Okay, thank you. So this is also we accept that this is one of the flaw of our study, which is we don't have any statistical data because we only have one metagenomic analysis in each sample, no replication. But the way that we do it is we cut some of the sampling or leaves, and then we randomize, and then we put like a composite of those leaves from different sampling sites, and then we do the analysis from the composite of that samples. So of course that is not accurately present the diversity. So this is because of the this is the preliminary study, and we do that because we want to know what kind of the bacteria here, and how we can do that is we want to isolate the dominant bacteria from here.
Okay.
Yes, please.
But I think you have to mention that in your paper, or poster, if you are not using statistical analysis, that it cannot be generalize the result, right? So
Can I answer to that?
Is it okay?
and you said that there is no statistical analysis there, but I found that in one of your graph in that your poster, there is a graph showing that there is like a star, like red star. Usually it means it is significant. There is a significant difference between the samples. So how do you explain that?
Oh, so you mean like the asterisk? So the asterisk, actually we put that not in the bar, but in the bottom of the dot. That means that those bacteria is actually found abundant in symptomatic leaves. So it's not about the significancy, but it's just a sign for that. For the bacteria is abundant in symptomatic leaves. We make it like a sign like that. And then for the statistical analysis, actually we will know if there is a statistical analysis, there will be like a asterisk or something like that. We don't put anything in our poster or in these slides, right? So, yeah.
Please wait. This is gonna be your last session, so is this gonna be your question? No. All right. Last session. Decide. I'm gonna start the timer now because this is the last session.
Okay. In symptomatically, you do clustering analysis. You make like a dendrogram. What it means? Can you explain about that in your poster? And you say that more symptomatically is more more similar with the same type of symptomatic or asymptomatic, but it shows to me for the asymptomatic, yes, it is similar, but for the symptomatic leaves, for the, I don't know, I think it's pole, it's more similar to the asymptomatic one, if you look at the dendrogram.
So once again, the dendrogram in our poster is the beta diversity. So beta diversity is actually the diagram that see that the bacteria, how is the composition of the bacteria. It's not about the abundance of the bacteria, and it's from the category. Yes, right, that the pole has the same category, both symptomatic and asymptomatic. For the composition of the bacteria, and also tree also have the same composition of the bacteria, but not the abundance.
Yeah. But
Oh, sorry, that was the last question. Do you have any more things to add? You have 20 seconds.
If you are still confused, you can read Yamamoto 2020 for about the microbiome data.
All right, and that is the end of the first round. I am very proud. You keep the zeal. Now we have 5 minutes to kind of rest and prepare, and then we will move on to the next presentation by D38. I think this competition is tighter with a very tight scoring there. Currently D49 holds 14 and D38 holds 10 points. Congratulations. I just want a note. I noticed that D49 got 5 points just from the presentation, so you know you could catch up by that also. Right. Apa? 3 per judge.
Oh, I see.
Yeah, 3 per judge. Sorry. Okay, I will be more clear next time.
okay.
Up to 3 per judge. So you guys have improved a lot since last practice session. All of you, I mean, like, even your materials are much more detailed, and a lot of the discussions are much better. That was really good. Toilet break? Anyone? Huh? It's okay? Toilet break? Alright. 4 points difference.
Ini tuh break ya?
Dari green nggak sih?
Iya.
First round.
The other sides are also on this stage of the tournament. Okay, 1 minute. Very exciting. All right, 1 minute. Do you want to prepare there or? Up to you. Okay, up to you. Alright, since we're gonna start at 30 seconds, I'm just gonna say welcome to the second round between D49 and D38. The first round was already finished with D49 defending and D38 attacking. In 20 seconds, whenever you start speaking, I will start the timer for your presentation, and then we can start the second round. Whenever you start, I start the second.
So we're going to present that the impact of maternal SARS-CoV-2 infection on pregnancy outcomes. Next, so because the coronavirus or COVID-19 result impacted more on vulnerable population or pregnant women, that's why this study is conducted. We enroll pregnant women at the first trimester, and we take blood draw twice. The first blood draw was taken at the first trimester, and the second blood draw was taken at the third trimester. For the lab, we do the hemagglutination assay to detect the antibody, and we scale the result from 0 to 3, where 0 is being the negative result. Next. So for the logistic regression, we see that low birth weight was the most impacted clinical pregnancy outcomes impacted by the COVID-19 infection, where the odds ratio increased by 23%, also combined with other variables impacting like the maternal age, sex of the baby, nulliparous, and hypertension of the pregnancy. Next. Also, we found that 50% of our population of pregnant women lost their antibody at 143 days, faster than the general population, where the age of the mother or the maternal age impacted more, where mother with older age tend to lose faster than the younger pregnant woman. I think that's all. Thank you.
Very good. 1 minute and 45 seconds. All right, we will start with the question and answer. When you ask your first question, I will start the timer.
Okay. My first question is, how much is your sample size and how do you calculate the sample size?
Okay, for the sample size, we have 956 for the antibody in COVID-19, and then
956.
yeah, 956, and then we for the calculation, it's from the sample for this, because this is a observational. We don't have like a control or control and case study. It is observational, so we calculate it by the there is actually there is a formula that is z of 1.9 z of 95 confidence interval times p times q per d square.
Stop. All right.
That is
On time.
Actually, that's what we are asking for. We are asking for the eligibility criteria of your study, because you said you only took from the first 3 semester pregnant women, and how do you justify that?
Oh yeah, for the eligibility, actually, we enroll all all ages of pregnant women at a specified like what healthcare we work with, and the reason why we took at that place is because the index human development index is very low at that place based on release of health profiles Kemenkes in 2022.
And for the trimester, we calculated by the gestational age.
Okay, thank you. I see that you do this in several days. So how much percentage of missing do you calculate it, and how do you handle that loss of the participants?
Okay, for the missing value, it is for this one, we have at least I think the most missing value is the MUAC and the mid upper arm circumference. It is around more than 10%, but then it's because it include that in the analysis. So it's only contain not non-missing values.
Okay, so missing data right from that, and I think we have another question. So
you said that you are having the participants. How do you confirm that the participants participated from the beginning to the end of your study?
Yeah, so we have so we have that informed consent that they need to sign, and we came to their house to make sure we explain what they need to get, what will they get, what will they need to do the whole study, and explain them. After that, they will need to sign with one another family member at the witness.
No, I mean, you said that your study is held from the January a whole year, and you took several samples from each month. So how do you confirm your participants is stable attending your study?
Oh, we're not collecting the base blood samples each month. It's only 2 times, the baseline and end line. That's how we collect the sample.
Yeah, we mentioned it like we took the blood draw twice, the baseline at the first trimester and the end line at the third trimester.
Okay, but I see in your poster you showed the graph with the green line and several colors of bars.
Okay, so because the enrollment is not one time, it's every time. So one mother came, they will get an ID, so they will not enroll again in the next enrollment, you know. So if one mother enrolled, we identified it as enrolled, and then we enroll another woman in other area, or because mother will. Okay. Yeah.
Oh, okay. Please.
okay, so about the HAT score, do you have any references about that for the qualitative score for HAT score? I see in your poster, and is that reliable?
For the HAT assay, we only do like the scale, qualitative scale. It's we're still there's another step actually we do with this study where we measure it quantitatively, but for this one we do it like with a scale from 0 to 3 as qualitative score. Yeah, it's reliable because we're using HAT because HAT as far as I know from I forget Galileo in 2020 that it's the most with the highest accurate with the lower price because we're conducting our study in a low resource place in Lombok. We use that specified assays.
All right, so this is a very tight competition, and this is the last session. So make your last question count.
Okay, so beside the antibody drop, do you also assess the physical condition, and do you have any external assessor for that kind of the condition? And is that reliable assessor? Or?
Okay. Okay. Yes, we look at the physical assessment of the mothers, but we're not included here. And the one that do that, we have a healthcare worker in our as our staff. So it's, and we do training before we do the study.
Yeah, and they have like a minimum score before they're going to the field. They cannot just go without any training in advance.
You have 20 seconds. Do you want to add your answer?
And also we collaborate with the midwives in each of the health care center. Yeah, we make sure that they have like a better treatment for their symptoms.
All right. Thank you. And that is the end of the second round. Have all the judges put in their scores? Okay, that means we have a tie. So in the case of a tie, we have overtime. Basically, it's 2 minutes of each side. So that means your side will get to ask questions again for 2 minutes, and then the other way around. So it's basically an extra time of 4 minutes. So that we have a 1 minute time for preparation for this extra time. This is really tight. This is actually exciting. I am amazed. Some input. I think both of you can be more elaborate on your questions. Like, try to ask questions and add comments for that, like the previous match. Huh? Yeah, because then it will be more specific on the discussion. Alright. We have 30 seconds, and then we begin by questions from D38. D38, are you ready? Okay. D49, you ready? Alright, please.
So, in one of your graph, in your poster, in the clustering, in the dendrogram, there is a distance in the below the dendrogram. How to read that?
Okay, that is the phylogenetic distance. So it's like a distance how close the composition each other from the sample to the one sample. So it can indicate how similar the composition of the structure of your bacteria from one sample to the other sample. So this is like the distance, how close they are.
Okay. Can we ask? In the explanation, you explain symptomatic more similar with the symptomatic group, and then asymptomatic more similar with the asymptomatic group. But if you look at the distance, the distance between how can you correlate with the distance? Because to explain about the similarity of the sample, we use the distance, right?
Mm-hmm. Yeah.
Yeah, but if you look at the asymptomatic, the upper one, and then the second row, it's more close to the bottom one than the upper one. If we look at the distance. So.
Actually, we did weighted unifrac distance also, but we didn't input it in the poster. But I can say that if we see the branches of the asymptomatic L2 to the SL2, IL1, and SL1 is divided by 2 categories only. So as IL1 is significantly different between the others. So that, I think that's answering your question.
Yeah, you answered the question, but in the poster, it says different, I think.
What does it say in the poster?
It says symptomatic, more similar with symptomatic, and then asymptomatic with asymptomatic.
I think Okay, so I would like our poster is the asymptomatic in the poll category is almost similar to the symptomatic in the poll category, and also in the tree category is also close to the tree category. That's what I think that's what on our poster.
Yeah, sorry. Yeah. Okay, now it's your turn for questions. I will start timer when you ask questions. Okay, ready? Ready? Please.
I see in your poster that in the figure T you did not have any explanation for the green line. First, that's your flow. And the second, it is very confusing since the bar is too much color and you don't have any statistical significance. First. And then the question is, in that term, can you generalize the result of your study?
Okay, first I want to answer about the statistical analysis in the graph. So it's not about statistical analysis. We only want to show the timeline of our study for each ID, for each mother. So that's why there's no statistical analysis there. And then
because we want to show that our study is happened during the Delta wave, where it's really important because Delta wave is known like where we haven't get like vaccinated, but so that our data is really important.
And also for the line, sorry, yeah, that's we don't add the explanation of the line. We will add that later. Thanks for the reminder. So the line means the dark, the red one means it is infected. And it shows that.
Okay, do you do some statistical analysis, and what did you use, and then why you use that, and is that make your data become confident and reliable?
Okay, to answer our research question, we do a statistical analysis. First, we do logistic regression to look at the impact of infection on the birth weight, on the birth outcomes. We use logistic regression. So the Y, the response variable, is the low birth weight or not, and then the predictors. One of the predictors is infected or not, and then the other one is MUAC, hypertension, and so on. That is the first statistical analysis. The second one is the Cox regression. We look at the event of loss of antibody during a period of time. That is to answer how the antibody of COVID-19 works in mothers, pregnant mothers. Sorry.
Last question.
Okay, so I would like to say that this is about the COVID-19 on the pregnant mother. But so I think that like do you have any similar like study on your case, and how is the result and how you collaborate that into your result? Okay, so do you have any?
Okay, so this is.
Is that your last question? You still have 30 seconds if you want to.
Okay, I want to add what the real implication of your study regarding the whole population of the pregnant women in COVID-19.
Okay, so the first question is.
So you can ask like 2 questions.
It's kind of related though.
Okay.
Because but previous studies.
Oh, for the study, this is the in our study. This is the beginning of our study, and then after we got the conclusion here, we continue with another study to look at the impact of vaccination on pregnant women.
Also, I want to add more, like from study of Gaffery et al. In 2020. They mentioned that they instead of using PCR, we know that PCR is the gold standard for COVID-19. Instead, they use like antibody, like neutralizing antibody is needed for like we want to know also the past infection instead of the recent or the current infection. We also want to know the past infection, so we will know that the pregnant woman will have like antibody that is important for us for a pregnant woman to protect them throughout the pregnancy.
All right, thank you very much. That is the end. Okay, and again, please come forward, both teams. I think both did really great. Shake. Shake. Don't worry. This is a double elimination, so there's another one, and all the judges are fixed. What was the score? It disappeared.
D38 27, D49 28.
Oh dear, there's just one. Please shake, please.
Thank you so much.
Okay. Thank you. Double elimination. Yeah. There's one more. There's one more. Okay, thank you. Picture. Once you document it, very fun.
Okay, 1, 2, 3. 1, 2, 3. 1, 2, 3.
Another one. Okay. Thank you. Good job. All right, next.
So D49 will move on to match 19 in Arena 1. While D38 will move on to match 26 around 2 p.m.
Okay, and then
Next up is
for the scoring ini D05 kiri atau kanan? Coba lihat scoringnya. Kanan.
So next up is D05 versus D51.
D05. Gimana Pak? Oke.
Cek.
Oke. Oh yes, please. Tuh, mau nunggu skornya biar pas mana yang kanan kiri ya.
Check 1, 2, 3.
05 sini, oke. D51 sini. Alright, oke.
Matching kita.
Alright. Oh my. This is an exciting one. Okay, so as usual, please introduce yourself and your team first, and then we'll continue with the coin toss.
Okay. Hi everyone. My name is Corey, and this is my partner Sri. We're from Team D05. We're from the Eichmann Research Center for Molecular Biology, Brin.
Hello everyone. My name is Christy, and this is my partner Aulia. We are from Universitas Gadjah Mada and Dr. Sarjito Hospital. Thank you.
Thank you. Fantastic. All right. Coin toss. Who picks head or numbers? Huh? Heads. All right. Okay. Heads. So you got numbers. It's numbers. So do you want to present first, or do you want to let them present?
Yeah.
Present first.
Yeah.
Okay. All right. Please prepare about 1 minute, and then whenever you start speaking, I will start the timer. Judges ready? All right. Thank you very much, judges. That was very intense. I think we're doing a good job. Okay. Whenever you start speaking, I will start.
So our study is about exome sequencing, facilitates personalized treatments for encephalopathy patients, transforming national clinical practice guidelines in Indonesia. So DE is the most severe group of epilepsy characterized by severe seizures and cognitive and behavioral impairments. By understanding the gene underlying mechanism and inheritance of seizure disorders, we can transform the clinical diagnosis and treatment. Currently in Indonesia, the guideline is using empiric therapy based on the type of seizure, in which may have the contradictions of underlying mechanism. For example, we have the focal seizures, but it's actually the natrium channel blocker. The study aims to look at the genetic profile of the patients, directing patients' therapy management in Indonesia. Next slide. Our study was conducted in Sarjito and UGM Academic Hospital. The DE patients was phenotype and do the whole exome sequencing, and then we can determine the genetic etiology of the patients and conducted the personalized treatment for the patients. Next. This is we've identified the genetic etiology in 20 out of 47% patients. And the most common genetic etiology was SCN1A mutations, which is responsible for Dravet syndrome. And management changes was conducted in 9 patients with 7 drug changes and 2 avoidance of brain surgery. Next slide. So this study illustrates that whole exome sequencing in clinical setting in Indonesia is feasible, has high diagnostic and clinical utility, and shed light on the possibility of using exome sequencing to modify the treatment for the patients in clinical setting. Thank you.
Fantastic, very good. All right, to begin, again, every question or every answer is limited to 1 minute. Total session of 6 minutes. I will start the timer whenever you start asking.
Okay, thank you for your team presentation. So I'm just gonna talk about the technology itself. So whole exome sequencing is a relatively new technology, right? So have you validated your approach? And then I think I've Googled some reports on whole exome sequencing, and they said that this method is not validated for identifying structural variations in the genome. So, but you said that copy number variance is identified in your study. How you justify your findings? Thank you.
Yes. So, you are right. So actually we justify with the next chromosomal microarray to justify the copy number variant.
Is that your answer?
Yeah.
Okay, let's move to the next. Okay, please.
So do you put in the inclusion criteria? So do you have any exclusion criteria? And do you have like age limit for the? Do you test it only the patients, or you also compare their genomic data with the siblings or any other members of the family? Because it might reveal more background on the family itself genetically.
Yes. So if we find the variant of unknown significance, we will do the segregation analysis by detecting the parents, and then we can know whether that is our gene or not.
Please.
Thank you. It's very interesting studies, and I'm really curious about what is the rationale behind the sample sources, because here you use whole blood serums from blood, right? What is the rationale behind it? Because how it can be representative for what happened in the brain and then what you detected in the blood? Because here we have the complexity that we have. Brain is very complex because they have blood-brain barrier, and how we can tackle this issue. I mean, how it's represent what happened in the blood represent what happened in the brain.
Yeah, because DNA is represents in all cells of our body, and this is a genetic disorder, so we can isolate DNA from everywhere in our body. It's not the protein expression, it's the DNA data.
You want to add? We have 20 seconds. All right, okay. You want to add or clarify, please?
And also, since you target that it will be for a personalized medicine, right? So what we see from your studies is happen with the genetics causes, but how about the non-genetic causes? I mean, how we will have the guidelines for that? Because you have only 47% of genetic causes in your cases, but how we can etiologies?
Yeah, so this DEE is a Mendelian genetic disorder. So when we don't find the other than 47%, it's actually because of the machine, the technology cannot identify it. It's not caused by another genetic disease, another non-genetic issues. Is that enough?
It's up to you.
Yeah.
All right.
Okay.
If you want to clarify, if you want to chase, you should, chase down.
So you're confirmed that it's only genetic related. It has nothing to do with infections or like
yes,
other pathogens.
Yes.
Okay.
Because we exclude the infections by the pediatric neurologist, so we already have expertise on that matters.
So that will be your exclusion criteria,
Yes.
then, which is not mentioned. here.
Yes.
Yeah.
Thank you for
Yeah.
reminding us.
So you claim in your title that it's going to change the national management of this kind of patients. How practical do you think the real life application it is? Because the technology isn't cheap, I guess.
Yes, that is not cheap, but it is affordable. It is 5 million rupiahs, and if you you do the brain surgery, it costs like 100 million. Okay, so it is not cheap, but it is cost effective.
Okay, continue the question about that because it will be like a targeted therapy. But can you analyze from this data if there is any different treatment responses that can be appears from these genetic variations? I mean, if you can use maybe there's some genes that responds better with the treatment, but the other genes is not. Maybe we still can use drugs for the specific genes, but not in the other genes.
Could you rephrase?
Yeah.
Sorry.
So what if you can clarify the different treatment responses based on your findings, if there is better response to therapy and some are not a good response, have a better response to therapies. From the findings, if you can analyze the different treatment responses, because you have a variety of genes there, right? If each genes has a different treatment responses for our already available drugs. Yeah. Yes. Okay.
Yes. The patient first the therapy with carbamazepine, and after we know that this is the genetic is SEN1A, we therapy with the diet. Okay.
So SCN1A is a natrium channel blocker, but carbamazepine is also a natrium channel blocker. So it make the things worse. So we should stop the carbamazepine and change to another drugs. So that's the point, to know the genetic. But actually with the time, actually if there's the disease itself is focal, is in current guideline, it's use carbamazepine, current guideline. But when we check it's SCN1A, then we should stop the carbamazepine because it makes things worse.
Last question, last session.
Okay. I'm just continuing. So now I assume this is like a preliminary study. Would you increase more samples, I think? And then, like, if you have already identified the SCN1A gene as one of the most prominent gene for this disease, wouldn't it be better for your screening of patients to design like a rapid detection, whole exome sequencing?
No, I don't think that's a good idea because the DE is a very disease. It has more than 400, and if we do the targeted, we close the opportunity to find novel genes because this very complex mechanism underlying the DEE. Thank you.
Do you want to add anything? It's 20 more seconds. Nothing?
No.
All right. Okay. Thank you very much. That was the end of the first round. It was really great. So as usual, we have a 5-minute break, and then we continue with the presentation from Group D05. Remember, the presentation itself can be graded up to 3 per. Yeah. Okay. All right. Okay. I need some toilet break, so please give me a moment.
Kobe dulu. Sama-sama di Kobe. Iya, iya.
Kita banyak kenal sama-sama Unhas juga sih.
Oh gitu ya.
Banyak banyak sama juga alumni Unhas soalnya.
Oh, terus sekarang di BRIN.
Iya.
Keren ya.
Jadi datang ke dojo dari Jogja. Kalau ya. Dikasih ongkos.
Kasih ongkos sih, gratis gitu.
Okay, 2 more minutes. You wanna prepare? Open the slide, anything? Ngomongin price aja ngeliatin saya. Ah, Dr. Kristi. Price tadi. It is price sih. Terus saya dilihat. Parah banget. Parah banget. Inside joke-nya parah banget nih kak. Alright. We're gonna start the second round. You just ready? Alright. Again, the second round will start by a 2-minute presentation, up to 2 minutes presentation by Team DO5, and then continues to the question and answer up to 6 minutes, with a limit of 1 minute per question and per answer. Whenever you start speaking, I will start the timer.
Thank you. I'm here to explain to you our study about geospatial approach for hepatitis B virus in Indonesia. Next, please. So why hepatitis B? Because chronic hepatitis B virus infection is the major risk factor for liver cancer, and the genotype of HBV vary across the different geographical areas. And these HBV genotypes are associated with distinct clinical implications. Next, please. Due to this background, then we perform a geospatial approach to analyze the regional molecular epidemiology of HBV genotypes. We use clinical isolates from 2 referral centers, one at Jakarta and one in South Sulawesi, and then we perform the HBV genotype profilings, and we perform the geospatial analysis based on that. Next, please. As you can see here, these results, from the 2 referral centers, we don't find any significant differences for sex and age. Meanwhile, for the subtype, we found ADW as the dominant subtype in Jakarta, and ADR for South Sulawesi. And as you can see, the genotype distribution, red for B and blue for B and red for C. And from the hotspot analysis, it reveals that North Jakarta and also Makassar is the hotspot city for the both genotypes. I think that's all from our team. Thank you.
Thank you. Please return. 1 minute 40 seconds is pretty good. Okay, so now we continue to the question and answer from D51. Whenever you ask your first question, I will start the timer.
Thank you. My first question is how and where you collect the sample?
Thank you for the question. So I think Sri mentioned a bit, but we collected samples from 2 referral centers, one in Jakarta and one in Unhas, a place where our collaborator is. So we recruit the patients during 6-month period in 2018. And then we have the inclusion criteria that all the patients have to be adult, and the exclusion criteria and the hepatitis B infection is confirmed by the HBsAg markers, and then the exclusion criteria is any patients that have co-infections or any comorbidities. So we are convinced that all the any findings that we have is true from hepatitis B.
Your title is East and West. Do you think your referral centers already representing the East and West sections of Indonesia? How about the Papua? I think the Papua has high HIV and also HBV. populations? Thank you.
Yes, that's also our limitation on the studies because we acknowledge that it might not represent the whole East and West. That's why we didn't claim, we only claim this is a representative. Why we chose Jakarta, because of course it's the most populated cities in the west part, and Makassar, even though it's like you say, Papua has higher hepatitis B and HIV incidence, but the most referral centers is actually located in Makassar. So that's why we chose that as our study site. Thank you.
I see you have a different sample. In Jakarta, you have 192, and in Sulawesi, you have 82. How do you calculate and compare? Because it's a very big different sample.
Okay, so the sampling is a total enrollment cases that we have because we have like almost 1 year of enrollments, 6 months for enrollments, and then we. Of course, it will be different for sure because the two cities has different density of populations. For Jakarta, we have 16,000 people per kilometer square. Meanwhile, in Makassar, we have 200 people per kilometer square. So it can be different.
So, do you think because the health system management is different from with Jakarta and Makassar, do you think it's only the iceberg of the whole population with HBV?
Okay, thank you so much for the questions. And we like the question because actually why we want to represent it as this data, because we want people to question it's why we have the hotspot here and here. But as you can see in the HBV genotype analysis in the here part, and with the statistical significance that we have, it's clearly shown that the two difference proportion happens here and is statistically significant about the difference. As you can see that the Jakarta have the B, more dominant of B genotype and C genotype for South Sulawesi.
Okay, so you mentioned that even not representing all the population, but you can find the differences between Jakarta and Sulawesi. And so, can you explain what is the background, why there is differences in the genotype?
So a lot of things can affect the genotypes, the genotype prevalence in each geographical regions. First, the urbanization, because people migrate. Also, Jakarta has a higher migration compared to Makassar, of course, because you have international airport also from everywhere here. The other thing is cultural differences can be affected because, for example, some in Makassar I can compare, there is still hypothetical, but they have a practice of tattooing. Tattooing is very different. You can, because hepatitis B is a bloodborne diseases, so it can be also affected. Socioeconomy may be different also in both cities, so it can be many things can affect it. And we want these studies triggers another study that will complete this data.
I mean, I meant that why there is a difference in ADR subtype much more in Sulawesi than in Jakarta.
I'm going to add more to what Sri has explained before. So our previous study has characterized the availability of the prevalence of different genotypes in Indonesia, and our previous study has shown that in the western part, so from Sumatra, Jawa, and part of the Nusa Tenggara, is mostly genotype B with serotype ADW, and for the eastern part is also genotype C with ADR. So we think it is so hepatitis B clinical manifestation is really related with the host immune response. So we think that there's also the contributions of different ethnicity background in these different locations that are actually preferring which genotype is more predominant in this certain area.
Yeah. Do you think the migration of the population play a role in these differences of genotype, the B and C genotype between Jakarta and the east part of Indonesia.
Okay, there is no evidence yet for Jakarta and Makassar for specifically, but in the studies, in other studies, it's already proven that migrations can affect because it's blood-borne disease. So people who carry the HBV from everywhere in the town can be transmitted the disease. So of course it can be affected.
So what is the countries outside Indonesia which has dominantly B and which has dominantly C?
So yeah, so if you look at the worldwide data, we can see that certain genotype only exists in certain areas. So for Asia, Southeast Asia, East Asia, usually dominated by B and C. So that's pretty typical for Asian countries. I think it's based on our genetic background. For Americas, there's are more variety because I think there's a lot of migraines there, so we see a lot of genotype A, B, C, and D. And for the Middle East, usually genotype E. So that's why I think because it's as I mentioned before, it's closely related to the host genetics. So that's why I think we can see that certain distribution.
You mentioned the host immune problem. So I think the HIV carrier and data should be I think it's yeah.
Sorry, you want last additions or any last comments, questions? Because you have 40 seconds.
Yes, you say in the background, but hepatitis C has a worse prognosis, and your conclusion is for management and improve management and treatment. So I think it's good if you input like the clinic patient, like the hepatocellular carcinoma patient for this study. Okay.
Yeah, thank you for that. So this is only Hep data because we exclude any other infection by any other viral infection. So Hep C, patients with Hep C, patients with HIV positive are excluded from this data. Okay, and regarding for the clinical treatment, of course, there's our main goal to improve the overall management and therapy. But this is a cross-sectional study, so we only recruited patients with confirmed hepatitis B infection with no treatment. So I think if there's a follow-up study, we can also expand more for the cirrhosis, HBV-related cirrhosis or hepatitis cellular carcinoma, but there will be, it's not in the scope of our current study. Thank you.
Thank you very much. That was the end of the second round. Okay, please come forward. Judges, finish everything you put up? All right. Please, shake. And so congratulations to Team DO5, which is the champion for this part. But don't worry, again, double elimination. Team B51 is another one. So, Raka.
Yes.
Picture. Oh, there's another one. Thank you. All right.
D51 will be moving to match 15 in Arena 2, while D05 will still be in this arena, but after the lunch break.
D05.
The next match is between D13 and D32, but I've been informed that D32 is absent. So by
Win
default,
by default. Jadi itu tadi D32. Oh D13 tadi already. D32-nya absent ya.
D32 is not here.
But I start the game first.
Okay. Oh, so this is your second match?
Yes.
Oh. And so, okay, you won by default.
So D13 will move to match 22 in Arena 1 after the lunch break.
After lunch break.
We'll be waiting for the other arenas to finish. But for now, this arena can take a break for 10 minutes for
Yay.
snack break and toilet break.
Thank you. Bisa win by default gitu. Tapi kalau kan itu nggak ada ya, Mas?
Enggak, ini aku
Enggak, biasanya kalau laptop di rumah nyari. Tapi masalah koneksi kan.
Makanya harus buka dealer.
Berapa skornya? Berapa, Cek?
Nanti akan lawan di
Gitu, kenapa English?
How's it going? It's going okay? Thanks so much. Okay, thank you. How's it going?
one time.
Yeah, that's good. That's very interesting. I think that we should do this.
I think so. But I think the practice section helped them get the.
Saya terjebak tadi ya.
Yeah.
Saya
Because
terjebak
then
tadi.
they become more better formed. Yeah, he's single fighter.
Single fighter. All right. Okay, great.
Ini kita break ya? Break ya?
We had a tie also.
10 menit.
So I went to overtime.
Oh.
And then it was broken in overtime.
Nice.
Yeah. It was the SID versus the. It was Debbie. Febby, yeah. Febby and. It was the three one, the three infections.
Oh,
Yeah, both of them really know their
So,
stuff,
so
man.
who won finally?
The three infections. By 1 point.
We had several that were 1 point.
It's crazy.
Tiba-tiba turun ya.
Oh, berarti dua-duanya nggak penalti. Jadi ini babak kedua ini tadi kalah. That's a problem.
Lihat match berikutnya.
Padahal selama
Itu pun gara-gara
Oke. Alright. Yuk, semangat.
Tadi nomor kelompoknya di situ. Oh iya iya betul. Iya sampai majalah Tempo. Padahal mana mana benar. Kata itu sama. Iya. Iya iya.
All right, let's go. Let's start. Last match for this set. Mana itu? Where's your apa namanya? Where's your number? Itu dia.
47 hilang.
Hah? 47 hilang?
Ya udah biarin nggak apa-apa.
No, it has to be over there, right?
Oh. Soalnya kita nggak dikasih kan tadi.
Oh gitu. Berarti masih di sana dong? Hah? Enggak? Oh ya udah, oke lah.
Mau saya bikin yang sini jadi 47.
Bah, nggak usah lah.
Nggak usah.
Tapi di itunya udah ada kan? Di scoringnya sisi mana? Scoringnya coba. D33 di mana? D47 di mana? Apa? Ini satu match lagi tapi telat ya. Aku match berapa nih sekarang?
Match 16.
Oh.
Kita kayaknya udah kalah.
Coba, ada nggak?
Ada nggak? Ini nggak ada. 47.
47 duluan.
Enggak maksudnya. Ini ada, ini ketemu.
Di sebelah ternyata.
Enggak maksudnya.
Ketutupan.
Udah ada belum? 47 sama 33 presentasinya. Ini belum ada. Nah lu. Tadi presentasi
Tadi
pakai apa?
lagi di sana.
Oh itu dia tuh di bawah. Dojo presentasi. Beda dia.
Dia.
Oh.
D47-nya di belakang, baik.
Alright. Mike's ready. Alright. Okay. So thank you. Judges ready. Alright. So this is second match. So this is your elimination match, yeah? This is first last. Alright, okay. So this is an elimination match between D33 and D47. Oh yeah, the score is okay. Switch D33, D47 because of the score.
Oh.
Okay. Please. Alright, so we will start by introductions. Please introduce your name and your background, where you're from, and then we will continue to the coin toss to decide who can decide who presents first. Please, D33 with your introduction.
Okay, good afternoon. My name is Yusrita Froji, and I'm from Kirei Imeri. Thank you very much. Nice to meet you all.
Please, D47.
Good afternoon. My name is Gabriela, and this is my team, Sri. We are from Brain. Nice to meet you.
All right, thank you very much. Now we're gonna do a coin toss. Who wants heads?
We had.
Heads. All right. Okay. It's numbers, so you decide whether you present first or they present first.
Okay, I will present first.
You'll present first. All right. Please prepare about 1 minute, and then I will start the timer when you start speaking. Judges ready? Okay. So just a reminder that in your presentation, you can be graded up to 3 per judges. So this is a good opportunity to bump up your score.
How many?
2 minutes.
2 minutes. Okay.
I will start when you start speaking.
Okay, I can start. So today, this afternoon, I will present to you the impact of PCV13 to the serotype distribution and antimicrobial profile of Streptococcus pneumoniae, because we are now implementing the PCV13 since October 2022. Next. So for the background, we have been implementing actually the PCV13 in Indonesia since October 2023, but there is no surveillance from the invasive cases and also this community for these cases, for these implementations. Therefore, we try to compare before and after vaccination with the secondary data, because we are now ongoing study with the previous pre-vaccinations data. So we collected 15,000 whole genome data from the pathogen watch, and we did the screening for the serotypes, for the sequence type and also antimicrobial resistance, and we use 5,000 genome for analyzing this one. Next one. And our finding, we observed there is declining of the PCV vaccine serotypes and also the antimicrobial resistance profile. So it's less resistant now. And we also find that the multi-drug resistant strain of Streptococcus pneumoniae decreasing after the vaccine introductions. Thank you.
All right, thank you. That was it. That was very good. minute and 20 seconds. So now we will begin the question and answer. 1 minute per question and per answer, to a total of one session of 6 minutes. I will start the timer when you start asking questions.
As you mentioned that this is already started at October 23.
Yep.
Maybe, I don't know, if there is another vaccine that already implemented before the PVC one that has the same function. So what's the difference with the PVC 13?
Is it in Indonesia or worldwide?
Yeah, because it's in Indonesia.
No, we don't have any vaccine implemented for pneumococcus so far. We just started.
This is the novelty that you are doing in your research.
Yeah, that's right. In Indonesia.
From many kind of vaccines, like from the bacteria, like what is the name of the bacteria? Sorry?
Streptococcus pneumoniae, if you can
from
find.
the Streptococcus pneumoniae, why you choose the PCV13 instead of others vaccine? Is there any others vaccine that related to the serotypes or?
Yeah, so this PCV13 contains 13 serotypes, and it is widely used worldwide now. So it's better to use these vaccines as our vector for the comparisons because it's widely used.
Oh, okay. So how about what is the name of the bacteria? Sorry.
Streptococcus pneumoniae.
Streptococcus pneumoniae in Indonesia itself. Do you already study the differentiate between the pneumonia in other country that you use as your samples in Indonesia? Like how is the related impact? Like let's say that in your data that the PCV have a great impact, like for the antimicrobial resistance and also for the serotype of the bacteria. How it's you can make sure that it is can be related to the pneumonia in Indonesia?
Okay, so in Indonesia we only have pre-vaccinations because we don't have any pneumococcal vaccine implementation yet until now. So I only have data for pre-vaccinations, and there is no any analysis for post-vaccinations because we just implemented the vaccine.
How about the diversity of the bacteria in Indonesia? Is it do you already study about it?
Yeah.
Like is it same with the other country that they use here?
Yeah, what I can say, before vaccination, we still have PCV13 types circulating in our community and also in our patients infected with this bacteria.
Do you have?
I think I saw that you extract like data from other countries. You extract data from other countries too. There's from Brazil or something, right?
Right. That's right.
So what the baseline of the data that you are implementing with the ongoing program right now?
So with this analysis, we can give a big picture to government that the PCV13 has impact for Streptococcus pneumoniae, for serotype, and also antimicrobial resistance, because we are now implementing the vaccine, right? So it's like preliminary information to the government. Yeah.
So what's the goal of even like now you want to suggest that this is the primary data. So what will the government do with that in your perspective from the conclusion of your data?
So we can see that there is increasing of certain vaccine serotype after the PCV introductions, and the emergence of non-vaccine serotype not included in PCV13, emerge after the PCV introduction. So it will be a warning for the government that we need to also pay attention on these serotypes. Yeah.
So you mean after you get this data, so you will suggest that the vaccine PVC13 is really need to be used in Indonesia, like something like that?
Yeah, because it reduces the prevalence of incidents, pneumococcal infections.
So you will suggest that the government should buy the vaccine, or you will recommend to Indonesia can produce the vaccine itself in?
It's already implemented in Indonesia as national vaccine program.
Oh.
Yeah.
So would you mind to explain us about this result, the before and the after one?
What do you want to explain? The explanation?
Like the impact from the, yeah, it's too small so we can see. Yeah, what the difference with the before and after?
This one is the name of antibiotics, and we can see that the prevalence is reducing. Okay, and this one, the MDRSP, the multi-drug resistant strain, it's also reducing.
What kind of antibiotics that that has a small impact to this bacteria?
Small impact, what do you mean with small impact?
Like I mean like after, before and after using the VCV, which one of the apa tadi? Antibiotics that like not have a significant impact with this vaccine?
Most of the vaccine has impact, but I think I can say that this one, linezolid, has less impact with this. Like most of the antibiotic will be less resistance after the PCV introductions.
Oh, so in this case,
This is gonna be your last.
okay. So it means that the antibiotics, you can also suggest what kind of antibiotics that can be used to.
I cannot suggest that because the antibiotic treatment should be delivered by the clinicians, not by the
I
researcher.
meant by your research, can we just take a
We just inform
conclusion?
that this one is the antimicrobial resistant profiles so far, but that's it. The decision, what kind of antibiotic that they can use, it's the clinicians.
Oh, okay.
Right.
Okay, thank you, and then
Yeah.
it's the end of first round.
Thank you.
Okay, so now we have a 4 or 5 minutes break to prepare for a group to present, and then the questions from this side. Okay, I will start with
newbie jadi bingung nanyanya juga.
Saya belum baca tapi.
Nggak apa-apa. Maksudnya kita
tadi belum baca.
Kita belum baca video.
Tadi udah akan.
Ini kita udah kalah dari ronde pertama. Tenang mas.
Saya kalah 0,6.
Nah, kan itu masih
Sabar ya mas.
Gara-gara
apa? Kelamaan presentasi.
apa tadi ya?
makanya tadi pastinya dicepetin ya, maksudnya nggak sampai 2 menit masa udah selesai result gitu aja.
Kami jadi cepet ya, saya nggak tahu.
Mas sudah lama jadi peneliti ya?
Masa yang S2 di itu IMEI deh, tadi ngomongnya dari IMEI ya? Dari IMEI mas.
Oh, okay. Not both of you? Alright. Okay, so. Right. Now is the second round where we'll begin by the presentation by Group D47 and then the questions given by Group B33. Time for the presentation is 2 minutes. I will start the timer when you start speaking.
Okay, good afternoon everyone. This is our team's theme, sequence homology-based enzyme detection of plastic degrading gene using PCR approach from enrichment of plastic contaminated soil in Galuga, Bogor and Cipecang, south of Tangerang landfill. So basically, why the background is this: we want to screen and find and produce plastic degrading enzyme from new resources from to environmental sustainability solution for plastic degrading, especially from our local environment, from Indonesia, because the finding of Pycninia bacteriocinaensis that can produce PETase and MHTase that can degrading PET. Next. So this is what we do. We went to the landfill, sampling the soil, and then we went to the lab to culture the samples with minimum media with PET film. After we culture it, the piloting, we extract the genome and then amplify the targeted genes, which is PETase and MHTase with this primer. And then after we get the amplified gene, we sequence it and verify it in the gene bank. And the verified sample, we send it to analyze the 16S metagenomic sequencing. Next. So this is our result. That is the gene that we successfully amplified, the PETase and MHT. And the one with the flow sequence is MHTase from Galuga. It is near to, after the sequencing, we can say that it is from bacteria called Diphtheria staphylococcus, and with GE PETase, which is only a partial gene that we successfully amplified. It is from Acidophorus diffaldi. And it is verified with the 16S metagenomics. Thank you.
Okay, very good. All right, now let's continue to the question and answer. When you start asking, I will start the timer.
Okay, thank you very much. It's very interesting, actually, the research. First thing that I want to know, why you use the broth media to isolate this bacteria or to do the enrichment, because you will amplify a lot of bacteria with this media.
Okay, actually the media that we use here is the minimum media, so there is no like carbon inside there. So we change the carbon with the plastic itself. So the main resource of the carbon will be the PET plastic. So we just like enrich the potential of the bacteria that can live with the PET plastic only as the main source of carbon. And after that, we will get the several potential one to get the genome from the enrichment culture.
Okay, if you are expecting that, why you found many various bacteria in your 16S metagenome?
Yeah, perhaps it can be concluded that the bacteria we found here, there are many bacteria that actually could be live with the carbon source of PET. And if you can see here in our phylogenetic tree, the bacteria here actually from our sequence, what we get, it actually come from the same group of proteins with the same function. Like it means that the bacteria, and from the NGS result, it showed that the from the G2 metase is show that the Diphtheria surhatensis that our suspect bacteria gene come from is the Comamodaceae, which is the most abundant in the culture NGS.
Yeah, because I'm talking that since you have the target bacteria, why don't you use the specific media for that?
That's the specific media that we use, the same media like the journal says that how they can find the Idoneella sakaiensis.
Okay, so you expect all of these bacteria contain these genes, the GMSase and GPTase?
Yeah, actually, they can live with the plastic as the main carbon, but you know that many enzymes can be related to degrading plastic, but we will specifically try to search for the specific metase and pitase. So we can say that the bacteria that can live in the culture using the same gene, the same enzymes to live, maintain the carbon source of PET. But for this one, perhaps it's
not
the
very
one.
specific because I'm still wondering your nutrient of the media.
Yeah.
Do you have anything else instead of plastics in your?
Of course, we have trace elements and then a little bit of yeast extract first, like only 0.5%, and we leave the culture for 2 weeks. So every 2 weeks we refresh the media to maintain the degrading the bacteria to still alive in the culture.
How do you measure the degrading plastics in your broth media? If you use the agar media, you can see the clear zones as degrading plastic indicators. With this one,
Of course, the agar media is the best one.
Right.
But because we want to extract the genome in the community, that we want to analyze that we get the full potential of our samples. So we want to like enrich the number first. So and then because of that we can get the gene. But the second problem is putting a PET film in an agar plate is quite difficult. To be able to homogenize PET in an agar plate is quite an effort and require some reagent that we don't have at the time. That's why we are continuing with the prototype.
Okay, I don't see any significance. Why do you need to do the metagenome 16S, or just direct plating and targeting your bacteria? Can explain that? Why do you need to do the 16S, or just because you only want to see the gene, right? And why do you need to know with others non-potential bacteria?
Oh, actually, it's just because we want to know the bacteria. Is it the same thing like the gene that we get? Like we can only predict is it the which bacteria come from produce the gene that we get, and we just wanna make sure did we really get the Ideonyl asakaiensis on our culture or no? Because we don't know which bacteria produce the gene that we get from our research. Like it's just only for the confirmation of our data, like that.
Okay, thank you. Because it's like blunder for me. Why do you need to do the 16S? In the meantime, you have one single bacteria as your focus. Okay, I think, can you explain why there is difference proportions in each site? You have different proportion, right, of the bacteria. It's different actually.
Oh, actually, the above picture is from the NGS from the soil sample of the G2M metase comes from, and the one is from the G1 PETase, and it's just only show to you that from the metagenomic NGS sample, we can see that the most abundance from the soil sample is really related to the bacteria that we get, the gene that we get, which is the Delphtia sakaiensis, and from the NGS data, it showed that this is the abundance.
The abundance of,
yeah.
Okay. Last one. Okay, the last one. How do you generate this phylogenetic tree? Because you only do the 60 strands with the metagenomes, right?
Yeah.
And how you generate this one?
So actually, we use the NCBI database. So we just like make a blast, and after we get the same nucleotide that have a similarity, higher similarity, then we just try to make this phylogenetic tree.
The question is how to make the phylogenetic tree.
Oh, I do the cluster W alignment, and then I use the Mega 11 to construct this phylogenetic tree.
You have 15 seconds if you have anything to add. Well, now you have 10 seconds.
So the thing is, we get the gene from the Idionosakiensis, but from others bacteria here.
Oh. All right, thank you very much. That's the end of the second round. Please come forward. Please come forward. Please shake hand. Thank you very much. Well, congratulations, Group D of 33. You won by 4 points, but thank you very much, D47. It was great. It was great to see your progress, and it was a very good. It was very good discussion. Thank you. Maybe you want to take a picture? Yeah. Thank you. Makasih ya. Maaf ya, Mas ya. Good luck.
Terima kasih.
Nonton aja dulu, lihat dulu.
Now we move to lunch break.
Lunch break. All right. Thank you very much. Okay. Gimana?
Nunggu arena lain selesai 1 jamnya baru dihitung guys.
Oh ya udah.
Kalau di sini terus mohon Mas Didi kan kita nunggu ruangan lain.
Oh, berarti paling seleksi.
Jadi masih ya. Paling cepat aja satu berarti. Paling cepat satu.
Tapi mereka tuh justru
yang punya ya. Nanti di mana? Ini bukan orang sini.
Tadi tuh sebenarnya dari pakai sistem karena justru mau nyari dalam spesies apa lagi dari itu. Diskusi ya. Yeah, I was like, come on, man, that was a good question. Kalau bagus tuh nggak bisa mikir pasti. Oh gitu ya, gitu. Jadi kayak literally maksudnya itu. Jadi mereka justru nyari spesies
baru ya, yang bisa itu gitu ya. Tapi kasih Indonesia.
Makanya nggak pakai seleksi.
Oh, iya.
Makanya
break ya, makannya nanti di area luar yang sudah disediakan. Selektif ya.
Tes 1 2. Oke.
Baiknya sih fasilitas yang presentasi jangan terlalu jauh begini ya. Jadi kurang agak begini aja.
Okay. Get the candidate. The gladiator. The dojo might. Where is he? Are you not entertained? Oh, I saw someone watching. Alright. So this next match is the opponent is not here, so supposedly he wins by default. But for that, he will be given the presentation. He will be given the chance to present for 2 minutes, and then he can get questions from all of you. So he gets a chance to warm up. Yeah. The next match, the opponent is not here, so technically he wins by default. But to give him a chance to warm up, so he gets the chance to present his 2-minute presentation and then receive 2 questions from the audience or the judges. Yeah. Udah. Nanti dia yang lose by default nih kalau telat. Gimana sih? Oh yeah, what's this? What's happening here? Ayo. What's the team? Team number? Ada.
42. Oh, 47 we eliminated.
Oh ya, oke. Ini udah nggak usah di sini kalau gitu. Not very good. So please sit down and then short introduction of you and the team. And then since your opponent is not here, but you will be given a chance to present your 2-minute presentation. And then after that, the questions will come from the public. So basically, it's a chance for you to warm up for your next presentation. Please, introduction first.
Check. Bismillah. Okay, good afternoon, everyone. So we are from D42 teams. I am Faris Izzaturrahman. You can call me Faris, and my partner, Mbak Anissa. So we want to introduce to you our research about antithyroid cancers using bioinformatics approach analysis through network pharmacology and also molecular digging.
All right. So it's the same. It's a 2-minute presentation, and then we will take questions from anyone in the audience or the judges, and then after 5 minutes, then it's over. I will start the time when you start speaking.
Next, please. Okay, thyroid carcinoma has a lot of types. We focus on papillary thyroid carcinoma, which is about 70 and 80% occurred in all of the patients in the world. And the main treat by surgery and radioiodine remains ablation. So we propose an alternative approach using some alternative such herbs, that is Pogostemon cablin and also Muraya koeknegi, also known as daun nilam and daun temurui. We're using these two compounds with two plants with 65 compounds, and the studies explore these natural compounds to potential drug for antithyroid carcinoma agents. Next, please. Next. Yeah, this is the brief steps of our analysis. We do all of the analysis by in silico approach, so all of the analysis by the computationals, predictions, and models. Next, please. Yep, and we did this analysis until the molecular docking, and also our further research will be in the molecular dynamic simulations and also RNA sequence transcriptomics. Next, please. Yep, from all of the analysis, we found that 41 compounds from both of plants are shortlisted and can be a candidate for antithyroid carcinoma. The studies identified 242 genes that are related to PTC, and we found AKT1 as the key player and important genes that occurred in all the cases. We targeting these AKT1 proteins as our target genes, and we found these three compounds as the potential alternative drug from Pogostemon cablin and also Muraya koeknegi. There is clinesterol, Norens 12-ins, and quenimbin. The study provide novel insights can be potential antithyroid. Thank you.
All right. Fantastic. Exactly 2 minutes. So please, let's have any questions from the floor. Anyone? Yes, please.
I'm just wondering, you decided on to look at these two plants, nilam and temuri. So is that any reasoning why you picked these two plants? Is it already used as a traditional drugs or how?
Yeah, first of all, our collaborator, Universitas Syakwala in Aceh, are a leading of the research in daun nilam or Pogostemon cablin. And also Muraya koeknegi, also known as daun temuru, is a common herb that used for recipe in Acehese food. So we bring up from that conditions that nilam and temurui can be an alternative because it's commonly used in all of the populations, especially in Sumatra.
Fantastic. Next. Anyone? Yes, please.
Hello. Are there other compounds that are known to bind to AKT1 aside from the native ligand, and how do they compare compared to the compounds that you found?
Yep. Next. Lagi. Next. Lagi. Yeah, this one. Yeah, so we compare all of the analysis into the native ligands that allosteric. I mean, it's the natural compound. No, it's not. It's the native compound that found in AKT1. It's named AKT inhibitor 8, or also known IKO. We compare the binding affinity scores between all of the natural compound that we examine versus that native ligands. The difference score between binding affinity in all of the compounds, our compounds, and the native inhibitor are comparable is about -10 binding affinity in our analysis and -14 in the native ligand. So it's close enough, and we can move forward for other analysis.
I was asking, is there other compounds other than the native ligand?
Okay.
Sorry. That other people have found, that other people have synthesized. Does that exist, or is this the first? Your finding, is it the first non-native binders, inhibitors?
For the natural ligand, actually there is no publication about which natural active compound from natural compound that directly target the AKT1. There is a publication. Actually, AKT1 is involved in the ERK and then make pathway, which is it has correlation with the mortalin. So previous natural compounds, it's target mortalin and then it's inhibit the activity of the AKT1. But for the natural compound which target AKT1 directly, our research is like the first one who study about this. Thank you.
Yeah, thank you very much. I have experience in also designing these extract works, but I was working with the medicinal plants, but for antimicrobial testings. Actually, my first question is, how do you extract this biocompound? And can you, is there any possibilities the restructurization of your biocompound because of the solution that you are using for the biocompound extractions? And also, are you also just targeting the AKT1? Do you have a plan to also like see is there any cross reactivity with another pathway, so we can like ensure this compound is really visible to use as this biocompound?
Yeah, thank you so much. Very interesting. So, yeah, first of all, our previous studies with our collaborator, they found this 65 compound that we use in this in silico analysis are retrieved from LCMS and GCMS from the Nilam and Temurui. So we bring up all the analysis before that's using 65 previous identified natural compound from LCMS and GCMS, and we do this in silico analysis. For the restructurizations of biocompound, actually the limitations of our research not reach out that your questions about for the restructurizations of the natural compound. So yeah, I think we still move forward from the in silico analysis. And also the next one, another pathway that can be inferred from the analysis. Our further research, we want to investigate AKT1 correlations and intervention with mortalin, because we found that a lot of literature that talking about PTC, AKT1 always show up with mortalin activity. So you can find a lot of number in AKT1 and mortalin active in a lot of PTC cancer. So the next research, we want to find the correlations between AKT1 and mortalin with an RNA sequencing or transcriptomic analysis. Thank you.
Thank you. Congratulations. Thank you. That was 6 minutes, so I think you gotta warm up for your next one. Okay. All right. Thank you. Thank you very much. Thank you. All right. So the next match would be.
D36 versus D05.
D36 versus D05. Please. Which side switch? D36 sama D05. 36 sama 05. Oh, ini berapa beli? Oh, katanya 05-nya yang sebelah kanan. Soalnya kemarin tadi dia udah di kandang katanya, jadi dia di kandang. Jadi 36 terus D05. D05 di oh. waiting, as usual, we start by introducing name and background, and then we go to the coin toss to decide who gets to decide who goes first. Please.
My name is Nadia Fadila, and I'm here with my teammate Dr. Sriwadini. We are from Faculty of Medicine, Universitas Indonesia.
Thank you. Now, please. Team DO5.
Okay, hello. Good afternoon, everyone. My name is Corey, and this is my teammate Sri. We're from Team DO5. We're from the Eichmann Research Center for Molecular Biology, BRIN.
Fantastic. All right.
It won't let me switch. I don't know why.
Oh, okay.
Kitanya switch.
Boleh kok.
Iya deh, sudah deh. Supaya nggak bingung.
Iya, rasanya.
Nanti judgingnya juga ribet. Alright, so we're gonna go to the coin toss. Who picks heads? Who wants heads? Heads, alright, okay, heads. Yeah, sorry. Heads, please choose whether you want to present first or they present first. You want to present first? Alright, please prepare. About 1 minute to prepare, and then the presentation will be for 2 minutes. Remember that the presentation can be marked up to 3 per judges, so it'll be a good chance for you to, get a head start. Yeah, 1 minute to prepare. If you want. Do you want to prepare or do you want to start?
We can start.
Oh, you want to start? All right, okay.
As possible.
Okay.
As soon as possible, yeah.
All right, I will start the time when you start speaking.
Okay, thank you. Here I would like to present our study regarding the geospatial approach for a hepatitis B virus in Indonesia. Next, please. Why we choose hepatitis B virus? Because the chronic HBV infection is a major risk factor for liver cancer, and the HBV genotypes vary across different regions, and its genotypes is associated with the clinical implications. Next, please. Therefore, we utilize a geospatial approach to analyze the regional molecular epidemiology of HBV. We collected the clinical isolates from 2 referral centers, Jakarta and South Sulawesi, and then we perform genotype profiling to perform further geospatial analysis. Next, please. As you can see here from our data, from 2 referral centers, we found there is no significant differences between sex and age. But if we go to subtype, there is ADW, which is the highest one in Jakarta, and ADR is the highest in subtype in South Sulawesi. And for the genotype, we found the genotype B as the dominant one in Jakarta, and C is the dominant one in South Sulawesi. And from the distribution, you can see that the HBV blue for B and red for C. And from the hotspot analysis, we found that North Jakarta and Makassar are hotspot cities for both genotype. Thank you.
All right, thank you. Thank you very much. That was 1 minute and 40 seconds. And now we begin the question and answer. I will start the time when you start speaking with 1 minute per question and 1 minute maximum per answer.
Okay. First, why don't you make a geospatial analysis based on the subtype of HBV?
Thank you for the question. So subtype is important for epidemiological data, but if you want to look at the clinical effect, it's better to use the genotype data, because there are several literature have shown that certain genotypes have certain effects on the severity of the disease. So if you compare B and C, genotype C usually have more severe liver disease manifestation. That's why we use the genotypes data.
The only genotype, not subtype?
Yes.
Okay. Then, in the conclusion that can you explain and give the explanation, management and treatment strategy based on HBV genotyping, because you have made a map.
Thank you for the question. So here the case is that we are Indonesia is an archipelago country, right? We are have very wide area to cover with all the health problem that just is not only HBV, of course. So what we want to reach from this hotspot analysis, we know where we want to target for the prevention. We want more targeted problem. Here we can see that here is the hotspot and we need the intervention here. So that's the idea of these studies.
Example of the action, maybe for Jakarta?
So far, no. This is so based on our knowledge, this is the first geospatial approach for molecular epidemiology, particularly genotype. So far, we don't have any data for using this geospatial approach for policy yet, and we hope it can contribute for the future preventing our public policy or for public health.
I just make a comment, like the samples and data you were collected is during 2017 and 2018, which means the finding might not reflect the current situation, as changes in public health policy, screening and treatment practice since then could have influenced the prevalence and distribution, so it might not be generalizable. What do you think?
Yes, that's also a concern for the study. But I think looking at what the government, the available health program currently, so we still don't have any epidemiological data on the G2 type itself. So we have the national prevalence of hepatitis B infection, but looking down at that's on the general population, but no epidemiological data on the Hep B genotypes on the patient. So there's still nothing there. So yes, it might be a concern whether this data is still applicable at current situation, but we think that it's better that we do the analysis now to inform better approach for to encourage more studies on this field, I think.
My other concern is, why do you only use one center for one such big province?
So this is we use existing data on our previous study. So this is the only new novel things that we do is the geospatial analysis. All the genotypes data are based on previous studies that we've done. So at this point, yes, it's also concerns whether we should include different referral centers. We acknowledge that is one of the limitation, but at that point, it's our starting point. So if we can get more data, of course, the data will be more representative on the province itself.
Also, the study design is cross-sectional. I think it limits the ability to infer temporal change or trends in the geographical distribution of HBV genotype. So I think longitudinal studies would be more appropriate and would provide more insights.
Is there a question or?
No, like a statement. Like I think like longitudinal would be better for this type of study.
Please respond.
Yes. Thank you. It's a very good input for us, and we acknowledge it also. Of course, the temporal studies will be perfect for this because geospatial approach also very useful for that. But as my friend mentioned before, what we try to do here, and also like to utilize the data maximally, because we have this data from 2017 to 18. Of course, it's not an actual data, but what we are trying to introduce here also that all the data that we have, we can maximize it, the use of it. So even the genotype that previously no one care about this geospatial, but actually it can be useful. So we want to introduce this also that we can maximize the utility of our data. Thank you.
Last round, so you have 1 minute for your last question.
I think it's about the sample collected from referral center. I think it may introduce selection bias when you collect it from there, as patient presenting to those center might not represent the general population. And also, I see that in South Sulawesi, it's almost half of the samples that you collected in Jakarta.
So, sorry, can you make a question on that?
Yeah.
Like a statement, another statement.
Okay.
Okay, thank you. So yes, we acknowledge the bias, of course, because the two cities are actually different in terms of socioeconomic and also the density. For example, Jakarta has a density of 16,000 people per kilometer square. Meanwhile, Makassar has 200 per kilometer square, so it's totally different. But based on that, that's why we have a lower number of cases, because it's a total enrollment study for 6 months. So that's the represents also the density of the population.
That is fantastic. Thank you very much, and that is the end of first round. Okay. Okay, so for the next one, we have about 4 minutes to prepare for the next group, which will be D36, right? Yeah, not 32. I was like, yeah, for D36 to prepare the presentation in 4 minutes. Then. Yeah, D36, yeah, whatever. All right, okay. You want to start now? Yeah. Oh, all right. Okay. All right. Okay. Sure. Okay. In that case, we're gonna start round 2. Judges ready? Judges ready? Judges okay? Yeah. All right. So again, just to repeat, you have 2 minutes for this presentation, and you can be scored to up to 6 basically, because it's 3 per judges. So it could be, good to give you a head start. And then after that, we will continue with the question and answer. I will start the time when you start speaking.
Okay, today I'll be discussing phylogenetic analysis of Sarcoptes kbi in human population, a case study from Jakarta, Indonesia, and global comparison. Next, please. So scabies, as you know, is a contagious skin disease which is a rising public health problem worldwide. Our research will focus on genetic epidemiology using COX-1 as a gene marker. This study aimed to analyze this genetic diversity of Sarcoptes kbi in human from boarding school in Jakarta and compare it with mites of various hosts and region. Next, please. So we collected sample from students at boarding school X in South Jakarta that were diagnosed with scabies. So as of now, 21 samples were processed to amplify COX-1 gene and then analyzed with 62 gene bank sequences and then aligned together and then analyzed using maximum likelihood tree. Next, please. So our result that there are 3 genetically distinct clade. Clade 1, marked in red, is human samples from Jakarta and Panama. Clade 2, colored in blue, is global human samples from Australia, China, and South Korea. And clade 3 in orange is such a diverse samples and hosts from like humans, dog, wombat, and water buffalo, and also locations. Next, please. So clade 1, it suggests uniquely adapted lineage due to genetic conversion or geographic isolation. It also points at the common ancestry. While clade 2, it hints at how human migration might facilitate the spread of scabies. It also shows the strong mite adaptability and survival. While clade 3, the most diverse with samples from various hosts and regions, suggests might have developed genetic mechanism to evade both host defenses and thrive in various ecological niche. In conclusion, our study demonstrates the complex genetic relationship of Sarcoptes kbi. Thank you.
Very good. 1 minute 50 seconds. That was very concise. All right, now to start, DO5 can ask the questions. Remember, it's 1 minute per question or per answer. And I will start the time when you start speaking.
Thank you for your presentation. Very interesting. Just wondering whether the clade of the scabies tick actually affect the diagnostic treatment or even the clinical symptoms in the patients.
until now there is no data mentioned about how the relationship between clade and clinical manifestation. But in real patient, we can see maybe one area is most of clinical prevalence or clinical manifestation is different from other pesantren like that, and why the student in that school can get reinfection maybe every year, and other pesantren still clean.
I would like to add that this study has not yet completed, so that is one of our goal to make connection between the variant and clades into the clinical manifestation.
Okay, thank you. I want to ask, because scabies, as you said before, there is a common disease actually in the boarding schools or in the for example pesantren kind of. But actually I wanna ask to the more specific for why you select this boarding school among all school.
We studied in this pesantren since 2015, and every year we do the education. But after they back from home in holiday, then back to pesantren, the students still get reinfection. That's why we curious what happened with this school. Every year we do the education, then mass drug, then other support like what kind of baju dalam, yes, like that.
Undergarments.
Okay, thank you. Okay, yes, that's a nice finding also. But from there also raise another questions, because when you say that when they come back again from their home, they reinfected again. I missed the data of demographic data from your cases, and I wonder if you have or not, because what's important in my opinion also, because maybe we can track also the demographic data and what cause of this reinfection, as you said before, or maybe the origin of the students also affect. Maybe they bring back from their hometown and then go back to the pesantren and then they reinfect the other, or kind of.
Yeah, we have also data from the origin, the town. Then when they come home, the last, the duration when they go back to their home, and what else the data we have?
We have the basic demographic data, the data when they go back home, the duration, and when they go back to the school and their reinfection. And as of now, we do have those data, but even the PCR has not yet been completed 100%. So this is what we have right now. But that is also our main goal.
Okay, I'm just going to ask about the phylogenetic tree. So you're using the maximum likelihood method to generate the phyllo tree, right? So I'm just concerned about the bootstrap values in the roots. How confident are you about your phylogenetic tree analysis? And have you considered to add more isolates to increase the separation between the clades? Or because even with the outroot samples, it's very low bootstrap value. So.
Pit stop. I won't count. All right.
So for mainly for our branch for Jakarta, we have a nice value of 86%, which is pretty good. It's above 70, and we also have planned to add more samples from our own sample from Jakarta, and also about 200 more samples that we will retrieve from gene bank. So it will be more completed.
Continuing from that. So you said that the clade one has a localized genetic adaptations, but the origin of the samples are very far apart geographically, right? One from Jakarta, one from Panama. So what do you think happened? How do you explain the, I don't know, evolutionary something for the tick, or how do you explain the clade for me?
So this is our hypothesis is that they may have a common ancestry, and number two, they may have a parallel evolution. So they independently evolve, but because they have the same environment, I mean similar, not same, similar environment, they try to adapt to similar environments, so they have similar evolution.
Last round.
Okay. Correct me if I'm wrong, because I found that during my study, I also we find that scabies is more active during night, right? So when you do the sampling, when did you do the sampling collection? Do you collect during the night, or and is it apply for the same collection time for all the samples?
We do the collection in the same time, but not in the night. But because it's active, it means that the might make a burrow. But we do the skin scraping from the nodule, not the burrow. Not only from the burrow, but we can also take the skin scraping from the nodule or pupil, like that. It's also an active lesion in scabies.
All right, thank you. Thank you very much. Let us do round two. So please, everybody have a front shake while we wait for the drinks to come in. Please. Judges, done? All right, so congratulations for continuing. And thank you and congratulations to Team D36. It was a Oh, there's a. Oh, yes, please. Oh, wow, all right. Thank you, makasih ya,
For Team DO5, we'll be moving to match 32 in Arena 2. Arena 2. Probably around 40 minutes from now. Not yet. But for Team D36, we'll stay in this arena.
Oh.
And then we have 3 matches away from
Oh,
their
okay.
arena.
All right. Okay.
For now, team is D33 versus D51.
D33 and D51, please. D33.
33 is second phase of elimination. 51
Oh, okay.
the first.
D51. Oh, they're both second phase elimination, right?
He has presented two
Fourth.
times. This is the 4th time, sorry.
Yeah.
Yeah.
Yeah, so basically both are in elimination, right? Yeah. Oh dear God. So much tension. Give me a moment. Yes?
I don't know if this is a good idea or not, but can we try to, in the attack house, take the question? Because otherwise it's hard to respond to it, right?
Yeah. So that's why I always ask, like, is there a question in there? Because if it's just a response towards a statement, it is usually, it's like you're giving a free point to the defender basically. So if you do have a statement, you should at least phrase it as a question at the end, like, do you think your method is accurate, or how do you think you can make it more accurate, or something like that. So it's just better that way. You can have a good background based on the statements that you were given, that you give, but then it's great to still have a point to attack basically. Does that make more sense? Yes, Christina? All right, so where's my point? Oh, here it is. Okay, so for this round it's D33 versus D51. Both did their previous matches in this arena, so very, 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 very important match. As usual, we will start by introduction of your name and your background, and then we continue to the coin toss and the presentation. Please.
Hello, good afternoon, everyone. My name is Tafroji, and I'm from IMERI. My background is microbiologist. Thank you.
Hello, we are from team D51. My name is Christy. I'm a pediatric neurologist from UGM Academic Hospital, and this is my colleague, Dr. Aulia. She is a pathologist from Dr. Sarjita Hospital. Thank you.
Fantastic. Thank you. All right. Call. Who's heads? Who's tails? Number. All right. Okay. Number. So you get to pick whether you want to present first or not. Oh, all right. They pick you to present first.
Okay.
Okay. Please prepare the.
It's my fourth.
Your fourth. Your fourth first presentation.
Yes.
All right. I will, as usual. It's a 2-minute presentation. It can be scored to up to 6 because there's 3 per judge, and I will start the time when you start speaking.
Okay. Thank you very much. So basically, I just want to do the evaluation of the PCV13 impact to the serotype resistance of the Streptococcus pneumoniae, because we are now in Indonesia has been implementing this PCV13 as a national immunization program. So I think it will be good if our government can know these results and then they can plan how to evaluate in Indonesia. So for the materials that I use, I collected the whole genome data from Pathogen Watch for 15,000, and I do the filter and source selections, and I analyze 5,000 WGS for this analysis. Next one. And from these analysis, we have found that there are declinings of the serotypes included in PCV13, and most of the serotypes, serotype 3, 19F, and 4 is the persistent serotype after PCV13 introductions. And we also discovered there is a reduction of prevalence of the multi-drug resistant strain of this Streptococcus pneumoniae, as well as the antimicrobial resistance profile moved to less resistant after the PCV13 introductions. We did also the comparison in carriage. Carriage also do the same with the antimicrobial, but for the prevalence, there is no any significant of vaccine serotype circulating in our community. Thank you.
Fantastic. Thank you. Okay, so now we're going to go to the question and answer. It's 1 minute per question and 1 minute per answer. And from D51, I will start the time when you start asking questions.
Thank you. I want to ask, you see many study in a country where you not see the data from Indonesia, because you want to implementing the result to Indonesia. Yes.
Yes, right. So Indonesia has no publication for this WGS so far. That's why I collected the data from the Pathogen Watch. The secondary data, it's public access. It's free to access.
And how you pretend bias in data, because it's many country. So what is the criteria inclusion and exclusion for this patient?
Yeah, thank you. So the main criteria inclusion of the country that I use for this data collection is mainly for the completeness of the data from the serotype, antimicrobial testing, and also the antimicrobial profiles, also the sequence type of the data. And also to avoid the bias, because we are in tropical country, I also included like Cambodia, Malaysia, South Africa, and also Brazil as a tropical country, so we can let's say they are already included in our analysis.
Yeah. So do you make a subgroup analysis of developed country and developing country? And do you think there is different serotype or genotype bacteria circulating in those regions?
We did not do that because I just want to know globally factors. So I did not analyze according to the developing country, developed country, or not developing country. So it's only a big picture of what the impact of PCV13 of global implemented these vaccines.
So what would you guess in Indonesia would be? Is it the same?
Yeah, I think the pattern is so far the pattern is the same. We have the data from previous pre-vaccinations, and we still found like vaccine serotypes, the invasive ones, still circulating in our community, and if the government will design for the surveillance, it will be good to have the evaluation. So we expect we will have the same results, but we need to concern the non-vaccine serotypes not included in this PCV13 that will emerge after the PCV13 introductions.
Yeah, and I think because the introduction of PCV13 is late in our countries, it's just like 2023, if I'm not mistaken.
It's October 2022, yeah.
2022, and so in other region, it's already very long time ago.
Yeah.
So is there any differences because of the late introduction in our country?
I don't think so, because we design this vaccination is for the children. By implementing in the children, they can avoid the circulation of the invasive serotypes in the children, like they did, the previous country did for the implementation. So I don't expect any differences after the implementation with this PCV13.
Thank you. So I look at your graph, and there is declining in some region, in some serotype, but in the 6B there are increase after the PCV13 introduction. So what is your comment?
Yeah, if you see the graph, the above one is for the invasive pneumococcal disease among the patients. The lower graph is for the carriage survey. It's from nasopharyngeal. So from the invasive cases, we found only serotype 3, 19F, and 4 that are persistent after the PCV13 introductions. But for the carriage, 6B can be the major serotypes. That's only.
But because the immunization should suppress the bacteria, right?
Right, but not in nasopharynx, because not in the immunization will condition in your bloodstream mostly. That's why there is a reduction of pneumococcal disease when they infect the blood, CSF, and pleural fluid, but not in nasopharyngeal.
Oh, the first one is in the
invasive.
Invasive.
Yeah.
It's also
This one?
high. So for the 6B.
This one?
No, the first one.
No, it's serotype 3.
Oh, that's serotype 3.
Serotype 3.
Oh,
Yes.
not 6B.
Not this one, 6B.
Oh, okay.
It's
So
Yeah.
this vaccination is not good for the 6B, and it can be contagious to other patients.
The vaccination will not affect to the carriage, but it will affect in reductions of the invasive cases. Yeah.
This is the last section. So
How about the antimicrobial resistance? So I don't think, you mentioned that there is a reduction in the resistance. But how about the PCV13 introductions in the resistance? So can I use more antimicrobial in my patients right now?
So the principle of the PCV13 introductions, serotype included in the PCV13s is mostly resistant to antibiotics. So by eradicating these serotypes, you will decrease the resistance for the Streptococcus pneumoniae.
Yeah, and one more.
Yeah.
PCV in Indonesia, the national immunization is using PCV10, not PCV13. Do you think it will have another circulating serotype in our country?
No, actually it is PCV13 now.
For the national immunization?
The national immunization program, yes.
Oh, really? Okay,
Yeah.
thank you.
and that is the end of round one. Thank you. All right, so next round we have about 4 minutes. Maybe just a bit of an interesting background. The reason why they're so back and forth because both of them really know their stuff.
Yes.
Because vaccination, I believe being a pediatric neurologist means that you're very familiar with the vaccinations, but you are also
The microbiologist.
Yeah, you are also a microbiologist. So that was one of the most intense sessions I've ever refereed. So that's very exciting. Thank you for that. Okay. Again, I'm gonna give you the option. We always have like 4 minutes for preparation if you want, or you can start now if you want to. I wouldn't stop you. So, but I will start the timing for 4 minutes from now. Just tell us when you're ready. Oh, you want to start? All right. Okay.
Okay, thank you. Our study is about exome sequencing in DEE, Transforming National Clinical Practice Guidelines in Indonesia. Next. DEE is the most severe group of epilepsy, severe seizures, drug resistance, early morbidities, early mortality, and cognitive and behavioral impairments. You see, there's a lot of gene responsible for DEE, but finding the gene, the etiologic gene, can know the underlying mechanism and transforming clinical diagnosis and management. Next slide. Currently, guideline is using empiric therapy based on type only, in which maybe the contradictions of underlying mechanism. For example, focal seizure, you use carbamazepine, which is natrium blocker. But if your which is also a natrium channel blocker, then giving making it worse. The study aims to look at the genetic profile of the patients, directing patient therapy management in Indonesia. We use the patient's phenotyping, whole exome sequencing, and find the genetic etiology and use personalized treatment to the patients. Next slide. We identified the genetic etiology in 47% of our patients. The most common genetic etiology was SCN1A. So you see, we manage our patients wrongly currently. Management changes was conducted in 9 patients, 7 drug changes, and 2 avoidance of brain surgeries. Next slide. The study illustrates that whole exome sequencing in clinical setting in Indonesia is feasible, has high diagnostic and clinical utility, and can modify the treatment for the patients in Indonesia in clinical setting. Thank you.
All right, so for the question and answer session, again, 1 minute for each part, and I will start the time when you start asking. Oh, pit stop part. It's okay. I won't count. All right.
Okay, I'm quite skeptical with your subjects number. It's only 50. What power do you have for these subject numbers?
Yeah, this is
because you are talking about the etiological, right?
Yeah, this is actually a rare disease, 1 in 1,000 patients. So we collect all the patients who comes to our referral hospital and meet the inclusion criteria. So in a national study of Australia, they only use 100 patients. So in only one referral center in Indonesia, 40, 50 is, I think, is makes sense. Yeah, maybe the sample size is different for rare disease compared to common disease.
Okay, so it's, okay, like if you have like 100, I think it will be good to say that this is the etiological agents. And yeah, I think for you collected the demographical profile, right, from the patients. Yeah, why don't you try to analyze this one? Is there any? There is no any correlation so you found with the SCN1 with the symptoms or the demography from the patients?
What kind of demography?
Symptom, I mean.
Oh, the symptoms. Yes, the phenotype is very one for another. Maybe you can see at the first slide. They have like developmental delay. They have and epilepsy. It is very hard to differentiate only from the phenotype, only from the symptoms. That's why we need a genetic testing for the patients.
Don't you have any scales for this grouping of the symptoms so you can do the analysis, the severity of this SCN1?
No. They are all resistance to drug. More than 2 drugs we apply to the patients and there's no decline in seizures, then we said this is intractable epilepsy.
Yes, and we make some point for inclusion criteria like EKG, EEG, and MRI, and also the seizure.
Okay, so from these clinical symptoms, why don't you try to find the risk ratio or the old ratios, because you have the AZN, you have the mutations on the SNP, so you can actually you can do this analysis, right?
Yes, if it is available, maybe there's already a guideline in the world, because it's not us who knows the genetic of the symptoms, but because we don't know, because it's too similar one another's, then we still have to conduct the genetic studies. In fact, I suspect this is a SCN1A mutation, but when I test, I'm wrong. Yeah.
Oh, really? Okay. And I thought that you performed the cohort study for this one, right? And you also did you also collected all the symptoms during your cohort study? And
Yes, of course.
did you analyze in your
Yes,
research?
of course.
And-
Because in bioinformatic analysis, we have to compare our result with the phenotype of the patients, with the human phenotype ontology. So every one single phenotype, we have to compare whether these variants causing this phenotype. So we collect all the symptoms of the patients.
Okay, when you collect the symptoms, it's cohort study. Did you analyze? Is there any differences, like possibilities SCN is not expressed and causing these symptoms in what phase during your course study?
We did not, but thank you for your suggestions. But from previous study, because we have OMIM, ClinVar, we can already know like the SCN1A, what is the symptoms of SCN1A. But you know, because it's really hard to differentiate between one seizure to another seizure, because like both of mutation can have the same seizures. So yeah, but it's a good point. Thank you.
Yeah, I think it will be very impactful for your etiological agents. It's very important. Okay, and the last, I think not the last one. What is the correlation of your SCNA1 and other SNP that you found in your study with the drug and diet treatment that you are planning?
Yes, there's very much correlation. SCN1A, we cannot give natrium channel blocker. For KCNQ2, we must give a natrium channel blocker, because that is a potassium blocker. And then for GLUT1, glucose transporter 1, we have to give ketogenic diet, which is a fat, but the defect is in the glucose. So we have to give calorie from fat, not from glucose. And it's exhaustive data. So yeah, I just give you an example. But very much what we can do by we know what is the responsible gene.
But okay, but you need remember you need to do more analysis for the whole genome sequencing profiles, because it's talking about the exomes. Probably there is correlation with all the genomes of the patients. And how do you think about that?
Yeah, thank you. So
Because this is his last session, so are you finished?
Yeah.
Because you still have like 40 seconds. Is that okay?
Yeah, okay, I think.
All right, okay. Please.
okay, thank you for the questions. So yeah, so most of the mutation we found here is single nucleotide variants. So we use exome sequencing, but in parallel, because this is part of the BGSI program from Kemenkes, in parallel we do the whole genome sequencing in Jakarta, and we do the whole exome sequencing in our center. Thank you. Okay.
Anything to add? No?
Yeah, maybe because you raised question about copy number variant and structural variant that can happen in our patients. Yes, you're right, and we already back up with whole genome sequencing. But our publication would be the whole exome sequencing, given that this cost effective at this time, because whole genome sequencing only raised maybe 5%, not too much, but the price is very high. Thank you.
Thank you very much. And that is the end of round 2. Please, everybody, to come forward as usual while we wait for the judges to finalize their gradings. Judges, is it final? Everything's good? All right, so thank you very much and congratulations. Team D51.
I just enjoyed the discussion.
So, okay. Congratulations.
Makasih ya.
Makasih ya. Thank you. Selamat lagi dong.
For Team D53's final finally eliminated.
D33.
51.
Good game, good game, good game, good game, good game.
Match 28 in Arena 1. Arena 1.
That's good, right?
We go Arena 2, but the opponent is not yet decided.
Oh, waiting for the wins.
As for this arena, the next teams are both still in Arena 2.
Alright,
so
so we wait. Alright.
we get a little like
Okay, we got a little time. Alright. Good luck. Arena is very chop chop. in D41 and D42. Ini kebalik ya? D4. Okay, so please switch sides. D41 on this side and D42 on the other side. Just to assist in scoring and understanding the situation. Thank you. Right, so in the beginning, we're going to start with introductions, a name and background from each team, and then we continue to the coin toss to decide who presents first, and then it goes as usual. Please, D41.
We are team 41. I'm Daily.
Dr. Selfie.
Yeah, both of us from the Department of Microbiology, Faculty of Medicine, Universitas Indonesia. Thank you.
Okay, thank you. We are from team D42. My name is Anissa and my partner is Faris, and we are from Bioinformatics Research Center, Indonesian Institute of Bioinformatics. Thank you.
Fantastic. All right. Thank you very much. Now for the coin toss, heads or tails? Who calls first? Heads or tails? Head. All right. Heads for D42. And it's heads. So please decide whether you want to present first or you want to let them present first. Present first? All right. Please prepare D42's presentation. The presentation will be 2 minutes. Remember that the presentation can be marked by the judges to up to 3 points per judge, so that's 6, which should be a very good start for you. I will start the time when you start speaking.
Okay. Good afternoon, judges. So we want to introduce to you our research anti-thyroid cancer potentials of Pogastemon Kablin and Murray Kognigi in bioinformatics approach analysis. Next, please. We targeting thyroid cancers. There are 4 kinds of thyroid cancer, and the most common is papillary thyroid carcinoma. This cancer is challenging to treat, require advanced method with a lot of side effects. We propose a natural treatment for papillary thyroid carcinoma using Pogastemon Kablin or Nilam and Murray Kognigi or Temuru with these 65 compounds we retrieved before. The study explore natural compounds against death papillary thyroid carcinoma. Next, please. Yeah, this is the steps that we required into this analysis. They are step 1 until step 6, molecular dockings, and our research, further research, we want to do step 7 and step 8, molecular dynamics and also RNA-seq database analysis. Next, please. Yeah, we shortlisted 41 compounds from all of the steps, and we identified 240 to identify genes that link it into the papillary thyroid carcinoma. And we found that AKT1 is the most playing important role genes in our network analysis, promising candidate for further investigations. We do molecular docking AKT1, and we found these 3 compounds, cleonasterol, Norans 12L, and coenimbi, as the promising natural compound that we can suggest into the natural herb alternative agents. Next, please. Yeah. Also, we found that AKT1 and mortalin have overexpressed in all the thyroid cancer. Our further research suggests that transcriptome analysis to find number of AKT1 and mortalin in PTC cancer using transcriptomic analysis. Thank you so much.
Thank you. Thank you very much. All right, so now we go to the question and answer session. Each question and each answer is only up to 1 minute, and I will start the timer when you start.
Thank you for the interesting presentation. First of all, maybe I would ask about the AKT1. You mentioned previously as the key player in the thyroid cancer. How do you think so? And also, we know that in cancer we have multi-genomic and it will influence one another. And how come you produce a drug that only target one gene? Thank you.
Yeah, we do network pharmacological analysis. Can you go to the previous slide, please? Yep. Yeah, in this one, we identified there are 242 linked to papillary carcinoma and the potential drug in our herbal compound. We do all of the analysis and we found that AKT1 is the most important by degree filter. So we choose that AKT1 as our target.
Yeah. How do you know that each protein didn't affect one to another?
Sorry?
So you found 242 genes, and I believe all of the genes actually interact to each other. And how do you know that when you target only one gene among 242, you can already target something in there in the cancer?
Okay. Can you go to next slide, please? Again, next. Yeah, in this one. The color is so bad. So we also do enrichment analysis. All of the genes, the 242 related into our analysis. We do the enrichment analysis and we found that 242 related into the pathway in cancer. And we found AKT1 in all of the literature are correlated into the cancer in the commons and specifically in papillary thyroid carcinoma or thyroid cancer.
Yeah, I understand. But then when you have a lot of genes actually related to that cancer, when you only target one genes, the other genes actually can interact with each other and also causing the cancer still been there.
Yeah, actually, if we are talking about thyroid cancer has specific mutation. It's called KRAS mutation. Thyroid cancer has different mutation at it and relation with APK mTOR pathways, right? And also including AKT1. And then how we can know that it's only targeted AKT1. So the next study we want to do the in silico study. So we will knock down or overexpress only AKT1, and then we will know what happened, and then we will check the downstream of the AKT1 and the mTOR pathways. Thank you.
So actually, from the previous study that I learned from Kumar in 2019, the interactivity in the genes in thyroid cancer also, it can still affect the cancer itself. So you cannot actually really only docking one single gene and then expecting the patient to get better.
Yeah, so that's why.
Is there a question in there or? No, that was just a statement. Please respond.
Okay. Yeah, thank you. That's true. So actually, I started this study and in collaboration with professor in the USA. His name is Professor Jong In Park. So we already study about it, and then actually we have data, also data patient that in the patient actually AKT1 is also higher, and it has correlation about with mortalin, and also of course it will be has correlation with another pathways, but our study is only focused in these pathways. Yeah.
So okay, just say you're successful with this compound. What is the best way for administration of this anti-cancer? For the administration.
Oh, you mean oral or? Yeah, something like that. Oh yeah. Okay. So based on my experience, when I did this research in the USA, actually for the treatment thyroid cancer, it's not only one way. So we can mix maybe by surgery or radio iodine, and also we can combine them also with the NON inhibitor for thyroid cancer, for example small molecule. So for that, actually for now we don't know because it's only prediction, but our future research, I want to adopt also my previous research that I will try like several treatment based on the stage or the status of the thyroid patient.
So in which stage that you plan to use this compound? With stage?
Based on my experience, we use it in the stage 2, but we have to check like the condition of the patient, right? Because even if they have thyroid cancer, but they have different immune in the body, so maybe there will be different also respond for the drug. So that's why for the study in the future we will try 2, because in the stage 2 talking about AKT1, okay, AKT1 is increased and also mortalin is increased, right? But we still can cure them. But for the stage 3 or step 4, it's maybe difficult. So maybe we need like surgery or other treatment. So we can start from stage 2, because for the stage 1 maybe we can just remove by surgery. Yeah, thank you.
Last session.
Okay. So do you can you confirm that this therapy actually will be very specific to the cancer gene and will not affect the normal cells?
Yeah, of course, because we have to check using in vitro, in vivo analysis, right? So we will use control. So we will use normal cell and we will check whether when we give treatment to the thyroid cell or to the normal cell, it will only give effect to the thyroid cell, but it will be safe to the normal cells.
Thank you. Thank you very much. Let's get another round. All right. So just give us a short comment. I think it was very good and very intense to start from the basics parts, and then you extended it into how this could be useful. So that was very interesting. But again, just to remind, it would be better if you can formulate, if you have a statement and then you formulate a certain question out of it, so that you can be more targeted in your session, basically. All right, so usually there's a time for you to prepare, but we give option if you want to start directly, you can do that also. So yeah, because it's the same. So usually there's like 5 minutes to consult the coach.
Yes, okay.
It's
okay. Just consult or just directly, yeah? Okay, just
Directly? All right, okay. Okay, in that case, I need to remind you that the presentation is at 2 minutes. It can be graded up to 3 per judge, and that I will start the time when you start.
Okay, thank you. Good afternoon. Our project entitled Sputum Microbiota Profiles in Hospitalized Adults with Community-Acquired Pneumonia. So, in imbalanced bacterial composition or dysbiosis in the respiratory system has a play role in the incidence of community-acquired pneumonia or CAP. So, this study aim to identify the etiologies and the microbiota profiles of hospitalized patients with a CAP. So this is the workflow of the study. We did a culture and real-time PCR to confirm the etiologies. In addition, we conducted 16S sequencing to profile the microbiota from the sputum samples. Next. So we divided the subject into 2 groups, mild, moderate, and severe, and then we checked several variables that contributing to the severe CAP, and from all the variable analysis, only age in the study statistically significant contributing to the severe CAP. So most of the patients are polymicrobial infection followed by bacteria and viruses as a single pathogen. Next. So we compare the alpha diversity between mild, moderate, and severe cases. Even there is no statistically significant difference, however, we can see the trend that in the severe cases, the alpha diversity higher compared to the non-severe. And it also confirmed in the Spearman correlation that we found in the severe group dominating by the pathogenic bacteria. Next. So in conclusion, CAP in adults was mostly caused by multiple pathogens, and in patients older than 65 were 6 times more likely to have severe CAP than younger patients, and patients with severe CAP had higher alpha diversity but had more pathogen and fewer commensal bacteria. That's all, and thank you.
Thank you. Very good. 1 minute 50 seconds. That was fantastic. All right, next we go to the question and answer session. As usual, 1 minute per question, 1 minute per answer. And I will start the time after you start.
It's interesting, examines metagenomics using 16S approach, but you state that using Illumina just in the V1 and V2. A lot of research state that using long read sequence such as nanopore that covered full V1 until V9 is more accurate. Can you make this state valid in your research?
Thank you for the question. So yeah, each platform actually have its own pro and cons, and we know that in nanopore technology for the microbiome analysis, it actually have some flaws that it contains more error than the Illumina one. So at that time of this project, actually Illumina is still the gold standard for the microbiome analysis. So that's why we choose to use the Illumina platform.
Yeah, okay. Yep. And you state that in the severe patients, you got more alpha diversity. That means it's more richness. Can you make sure which, I mean, like some pathogenics can be higher abundance or just extinct. Exist some pathogenic bacterial species. I think more diverse cannot be make a validations for your pathogenics conclusions.
Yeah, so we performed the Spearman correlation test, and in that test we can see that the pathogens are more significantly more found in the severe in patients with severe pneumonia, and the normal common cell bacteria are found less in patients with severe pneumonia compared to the mild and moderate patients.
So just you have a list of species that you targeted.
Indeed, yes. In the previous slide that you see the heat map, it is actually showing different genus of the bacteria that we found, and the blue dots and the red dots showing the correlation of the genus to the severity of the patients.
Yeah, more interesting thing that I found in your research is you are looking for microbiota profiles and community, but you also look out another risk factor. Can you explain what the correlations between all of the factors that you examine and also synchronized microbial community?
Okay, so at first we in all patients with CAP will be diagnosed, will be confirmed what is the etiology of this patient. So and at the time, and then we assess the severity of the patient. So using pneumonia severity index, it's called. So it's 1 until 5. So mild and moderate will be PSE 1 to 3, and 4, 5 it is severe at the time, and then we separate it. So what is the risk factor of this patient of having severe CAP?
So you split all of the group into mild, severe, and also healthy by the risk factor, or?
No. It is based on the pneumonia severity index. It is the one that doctors usually use in the clinical setting. So it is in the scoring system. We see the clinical condition of the patients, and then we give some scores based on the variables, and then after that we confirm if the patients have mild or moderate or severe pneumonia.
Yeah, I think it will show up in other questions. If you have split the group into your pneumonia conditions, and you combine with risk factor, you should split it again, like mild with some risk factor or severe with risk factor. So it should not just 3 group, but 3 with the child group, I think, like that.
Sure, sure, we're working on it actually. So the analysis is still running right now.
Yeah, it's really interesting. Still, we have time?
Yes, you have about 2 more questions.
Yeah, in the richness analysis, you state that alpha diversity in your severe conditions is more higher, but commonly to inspect the microbial community can have a significance. We using beta analysis that examines which barcode or like which conditions higher using some distance algorithm.
So yeah, in some studies, in some previous studies, for example, from Pettigrew and Wong, we can see actually the alpha diversity is also changing. But in this, the main problem in this study actually, because we only took samples in one point and we don't have any follow-up samples for that. So yeah, but already in the beginning of the hospitalizations, we can already see the trend that the patients with severe pneumonia already have more pathogen bacteria. So yeah, it will be very logical if we have follow-up, it will be even more higher. So that's the main point of this project.
Okay, the last questions. Yeah, I mean, follow up for the previous questions. Beta diversity analysis is usually used to check the distance between each species richness between the sample. If you examine by the alpha diversity, just like you cannot see the correlations of the species exist in the healthy and the pneumonia patients.
Yes, so
what do you think?
We actually already also diversity analysis, but we didn't really show it in here because it hasn't finished yet. So, but in the main manuscript, we will also show the results.
Yeah, in preparation actually.
Yes.
This is your last. I mean,
Yes.
then you can't give, but this is the rest of your. You have 40 seconds if you want to add any more points.
Yeah, because of the limited space in the poster, we actually want to focus because we will also have limited time, so we just here focus on this. Thank you.
All right, thank you. Thank you very much. This was a tight match. Please come to the front, and then we can shift on it first while we wait for the judges to finalize. Yeah, yeah, benar.
Oh, tadi kita nggak foto ya?
This arena takes a picture after every ad.
Ya, harusnya gitu. Harusnya dari sana melingkar aja.
Oh iya.
Melingkar aja.
Pura-pura serius nggak?
Iya, oke. Kesini ya, kesini.
Nggak berdiri. Oh, berdiri.
Oh, kita duduk.
Dojo.
Oke, Dojo.
Oke.
Oh ya, oke.
Sama juri dong harusnya ya.
Oh ya, oke.
Oke, 1, 2, sit.
Thank you. And so we have the result. Wow, tied by 1 point. D41 is the winner. D42 at 16. So congratulations, D41, and thank you very much.
Thank you.
D42.
Makasih ya. Tadi pertanyaan diversity belum terjawab nih.
Iya. Belum
Lagi
dikerjain, tapi lagi dikerjain.
Iya, thank you. Lanjut.
Iya, 1 poin.
Rasanya gimana ya? Dari pagi 1 poin.
Thank you.
D41 will stay in this area.
Okay, D41 is still in this area. All right.
Siapa lagi?
We are against?
This is 36 against Ananis.
Ada.
So we wait until
Lulus apa ya? Yang apa? Mau kerja. Mau kerja.
Banyak sih.
Banyak ya? Udah, ya.
Ya, kerja gimana?
Enggak sih, saya punya project kebetulan seperti ini sih kan buat edukasi tentang sequencing, kemudian juga apa? Ya kita banyak pemeriksaan juga mikrobiota kan. Tapi kalau bisa dia juga bisa pipetin gitu.
Oh.
Kalau orang biasa nanti nggak pipetin kan biasanya ya.
Ada sih salah satu peserta apa dari banyak sih sebenarnya.
Banyak ya? Ada aku minta itu ya?
Iya, Prof kasih aja spesifiknya di mana gitu. Nanti kita kasih tahu.
Oke.
Soalnya kita punya banyak alumni magang.
Magang ya? Oh iya, nanti itu ya saya kasih.
Oke. Siap.
Kalau bisa saya dapatnya.
Iya.
Oke, makasih ya.
Iya, makasih.
Udah pindah tempat lagi ya? Apa dokter satu di sini lagi? Pindah lagi. Oke.
D41 stays here.
Lanjutnya di mana?
Udah
selesai. Udah selesai. Oh, asik.
Jadi kita mau
Makan dulu.
Anak break dulu, kedinginan di sini.
Iya, dingin banget.
Nggak dinaikin aja nih.
Oh iya, 20.
Dingin ya.
20.
Dingin banget.
Juga deh. Pada pipis. Iya, dingin banget.
Jadi habis ini istirahat dulu 10 menit buat sholat asar, and then we have one more match, and then this arena closes.
Okay, so 2 more matches.
Nanti setelah match ini kan match 30, habis ini ada match 33. Setelah match 33 mulai mengerucut, jadi arenanya satu-satu mulai closing 3 dan 2.
Closingnya per arena.
Iya.
Wow.
Ini udah mulai mengerucut sih, makanya jadi agak kejar-kejaran sih.
Oh, jadi ada yang nungguin arena kita?
I'm not sure either.
Enaknya sih gitu ya, I think so.
Jadinya bisa nambahin 3.
I'm gonna do that. I'm gonna point that.
Tapi sekali presentasi bisa langsung 3 kali 6 just beda 18.
Iya, jadi poinnya tinggi-tinggi ya.
Iya.
Poinnya tinggi-tinggi. Alright. Tapi ada yang nungguin arena kita?
Jadi so far kita masih in sync sih.
Awesome. Jarang
Ini
ya.
berarti kelompok ibu udah elimination kami. Tadi sempat kalah.
Iya, ini elimination. Tadi kalahnya di sini juga.
Soalnya ini dari tadi sama
kita jadi.
Oh iya, kalahnya di sini juga.
Jadi tadi di sini kalah sekali, di arena lain menang sekali tuh.
Iya. Jadi mereka lanjut. Hebat. Tadi kan menang kan duluan. Satu kali menang kan. Terus kalah. Kalau ini kebalik, kalah terus menang. Seru ya. Double elimination itu seru.
Ini udah termasuk 15.
Seru.
Oh, top 15?
Yes.
Wow.
Seru for the judgment.
Oh gitu. Berarti tahun depan jadi referee sini. Tahun depan. Alright, okay, everybody's here. Alright, so this is D36 versus D38. We're gonna start by as usual, and a little bit of your background, and then we go to the coin toss, and then we continue the match. Please, D36.
Okay. This group from 36. My name is Sriwahdini, and this is Nadia, and we are from Faculty of Medicine, Universitas Indonesia.
Team D38, please.
Okay, we're from D38. My name is Miftah, and my friend is Fabi. We're from Summit Institute for Development.
Fantastic. All right, coin toss. Who wants heads or tails? Heads or tails? Heads. All right. Heads. Do you want to decide?
Present first.
You want to present first? All right. Okay. All right. D38 is presenting first. The presentation will be done in 2 minutes. It can be graded up to 3 points per judge, so it could give you a good head start. I will start the time when you start speaking.
Okay, everyone, we want to present our study titled The Impact of Maternal SARS-CoV-2 Infection on Pregnancy Outcomes and the Antibody Response. Next. So, we all know that COVID-19 resulted in over 7 million deaths, and vulnerable population like pregnant women tend to have experience adverse effect than the general population. So our study enrolling pregnant women at their first trimester, and we got their blood draw taken twice, The first blood draw was at the first trimester, and the second blood draw was at the third trimester. And for the assay, we do qualitative hemagglutination assay, where it is scaled from 0 to 3, where 0 means negative result, and 3 having the highest agglutination intensity. So we find that birth weight has the most, being the most impacted clinical outcomes in our study, where the odds ratio is 23% on adjusted, including other variables. Next. So also we found that 50% of our pregnant women population decreased their antibody at 140 days, where also their maternal age impacting more, where pregnant women with older pregnant women tend to lose more than the younger one. Thank you.
Okay, thank you very much. All right, so now we move on to the question and answer session. Each question and answer can be up to 1 minute, and I will start the time after you ask your first question.
Okay, my first question is, was there any consideration of the potential confounding factors like socioeconomic status, nutritional, access to healthcare that could influence the study outcome beside COVID-19?
Yes, we assess the other factors that might impact the low birth weight. That is, that's why we do like unadjusted regression and adjusted regression. In the unadjusted regression, we only see we only want to know how the the infection of COVID-19 impact the low birth weight, but we understand that it might there is other factors that contribute to the low birth weight rate. So we also assess other factors in the adjusted regression. Yeah.
Okay.
My next question is, include COVID-19 infection status like symptomatic and asymptomatic, because, and also the severity of the symptoms, because I think it will be really on your results.
Yeah, actually, the focus of our research is not that the comparison of symptomatic and asymptomatic. Also, instead of like the real time or gold standard to check if the pregnant woman is COVID, we assess more on the neutralizing antibody, like we do the assays, because we want to know the protection more instead of like, yeah, because we know that they are vulnerable, they need more protection throughout their pregnancy.
Please.
Can you mention the inclusion and exclusion criteria, because it's only mentioned about the first trimester mother? Inclusion and exclusion criteria for your data collection.
Yeah, actually, for the inclusion, we recruit every pregnant women at the area of our work, where at the woman under the care of the puskesmas. All of the pregnant women can join our study, whichever their age are. As long as they are pregnant, we can enroll them.
So, what the design of your study? Cross-sectional or longitudinal? Or
it's a longitudinal because we have like 2 times of sampling.
So when you know if the mother got COVID?
Maybe you want to elaborate.
Yeah. Okay, it's only pregnant woman. Is it should be a COVID positive or not, or something like that?
Okay, so we also actually test for their PCR, actually beside the antibody. But for the antibody, one of the strength of our study is that it happens before the mass vaccine. So we can know that we also know their vaccine history. So the infection means that they have antibody before their vaccine, or they have not any vaccine yet.
Is that your answer? Do you want to add?
So in order to classify a mother being infected or not, we have like HAT result, which is the antibody showing the antibody, but in qualitative way. So we have like the intensity of HAT. Yeah, and then also we also have RDT test. So for the the result is the IgM or IgG. So it is antibody RDT test.
Why didn't you elongate your study duration until the mother had their newborn? Because I think it's crucial after you follow up. Know like what are the effects after the baby is born, maybe.
So we have actually another study to look at that. So we have this is a separate study. So in the study we look at the impact of infection in pregnant women, but in other study we look at that what you mentioned. Yeah.
Yeah, actually we follow up the mother until their baby is pregnant. Actually, until the baby is 6 months old.
This is going to be your last session.
Okay, how about the like vitamin or a history of disease like of mother like diabetic, hypertension, and because it also influence the baby birth weight.
So for the hypertension, we also maybe not all of the mothers assessed for hypertension, but we also have we also collect the hypertension of the mothers from the what do we call that, the KIA. So we also gather other database from the data from the midwives they go to. So that's the source of our data.
Yeah, we actually have complete variables, but when we do the analysis, we try to assess almost every variables, and we found which variable impacted more, and we show that variables.
Oh, sure. One last one then.
Just a comment. I think that's very important thing to be written and to be reported in your presentation.
Last response.
Okay, because this is a limited, we also have a limited time for presentation, so we think that we only need to show the result here. But actually, we have explained about that in the poster.
All right. Thank you very much. That's the end of the first round. It's definitely very tight. Oh wow, it's getting even tighter. All right. Okay, so second round. Usually we give 4 minutes to prepare. But if you are okay.
I just need 30 seconds.
Okay, all right. Okay, 30 seconds. I just start the time for 4 minutes, but whenever you want to start, we can start.
Okay.
So. Wow. These matches are getting even closer and closer. Yeah. First round is there's a gap, but then this round is very close.
Right, I'm ready.
Okay. All right. So again, just to remind, the presentation is at 2 minutes. It can be graded up to 3 points per judge, and I will start the time as you start speaking.
Today I'll be discussing phylogenetic analysis of Sarcoptes KBA in human population, a case study from Jakarta, Indonesia, and global comparisons. So scabies is a contagious skin disease which is a rising public health problem worldwide. So we aim to analyze the genetic diversity of Sarcoptes KBA in human from a boarding school in Jakarta and compare it with mites of various hosts and region. Our research will focus on genetic epidemiology using COX-1 as a gene marker. So we collected samples from students. Next slide, please. So we collected samples from students at boarding school X in South Jakarta. Now, the study has not yet finished. We have 21 samples from students diagnosed with scabies. So 21 positive samples are processed to amplify COX-1 gene and then analyzed with 62 gene bank sequences, and then align and analyze using maximum likelihood tree. So from our result, showed three genetically distinct clades. Clade 1 is from Jakarta and Panama. It suggests uniquely adaptive lineage due to genetic convergence or geographic isolation. It also points at a common ancestry. And clade 2, colored in blue, is a global human samples. It hints how human migration might facilitate the spread of scabies. It also shows that they might have strong adaptability and survival. And finally, clade 3, colored in orange, is the most diverse with samples from various hosts and region. It suggests that might have developed genetic mechanism to evade host defenses and thrive in various ecological niche. Next slide, please. So the conclusion is our study demonstrates the complex genetic relationship of Sarcoptes scabiei. These findings are valuable for developing targeted public health strategy to control the spread of scabies effectively. Thank you.
Thank you. Very nice. Very good. Okay, now we go to the question and answer. 1 minute per question and per answer, and I will start the time as you ask your question.
Okay, so you have 21 samples. How are you confident with that 21 number of samples? Like, what makes your study is like relevant to the global with that number of samples?
We start the study from the screening all of students in the pesantren, and we have count based on the Lawson method. We need a minimal sample about 88. Then from the 88 century, we do the clinic physical examination, and we got about 45 positive by clinical examination. Then we do the skin scraping. It's only about 30 positive by microscopy. Then we just finished about 21 sample for the molecular.
So from those all the screening one, you assess only 88 or?
Based on the minimum sample that we need, only 88 sample.
So you
based
only
on
assess
counting also.
88.
Yeah,
So,
88.
but in statistical analysis, statistics we have like a margin of error. We have to count a number of loss to follow up or something like that.
Because this is cross-sectional, so we don't need to
okay.
drop out.
Okay, so the base student in a pesantren, right? So how is that gonna be represent like the population? Because you started with a small area, it doesn't like represent like for how it will be more have impact for the global, because you started within the pesantren instead of getting data from the whole Indonesia.
Perhaps so kind sampling, active sampling that we goes to a population, or passive sampling that patient came to the hospital or first FASCUS like that. So this is one kind of the active sampling to know the prevalence in one kind population. Well, the population is a pesantren, not people in global.
But you need to have like global impact for your title, because it said that how it will be impact compared to the like other areas like China or something, but you only assess that specific pesantren, and it doesn't really represent Indonesia or something that you want to have impact on.
It doesn't represent Indonesia as like as a country, but it is a novel. This study has its novelty, which is there is no published data on Scabies farhominis in the Gene Bank or any other published articles elsewhere. So this will be the first. So it is a step in the right direction, I would say.
Okay, regarding your result about the 3 groups you have here, grade 1, 2, 3, how do you decide the cutoff for each group?
So with maximum likelihood method, the cutoff is numbers 70 of the branch. If you can see like in a phylogenetic tree, there would be numbers, right? So in maximum likelihood, if it's the number is below 70, then it's not very reliable, the branching. But if it's more than 70, it is stronger and more reliable, and we can say that it really the branch out is really you can trust it.
Okay. You said that maximum likelihood, right? To divide to get the cutoff. What method that you use? I mean, maximum likelihood is the method to do estimation actually. So, what specific? Yeah. So what? Why do you choose maximum likelihood other than other statistical method to decide the cutoff?
So for making a phylogenetic tree using sequence data, maximum likelihood is the most robust one compared to neighbor joining tree.
Okay, you mentioned robust.
Yeah, more robust.
What do you mean about robust?
Like it has replicates. We use replicates of 500.
Mm-hmm.
We use replicate of 500, and we also test which model will fit best.
Usually, robust means when there is outliers, your method will not affect by the outlier. That kind of robust. So, when you mention robust,
it's more reliable, like the shape
I think
of
it's
the phylogenetic
contradicted with the.
Please continue your explanation first.
So why is that robust? I think it. Can you repeat the question again, because it's kind of.
You mentioned it is robust method, right? What do you mean by robust?
What I mean by robust is that it has a high replicates, and it is more trustworthy compared to the other methods of making a phylogenetic tree, such as neighbor joining tree. When you use neighbor joining tree, it also only used like algorithm to make that.
Oh, okay.
This is your last session, so be careful.
I think I want to
Yeah, go ahead.
answer about the.
Yeah.
Okay, so in statistics term, robust means your method is not impacted by any anomaly in your data. That is robust. Usually, it means in terms of statistics. Actually, yeah. So I think your answer.
You need to make it more detailed, like if you have some kind of terms, so it won't be another meaning and it won't conclude to other misinformation.
Please respond.
There's no question in that.
No, there's not a question.
There's not. You have anything to say? No. I'd say that I don't think it compare. I'm not a bioinformatician, so but I don't think like it's very comparable to statistic. Like we don't have any outlier or anything like that. We just know that if the branching have a node of number of less than 70 or more than 70, then we can trust it. It is it shows the number of confidence, and if it's below 70, and then we just think that it's not reliable and the samples are the same. There's no branching out.
All right, thank you. And that is the end of round 2. I think we need to wait for the judges to come with oh, you're done? All right, is this the last synchronized results? That means we need to go to overtime. In overtime.
I always have overtime.
Yes, you always have overtime, which is pretty good, right? All right. In overtime, basically, it's 2 minutes, 2 minutes. So in this case, questions will come from D36 for 2 minutes, and then another one, 2 minutes, questions from D38. I will give the chance to D36 first, and I will start the time when you start asking the question.
Oh, do we have more time to discuss first?
Yeah, 1 minute.
1 minute.
Yeah. My suggestion: change the topic. My
Yeah.
suggestion, yeah, change. You gotta discuss about other things, or it's just gonna be equal all the way. Yeah. And like, ask a question. Do not give points to your opponent by not asking a question. Right, 15 seconds. Okay.
Is it
5,
mine?
4, 3, 2, 1, and so the first round of this overtime questions will be from 336. Answers will be from 338. All right, this way.
How did your study account for variation in the timing of COVID infection during pregnancy and its potential differential impact of pregnancy outcome?
A very complete question, please.
Can you elaborate more, like what is timing you mean?
The timing of COVID infection during the pregnancy. Do you account that in your analysis or in your study design? Because it might have differential impact on the pregnancy outcome, the baby. Like, you know? Do you understand?
Do you count when they actually got COVID?
Okay, so we're not counting, we're not assessing along the pregnancy of the mothers, but we assess it in the baseline. So we conclude it as a recent infection or past infection by the IgM and IgG, and also for the based on the seroconversion, and then we have baseline H80 and end-line H80, and we compare the end-line and baseline. If it is, for example, it is gain antibody, then we say, and we make sure, and we also have like historical vaccination data from the mothers, so we know that if the mothers have vaccination history or not. So we can make sure that if the mothers gain antibody and there is no vaccination in there, we conclude that as infected, something like that.
Last question.
In your conclusion, if I'm not if I'm not wrong, you mentioned about the older woman will give the seroconversion in antibody of COVID. Is it right?
The mothers will lose antibody faster if they are older. Women.
Why it can happen like that? How did it happen? Why you can conclude like that? Only based on the statistical or any?
Based on the statistical, based on the, you can see in the Kaplan-Meier curve, it is showed that the red line will have like lower in a lower place, and it goes faster to the end of the line, but for the mother, like a younger one, will have like more slower to go to the end of the line. So that's how we know that it has faster loss of antibody for the age of
Is that what you understand from our question?
older.
Is that what you? I mean, just is that what you understand from our question? All right. Okay. You have 10 seconds if you want to add.
Yeah, and we also assess for the using the Cox regression, and it is the p-value is lower than 0.05. So we conclude we know that it is significantly different.
All right. Thank you. Now, questions from your side? Answers from yours. 2 minutes. I start when you ask.
What really makes your study important? Because I don't really see you have like reference on how like what is the benchmark of your study will become successful or will be somehow impact have global, like you mentioned, it will have more global impact, because being the first doesn't always mean you're best at it.
first, we don't go for global impact, but it's about the clinical manifestation, which is can. I think you should explain it.
So the clinical manifestation, like a papule nodule or maybe, yeah, crusted. But in Indonesia, most of the student only mild one, not the severe. Severe like a crusted scabies.
I would add. Do I have more time?
Yes, you have 20 seconds.
I would add that we would connect and make the relationship of the clinical manifestation to the genetic diversity, the result of genetic diversity that we have. Because there are lots of cases of recurrence, reinfection, in our student population, because we have. That's all.
In your title, you mentioned about global, and also you compare like several areas outside of Indonesia. But I don't really see what is your benchmark of your successful study would be, because it's still scarce. It's still unknown when will your study will be called like successful. Yeah.
Can I ask you, what do you mean with successful study?
I think that's her question basically. Like, how do you measure your study to be finished or successful? Yeah. How do you answer that?
Because we want to make molecular or genotyping of the mite, and we think we can, when we can get the mite and we analyze the phylogenetic, that's success, isn't it?
Last question.
But when it feels like when you can do something that it doesn't have like reference or benchmark, is it really successful, or is it really important, or is it really useful because only you feel that, oh, finally we can do this?
Is that your question? All right. Okay, that was the last one. So please.
I think in genetic study, you gotta start somewhere. So this is also, like I said, the first one of the study that studying the genetic diversity of scabies in Indonesia. So like I mentioned that we wanted to make that relationship between clinical manifestation and the genetic diversity of scabies in Indonesia. And if we can make that, that would be a successful study, because it hasn't been, no one have made that connection elsewhere, and it also would impact heavily on the control, treatment, and surveillance of scabies in Indonesia.
20 seconds. You finished?
Do you have more to say?
All right. Thank you. And that is the end of the overtime. No? All right, then please come forward and then we can do our closing ceremony. So with that, congratulations to Team D36 for continuing. And thank you very much to Team D38. I think it was a very lively, discussion. Picture. This arena like
Yeah.
Okay, thank you. All right, that's great. Okay, 10-minute break, and then on to the next one.
You guys go through the next round. Okay, great.
Yes.
Okay, great. That was actually good. Nice work. All right. So with the last question she was asking you about, why is this, actually what she was asking was not so much does that statistical loss of antibody, is it just, it's not so much asking is that what your analysis shows, but asking is it plausible? Like what's different biologically?
Hence I try to.
Yeah, what's different biologically about an older woman that could be true?
That's why I was like, is that what you understand? D51. Wow, this is intense. Let's go, Dr. Christie. Let's go, Dr. Christie. D41. D51. Ini ada elimination nggak?
Iya, berdua sama
Oh,
saya.
both of you are elimination.
Iya.
Oh, okay. Intense. All right, D41, D51. On my right hand side, we will start with introductions.
Okay.
We'll start with introductions and then coin toss and then presentation. D41. D41, please start with the introductions.
Okay. Good afternoon. I'm Deli and my colleague, Dr. Selvi. We are both from Department of Microbiology, Faculty of Medicine, Universitas Indonesia. Thank you.
D51, please.
Hello, we are from team D51. My name is Christy. I'm pediatric neurologist from UGM Academic Hospital, and this is my colleague, Dr. Aulia. She is a pathologist from Dr. Sarjito Hospital. Thank you.
All right, and for the coin toss, heads or tails? Heads or tails? Head. All right, head. Heads for D41. And heads. So please decide whether you want to present first or let them present first. Oh, they will present first. Okay. Okay, please prepare the presentation for D51. And all right. It's the same. It's 2 minutes. Welcome back to the arena.
Maybe it is boring already.
No, definitely still exciting.
Okay.
So I will start when you start speaking.
Okay, our study is about exome sequencing in personalized treatment in DEE patients, transforming national clinical practice guidelines in Indonesia. Next slide. DEE is a most severe end of epilepsy spectrum, where there are severe seizures and cognitive and behavioral impairments, and it happens since children or childhood. There are a lot of genes responsible for the mechanism of DEE, more than 400 genes, and if we know the underlying mechanism, we can transform clinical diagnosis and management. Current guideline using empiric therapy based on the type of seizure, which may be the contradictions of underlying mechanism. For example, focal seizure, we should give carbamazepine. But if we find that the patient has SCN1A mutation, if we give carbamazepine, which is the same natrium channel blocker as SCN1A, then the seizure become worsened. So the study aims to look at the genetic profile of DEE, directing patient therapy and management in Indonesia. The DEE patients undergone the phenotyping, and we did the whole exome sequencing to know the genetic etiology, and we make personalized treatment based on the gene we found. Next slide. We identified the genetic etiology in 47% of patients, and the most common genetic etiology was SCN1A, which responsible for Dravet syndrome. So we know that we have wrongly give the medication of our patients. Management changes was conducted in 9 patients, 7 drug changes, and 2 avoidance of brain surgery. Next slide. This study illustrates that whole exome sequencing in clinical setting in Indonesia is feasible, has high diagnostic and clinical utility, and can modify the treatment for DEE patients in Indonesia. Thank you.
Thank you very much. And now we come to the question and answer session. 1 minute per question and per answer. And I will start the time after D41 starts asking questions.
Okay, so thank you for the presentation. So DEE is very severe, right? You said that. So it is inherited. If yes, do you think can we use this approach for the sibling of the patient that already have the DEE for the siblings to prevent?
Yes. Yes, it's actually one of the aim of the study. If we know the genetic disease, and then we can do only Sanger sequencing for the siblings in order to know whether he has the same genetic variation with the patients. Yeah, but it's not always inherited. Most of them are de novo mutations. Yeah.
And then how you stratified the patient? For example, I think you have 50 patient, right? So in which patient that probably you will have the prevention, for example, for the siblings. I mean, is it possible to stratify which patient will if it's they have siblings, then you do the prevention, you give the prevention.
So I will look at the patients one by one. So yeah, for instance, so it's genetic counseling. So if we have a patient, then we will consult the family. So and if we have like patients, and then we can tell them if they want to do the genetic analysis for the siblings. Yeah.
So whether the gene is de novo or it is inherited, it's not based on the genes, but it's based on the individuals.
Yes. If we found the VUS, for example, then we will take a look at segregation analysis for the parents. So if we have patients, for example, homozygous inheritance, and then we check the mother and the father, if they have both genes heterogeneous, and then we can predict the next siblings to come, right? So yeah, we do the genetic counseling in each patient, and in each gene, whether it is autosomal dominant or autosomal recessive or X-linked, etc.
Thank you. And I have a question about the, you mentioned in the title that you will change the clinical practice guideline because of these results. How will you change the guidelines based on this?
Yeah, it's difficult, but we are having the UKK, the National Working Group in Pediatric Neurology, and this study is only in our referral center. But I think multi-center study will have more impact to convince our working group to change this approach in managing our patients with DEE.
Yeah, well, for example, when patients come to the hospital, at first maybe he only have seizure and we don't know whether it is DE or not. And then when should we test for this exome sequencing?
Yeah. We have the inclusion criteria. Maybe you want to.
Yes, the patient is seizure is more one time, and we see the MRI and EEG. This patient.
So there is a diagnostic criteria. It has to be intractable, it has specific EEG, it has developmental delay, and it has changes in MRI. So that DEE is included in next for genetic testing.
It is like quite long pathway for diagnosing the patients. Yeah. So like, for example, when a patient got seizure and then the patient gets seizure again the next time, we are not usually directly ask the patient to do the MRI, right?
It depends. For intractable epilepsy, if you already give more than 2 drugs for the patients, I will go to MRI because I want to see what happens in the brain. But if only 2 seizures and I give 1 drug and it's over, I don't go to MRI.
Oh, yeah. So that's how you will change the guideline,
yeah.
I think.
How you collect the samples in this study?
So we took all the samples, came to our hospital that meet the inclusion criteria.
Yeah. Okay. So you said 50 samples. So and will make the national guideline, right? So how it is reflects all Indonesian?
It is not reflect all Indonesian, but I think 50 is quite a good number because this is a rare disease, one in every 1,000 patients, and we have compared to Australian national study, they use 100 patients for national Australia. So I think 50 is quite good number, but you are right, maybe multi-center studies reflecting our big ethnicity would be better to give more evidence.
Yeah, because we are multi-ethnic. Yes. So because we are multi-ethnicity right here, so it is west and central and east part in Indonesia is very different. So how it can be, so how to transform make it translation into the national guide.
Yeah, thank you very much. But based on our result, SCN1A, SCN2A, KCNQ2 is actually comparable with other international studies in different ethnic groups, including Caucasian and Asian. Although we haven't looked at the our ethnicity, but I think that's beyond ethnicity because every nations in the world has almost the same SCN1A and etc. Thank you.
Because of the speed, you have one last one.
Okay. Do you find novel mutations in your whole exome?
Not yet so far. So it's same with the international mutation.
Yeah, in that.
Sorry,
Okay.
yeah, that was the last one. Do you want to add? You have like 20 more seconds.
Yeah, there's no novelty. We find ultra rare disease, but it has been reported. But we find also the same gene, but novel mutation we found, but the same gene, not the different gene.
All right, thank you. And that's the end of round one. Thank you very much. Okay, so then we continue to the second group. Do you need time to prepare, or do you want to just go for it? All right, I just want to go for it. All right, okay. Please prepare a presentation for D41. And then presentation for 2 minutes. Remember, they could be graded up to 6 points because it's 3 per judge. So then we can have a good jump start to continue. Please prepare the presentation for D41. Okay. Pit stop. Pit stop.
Nilai-nilainya nggak terrecord tuh.
Alright, presentation for 2 minutes. My timer will start as you speak.
Okay, thank you. Our project entitled Sputum Microbiota Profiles in Hospitalized Adults with Community-Acquired Pneumonia. So, imbalanced bacterial composition or dysbiosis has play a role in the incidence of community-acquired pneumonia or CAP. So, in this study, we would like to identify the etiologies of the CAP and the microbiota profiles of patient with CAP. So, this is the workflow of the study. To confirm the etiologies, we did culture and real-time PCR, and then for microbiota profile, we did 60N sequencing. Next. So after collected the sample, we divided the patient into 2 groups, mild, moderate, and severe patient, and then we checked several factors that influence or contributing for the severe CAP. And we found in the study only age in the patient with more than 65 years old statistically significant of having severe CAP. So most of the patient here are polymicrobial, followed by bacteria and viruses, and a single pathogen. So for the microbiota profiles, there's no statistically significant different in terms of the alpha diversity between 2 groups, mild, moderate, and severe. However, we can see the trend that in the severe group, the alpha diversity higher compared to the non-severe. And in Spearman correlation, we also find that in severe cases, dominating by the pathogenic bacteria. So in conclusion, CAP in adult was mostly caused by multiple pathogens, and patients older than 65 years were 6 times more likely to have severe CAP than younger patients. And patients with severe CAP had higher alpha diversity, but had more pathogen and fewer commensal bacteria. Thank you.
Thank you very much. And now for the question and answer. Now we'll start the time as you ask your first question.
Thank you. Your background say reported correlated taxa in the sputum sample from infant and children. From your poster.
Adult.
Yes. From your poster say
adult.
Oh, so this is wrong. Yeah.
In your poster, you mentioned in the background, you took the sample from.
No, this is the no. It's the background. So we give the.
You use the adult.
Yes, it's study adult.
Okay, so I think the background also must be written in adults, maybe only like that.
Please clarify.
So because we would like to say that this biosis will influence the incidence of the CAP. So we put several studies that conclude that. So it's including in adults and also child.
Okay, so continuing to the questions, would your result will be different if we conducted the study in children?
Actually, the previous study from Pettigrew in 2016 showed that similar results from children. So the difference is we use sputum in here because adults are easier to sputum samples, but in children they use usually either saliva or buccal swab.
Yeah.
But the results actually quite similar.
Yeah, but it is more difficult to take samples from the children, right?
Yeah.
Okay. So I also read in your poster, you have to culture your patients before you do the real-time PCR. Is that correct?
Yes.
Yeah. So actually, for me, clinician, the importance is the time, because for culture it needs 7 days.
No, no, it can. The result can come out in 2 days.
Okay, so usually it's 5 to 7 days in my experience, and the patient is already become very worse. So what I want to is as soon as possible the result came out, and I think PCR is very fast. And what do you think if we cut the culture and directly to the RT-PCR? Would it be okay? And can we get the result sputum to PCR?
Yes, but we know that in PCR you cannot differentiate between live and dead bacteria. So as long as the DNA is still there or the genetic material is still there, it can be positive. Why we still perform culture? Because in Indonesia there are so many studies, especially in CAP, that mostly caused by bacteria. So it's still culture. Yeah, we still doing that, and we in the era of antimicrobial resistance now, culture is very important.
Yeah. Thank you very much. So. Like in clinician, 2 times 24 hours is the most. We have to change the antibiotics within 2 and 24 because the patient then degrade it very fast and then die. So we need a machine or a technique to solve our problem, because sometimes we just wait for the microbiologist to release the result.
Yes, of course. So actually, like the respiratory panel multiplex PCR, as we perform actually already in a clinical setting right now. However, for that we have to perform culture as well in parallel. So when the study conducted, actually it's not already applied in our hospital, in our setting. So we just perform the culture. However, when we want to continue with this study, we also perform the multiplex PCR, so we can really get more info about the pathogens.
So you do it parallelly?
Yeah.
Parallel.
Normally, when you have both measures, you can do both at the same time, and then you can use the multiplex PCR results in the beginning for altering your empirical therapy, and then after that, when the culture results show up, you can also change it to the definitive therapy.
Okay, so in your study, why you do the culture before?
Yeah, that's the reason, because like in the beginning, when we have not performed this as a research, we just do what the diagnostic in daily
Oh,
practice.
okay.
So that's but when we want to perform the microbiota analysis, we need to really get the more info, because like culture cannot cover some pathogens, and we would like to know more about that.
So it's not. So it's not the DNA amount that you do the culture first because before the RNA. You don't do the culture to make your DNA amounts grow. One last questions. Oh yeah. Why you do multiplex PCR? Because multiplex PCR has a number of bacteria and virus. How about novel or undiagnosed bacteria? Do you have another techniques?
So actually, in the panel system, they put the microorganism that commonly the etiology of the. So for example, the respiratory panel, they will put the pathogen that corresponding with the respiratory infection. So the composition of the panel test will be different in different infection.
Yeah, and in addition, also in the respiratory panel, usually it also contains the resistant genes. So you can already alter your therapy for the patients based on the PCR results.
Okay, so how about the unidentified bacteria? So, I mean,
You have 5 undiagnosed bacteria. Yes.
Unidentified one.
Unidentified. Yes.
Yeah.
How do you think about that?
We can actually perform the microbiome analysis, but for example, we cannot really know which one will be the pathogen, the true pathogen of the patients based on that results itself. So logically, for clinical practice, we need to consider the microorganisms that usually being pathogens.
Sorry, but time is up. All right, thank you very much, and that's where the round ends. All right. Do you need time to consolidate things, or are you just doing this out of spite? All right, okay. Because of the draw, we need an overtime of 4 minutes basically, so 2 minutes for D41 to ask questions.
One suggestion, and there was a couple of line of questions that were back and forth, but it was basically the same line of question. Our suggestion for this overtime is try to find a new question, so not related to your previous question.
Yeah.
Oh, okay.
New line of thinking, new perspectives. All right, we have 1 minute to prepare, and then Dr. Christy is also on overtime. Also on overtime. Extra 2 minutes aja, Dok. Extra 2 minutes. All right, ready? Okay. Questions from D41 first, while D51 prepares answers. And let's start.
Okay, thank you. So for your the DE is actually a rare disease, and well, the variety of the mutations in the genes may be very large. And how did you perform the external validity?
So yeah, our mutated gene will be analyzed also by the pediatric neurology, and we look at the oming, the phenotype of the gene, whether the phenotype for that gene is matched to our patients. Then we have we confirm that the patient has the right gene. Sometimes we get the gene that is very different to our patient's phenotype. And secondly, we will do the Sanger sequencing for the patients and also for the father and the mother.
Okay. And then previously you mentioned that you haven't found any novel mutations in the genes, no novel genes. Then is it possible to also just perform the targeted sequencing instead of exome sequencing?
Yeah. Yeah, because we don't know. We have only 50 patients. We don't know if the patients comes much more than that. And because we have 400 genes already added to the DE responsible gene, and the data list will be come bigger. And I think for 400 genes, it is more useful to use the more useful to do the whole exome sequencing than targeted panel.
Okay. In 28 patients where the pathogenic variants not found, did you look for the novel mutations, or which therapy did you give to those patients that you cannot diagnose?
Yeah, we considerate several other treatments such as epileptic brain surgery, and there are also vagal nerve stimulation and deep brain stimulation, but all those therapies is not readily available in Indonesia. And for the bioinformatic analysis, we are doing reanalysis to know whether there's new mutations or new reports from other region in the world found the mutations. Yeah.
Finish on that. So now questions from you and answers from them. Please, Steve 51.
Yes. Based on your study, do you any suggestion for the examinations so it will be more effective and efficient?
So for the diagnosing of community-acquired pneumonia, as I mentioned before, we suggested to use the multiplex panel-based PCR, but also in parallel doing the normal culture of the bacteria.
And how about the cost examination for this study? It's feasible for the JKN patient?
For culture, yes. I think it's in some region PCR also covered by the JKN, by the national insurance.
And how about the multiplex?
Yeah, which one would you choose between multiplex PCR and metagenomic?
It is quite different because like the multiplex PCR you use to confirm the diagnosis of the patients, so you found the etiology and you can treat the patients. However, in the microbiota analysis, you see how the imbalance in the patients. So for that, actually like maintaining the health associated homeostasis between patients and their commensal being very important based on this study. And if we want to adapt to it and then interfere the patients based on the microbiota, we need more data about that.
Yeah, I mean, in the clinics, there are a lot of type of patients with human immunodeficiencies or other genetic disorder that may have another not usual microorganism happen in these patients. So how do you prepare to detect that kind of unknown microba that is not listed in your multiplex PCR list?
Yeah, thank you. Okay, yeah, you can use the shotgun metagenomic approach for that, but in the cost-efficient considerations, we cannot really do that in daily practice.
You have 20 seconds. Or do you want to give it to them to respond? Last response then. I mean, last question then.
So, how do you think the next examination for the unidentified patient?
We are actually conducting more study about that. So for patients, we only split patients that with infection from unknown origin. So patients that has not detected the etiology, we can continue with the metagenomic to really seek for the pathogens in the patients. However, it's not in daily practice. So it is the second line diagnostic after the first line in clinical settings.
All right. Stop. Judges, please. Judges, please. Anything? Do you want to consolidate something?
Is it the same? Still the same?
If not, we're going to sudden death, which means whoever asks the question that you're not satisfied, then it's over. Yeah? Is that okay?
That's a big responsibility.
It is. All right, let's do a coin toss on whoever gets the coin decides whether they want to ask the question or they want to answer the question. All right, heads or tails?
Kita nanti
berdasarkan hasil toss.
Jadi cuma sudden death aja dok. Jadi ini kita coin toss nentuin apakah yang menang mau nanya atau mau ngejawab. Whichever yang nggak satisfied oleh judges udah itu yang kalah gitu, sudden death gitu.
If the question is not valid,
Huh?
if the question is not a valid attack, then that's.
Yeah, then you decide. Then well, that's not good
It's
enough.
unlikely that.
Okay, heads or tails?
So the
Huh?
first tail.
Tails, alright. Angka ya. Okay, it's heads. So you decide whether you want to ask the question or you want to answer the question.
Answer the question.
Answer the. So the question should be from you. So the judges can also decide that if the question is not valid, then that's a sudden death. Okay, 1 minute ya, 1 minute to prepare. All right. Yeah. Here, I show you the time to increase the tension. Please go back to the first one.
Lagi balik.
Udah ini. Oh, oke.
I don't have any idea.
Don't give up, Doc. Don't give up. All right.
Okay.
Ready?
Apa ya? Nah.
Okay. 5, 4, 3, 2, 1. Please. Team D51.
So how do you suggest the clinician if you mention unidentified microba is based on your result?
Wait, judges?
It's already asked. Yeah, it's the same question actually. Yes.
Congratulations, Cindy. Please.
Makasih ya.
Sama kayak gitu. Masih punya list pertanyaan nggak ada jawabannya. Udah nggak ada.
Come, Dr. Safi, Dr. Dari.
Oh iya foto ya.
Yeah,
Oh iya foto.
in this room we take photos. That room, I don't know why they don't.
Oh iya, nggak ada.
I feel like this room is a bit more serious. I gotta do this new one. Since this is the last match in the arena.
Oh yeah. Sorry,
Yeah.
it's not the last one.
Oh, it's not?
It's not the last one.
But it's okay.
Oh man, they don't want to. Alright, it's okay.
Ayo
Oh,
dong.
okay.
Agak ke depanan.
Oh,
Yes, on.
So there's another match here after this?
Yes.
Okay.
Okay, 1, 2, 3. Christina, I'm sorry, can you move? Okay, 1, 2, 3. 1, 2, 3. Okay,
Satunya, satunya.
Okay, 1, 2, 3.
Dojo.
Dojo. Thank you. Thank you.
Finally.
Who's next? Udah, who's the next team?
So I've just been told that we are absorbing the last 5 match of Arena 1.
Okay.
But I don't know if the judges are.
Oh yeah, judges
Are
are
we changing judges or are we adding judges?
Adding judges.
So yeah, we're integrating 2 rooms.
Everybody will come here, right?
Oh.
Everybody will.
3 judges in the room.
Number 1. Oh, everybody will move.
Right. The number 1.
All from Arena 1 move here. Alright.
So switch match 34. Right?
Okay. Am I reffing or is the I'm reffing? Alright.
Yeah,
Okay.
probably. They want to switch part with
Paham? Oh gitu. Oh ya. Tapi emang masih sekarang kerja. Ya udah coba aja datang dulu terus ya.
16 lawan 36.
Oke, gua arahin sini ya.
Ya.
Semuanya dong berarti.
Iya, benar.
Bapak dari berapa? Dari 16. 16? Ibu? 36. Oke, 16 juga. Oke, baik kita mulai ya. Langsung aja ya, kan udah tahu ya. Ada mikrofon? Di depan aja dulu ya, pinjam dulu ya. Okay, let's start for this session. As usual, I'll do the toast. Which one do you want to choose? Head? Number? Okay. Apa tandanya? Tandanya match. Oke. Number. Yeah. You first, yeah. Okay. You wanna present or? We want to present. Present. Okay.
Right. Hi everyone. Today I'll be discussing the phylogenetic analysis of Sarcoptes scabiei in human population, a case study from Jakarta, Indonesia, and global comparisons. So scabies is a contagious skin disease which is a rising health problem worldwide. Our study aimed to analyze the genetic diversity of Sarcoptes scabiei in humans from a boarding school in Jakarta and compare it with mites of various hosts and regions. Our research will focus on genetic epidemiology using COX-1 as a gene marker. Next, please. We collected sample from students from boarding school X in South Jakarta in 2023, and students that were diagnosed with scabies. So we collected total 21 positive samples that were processed to amplify COX-1 gene, and then we analyzed it with 62 gene bank sequences, and then we align, and then we built the maximum likelihood tree. So the results showed that there are 3 genetically distinct clades. Clades 1 is a human sample from Jakarta and Panama, and clades 2 is a global human sample from Australia, China, and South Korea, and clade 3, now colored in orange, came from diverse hosts: humans, dog, wombat, water buffalo, and locations. So the conclusion is that clade 1 might suggest uniquely adapted lineage due to genetic conversion or geographic isolation. It also points at a possible common ancestry. While clade 2, it hints at how human migration might facilitate the spread of scabies. It also shows the strong mite adaptability and survival. In clade 3, it suggests mites may have developed genetic mechanism to evade host defenses and thrive in various ecological niche. In conclusion, our study demonstrates the complex genetic relationship of Sarcoptes scabiei, and these findings are valuable for developing targeted public health strategy to control the spread of scabies effectively. Thank you.
Okay, let's move to the Q&A session. Are you ready? Timer? Ready? Yes, you can start, Pak.
Yeah, thank you. So the initial sample, there are 21. Would you actually cluster this 21 belongs to which clade? Because there are 21 students, right, from 21 samples, and you say at the end you have 3 clades. But for this 21, which one they belong to which clade?
The clades with Panama samples.
All of them in the Panama samples?
Yeah.
But I don't understand. Go back to the previous now. So initially I thought the idea is you're going to cluster these 21 samples into different clades. So when you look at sample number 1 to sample number 21, for example, if I'm going to ask you, so which one is sample number 1? Because the dots here, sorry. So for each one representing one sample, is that right?
Yeah.
But there are definitely more than 21.
Only 21. 2 sample from Panama. 3 sample from Panama.
Okay.
Then we conclude that Indonesia from this boarding school, one clade with Panama is in the clade 1.
Right. What it will be very useful if we can understand out of these 21 students, the distribution of these 21 samples, which one belongs to where? Is it all of them belongs to one clade? Because if that's the case, then you should mark, so say clade 1, right, is red. So that means it's this one, right?
Yeah.
So that means all the students is actually in this clade. But then we have to know which one is sample number 1, number 2, number 3, until 21. Can you confirm that this all belongs to sample 21?
Hold on a second. Do you understand what that is? Do you understand what that is?
Yeah.
Do you understand? Okay, can you explain it?
No. The number, right? Like there is the identification, the identification number. This one, like here, number 5, and number 6, number 19, 18, 14.
Is it belong to 21 students?
Yeah.
Okay.
Okay, if that's the case, then you should actually, basically the conclusion is very simple. There's only one clade in Indonesia, in Jakarta.
Yes, in this boarding school.
In this boarding school.
Yes.
But you say actually 3 clades.
The 3 clade is from the reference, SkyBS divided into 3 clade, not subtype SkyBS.
Okay, so that's why you have to be very clear for the conclusion. Pak Chandra? Okay, so if that's the case, then we have to understand. We have to understand also the significance of this, because this one from the one single school, how generalizable this to other outbreak, for instance. Can we say that all Indonesian cases of scabies outbreak will belong to clade 1 in Jakarta?
No, we cannot say that.
Okay, if that's the case, then what is the purpose of this study? Just only to solve Jakarta issue, or you want to actually, because at the end you want to say the implication, right, for health. Okay, next slide is for the health intervention. Is that right? No.
Yes.
Okay. Yeah, for public health to control the spread of scabies effectively.
Yeah.
How your result can do this,
I got it. So this study is still undergoing, and we want to connect the relationship of the clades and its known mechanism to the clinical manifestation that we found in Jakarta sample in the school students. So maybe Dr. Sri can explain more about how the school student may, a lot of them have some recurrence.
Why we do this in this pasantren? Because we have to follow this pasantren since 2015. Every year we give the education and the medication, but the infection is still positive. Yeah, it's a recurrent infection in this pasantren. That's why I want to know, is it any correlation between the clade to the history of the disease in these children? But what we get is it's only one clade in this pasantren.
Okay, so the original thought is that it should be different clades, but your data show apparently is only one. Okay, so how is going to affect these public health strategies?
It will give us an idea from the further, hopefully, this is our hopefully, it will give us, in the future, will give us an idea on how to tackle this problem with published references from around the world, from samples around the world.
Okay, isn't it as easy as to make sure they have good sanitation? You don't, in a way that you don't need to do this kind of testing. A good old sanitation practice would solve the problem, unless maybe if you show that certain clade has more infectious or something like that. But from this data, can we actually make that kind of conclusion besides just a simple public health strategies?
No, I don't think in reality it is not as easy as that. To control good public sanitation, we already done that in that boarding school, as mentioned before with Dr. Sri, that we have followed this school from 2004 for 2015, and it's still going. Recurrence is still often happened.
Oh, okay. So this 21 students is taken from one time or several times?
One time.
In when? 2000,
Two-
2023.
2023.
So, and, oh, okay. Oh, time's up.
Okay.
Is there 3 judges?
Yes.
Okay. And did we all already put in the presentation score?
You should.
Yeah. Or yes, I already put that. Just making sure.
Okay. So let's, yeah, D16. Okay. Ready? Time? Start?
Yeah. Bismillahirrahmanirrahim. Allow us to present our work, a detection of DNA methylation from buccal swabs using next-gen sequencing to study stunting. So stunting is still a health problem in Indonesia, 21%, but in Pandeglang it's up to 30%. And right now we understand that chronic malnutrition is a major risk factor to lead to stunting. And one of the proposed mechanism is because when you lack in nutrient intake, then it's going to affect the content of methyl donor. And methylation is important to reprogram the genetic in the babies as they grow into a healthy adult. So we wish to understand the epigenetic marker that have been associated with stunting using buccal DNA, because in the past, study of epigenetic actually using a blood DNA, which is very invasive in children. And we also use a very innovative method of NGS that they can detect native DNA methylation, which is actually easier than the traditional one, which requires a chemical conversion is going to degrade the DNA. Next slide. So in 2022, we collected 230 swab, and 30% of them are stunted. So in order to run this technical protocol, we collect the 12 top highest concentration DNA, and which is actually consisted of 6 normal samples and 6 stunted children. So using this, we run this NGS, categorized into 2, stunted without stunted, and we look at the 5 different loci of a different region that using this nanopore, looking at different genes. So next slide. So we for this data, we based on the few loci that has been published earlier. For example, the growth hormone IGF-2. If you look at the red arrows, there are some differential methylation. So the green lines including the extent of methylation, which is less in the stunted compared to the normal. And we also see similar patterns in other genes. And this is actually very encouraging because we can tell the differences between normal and stunted, which actually corroborated by previous literature. And our contribution is to use less invasive samples. We don't have to use blood. We can use...
Okay, D36. Are you ready with the question? Okay. Time.
So I still don't quite understand what is the clinical relevance of the observed differential methylation at the H19 IGF-2 locus in the context of stunting.
Right. Thank you very much for the question. So IGF-2 has been implicated previously. The methylation has been differentially seen in cases in the studies in Dutch famine, hunger, and also in the Chinese hunger studies. So in these studies, when the mothers don't get enough nutrients, then they establish that the IGF-2 methylation is less. And that's because the problem with the growth. Because the environment is detected being less food, so the programming of the baby's DNA is to make sure that they don't grow tall, for instance. But the problem is, this actually could be quite a serious one. If the child grow up in the abundant food, then they may not grow tall, but they may grow wide, because this is actually becoming an issue. And the reason we use also IGF-2, H19, because they have been established previously. So the difference is that we use actually techniques that is less invasive.
Okay. What is your study end goal here? Could the finding have potential therapeutic implication for addressing stunting?
Yeah, that's actually a good question, because right now, because this part of the 7-year prospective study, so in 2022 we take the baseline, and in 2023 we took the same children, and we found that 25% of them actually going from normal into stunting. So this actually studies to track these children, whether any intervention can also changing the dynamics of the DNA of these children. So in order to do that, we need to get the baseline level first, the baseline of this methylation in these children. So definitely we're going to expect whether the patterns we see here, the differences, in this actually an aggregate, is also going to change as the children has a different outcome. Because that's going to challenge the current notion that epigenetic is changing, but we want to show whether actually this changing is really relatable to nutrition intervention, and also whether they can be looked to predict also another issue of the risk of diabetes, the risk of obesity in these children based on what we see the methylation profile currently.
So the purpose is want to change the blood sample to the buccal swab, is it right? So why don't you also check the methylation from the blood as a control?
Right, it's a good question. The reason we did not do that, because it's part of the studies that has been going on for a while. We realized that when we take from the blood, there's a resistance in the populations to get the parents are resistant to get the children get the blood drawn, and also when we use the blood, it also create issue with the biosafety, because you need to make sure that you don't live around the needles and all that. So in public health, we want to make sure that, so another way to go around whether this actually validated, because most of the studies are using blood. So if it turns out that our data is no difference at all, that stop. That means our buccal swab is basically useless because they are not really similar. But we also understand from the literatures, apparently the profile of the methylation in children are actually quite correlatable between buccal DNA and also the blood DNA, and also with the neuronal DNA, which is actually quite interesting because this actually captured the early epigenetic program during the gestational period, which is it happens very, very early.
I read that in a journal that OMT is known for having high error rates. So why you choose to use it despite its limitation?
Right. So it has have a risk of high error rate, but also has other benefits. The first benefit, the obvious benefit one, is to detect the native methylation of the site. And as far as the error rate, we basically look at the very targeted areas. So we don't venture to unknown areas. If we go to the unknown areas, yes, you're right, it may have problems with the possibility of error. But we only go with the targeted one first, just to make sure whether we can replicate other previous data. And the good thing of the long reads like nanopore, at least we can tell whether there's also an imprinting one between father allele or the mother allele. So in here, at least we show first differences, whether it actually replicate the others, and but when it comes to the other novel ones, yes, we have to consider maybe there is actually an error rate. But in this area, we only focus on areas 30,000 sites of CpG methylated areas. So we don't venture all the genome. So we only focus at 30, so we can compare with other studies that use these 30,000 CpG sites.
And why you write down that? Is it mean that? Is 220 children sampling in one day? And why you write down the time, like 9 p.m. to noon?
Okay, yeah. Basically, we tried, the initial design is a cross-sectional studies. So we collect everybody from 12 different posyandu, and this is what we got, 230. And this is actually important for us to get the baseline level. Because the survey say 20% stunted, but now we can have data, but apparently in Pandeglang it's not 20%. And then from this 230, we collect only the DNA with the highest concentration first, but eventually then we can. Okay.
Judges, please finalize your scores.
Oke. Pertanyaan Anda terjawab nggak tadi tentang treatment?
Tentang treatment enggak? Sama jam juga enggak? Belum sempat.
Karena beliau memang tidak untuk treatment. Iya kan?
Pertanyaan saya, apakah ada implication untuk treatment ke depannya bisa atau tidak? Gitu.
Ya. Itu memang nggak ke treatment arahnya. Ya, treatment monitoring atau evaluasi ya. Oke, gitu kan?
So for D16, congratulations, you have won this tournament and you'll be moving to Arena 2.
Tapi kalau Anda ketemunya hanya di satu tadi, secara klinis apa maknanya?
Masih dicari.
Oh, oke. Karena kan tidak di semua biasa kan?
Iya.
Hanya satu, tapi secara klinis apa maknanya? Oke, masih dicari. Oke, fair enough. D16 ke
Arena
Arena
2.
2.
So next up in Arena 3 is D05 versus D41.
Madura. Oh ya.
Memang turun berangkat juga. Ini kayaknya ruangan turun mereka.
Oh iya.
Dan ini yang ketiga kali di ruangan ini. Iya, kayak jusnya kayak wah, jusnya nyambung. Sama jusnya banyak presentasi kita. Sekali di ruangan satu, sekali di ruangan dua, ada 3 kali. Oh ya 2 kali kesini. Kayak sekali lagi kesini. Oh ya ini keempat ya. Udah capek sebenarnya. Ini udah sore banget. Iya, udah kayak udah turun bener-bener. Oh ya Allah. Ini berapa lagi kan sisa lagi? Yang mereka atas berarti siapa yang menang? Udah ada pemenangnya udah belum kan masih udah berapa masih 2 lagi. Kita kayaknya yang di tim ini cuma ngejar 3 yang juara 3-nya. Yang di turun bracket itu yang buat juara 3 nya. 1-2 yang di bracket atas. Oh gitu. Kan nggak akan lagi mereka pas berarti. Oh gitu. Jadi kalau kita juara lomba mereka, kita akan melakukan juara masa gitu? Iya. Bisa kayak gitu? Ada tulisan biasa. Emang gitu gila. Iya. Kalau misalnya kita emang nggak akan selalu ya bu, apa kita masih ada di sini? Kalau emang mungkin benar. Sebenarnya lebih mending 1 kali presentasi terus lomba setelah pas jalan gitu udah ada karena kayak presentasi biasa gitu. Udah 1 kali presentasi. Total nilai berapa gitu?
Waktu ya. Oke, sorry ya. Ada janji yang udah lama mau janji. Oke, ini di 41 dengan 0. Oke, langsung tos aja ya.
Iya, langsung aja.
Mau pilih mana?
Kepala.
Yang ini ya? Berarti Anda yang ini ya? Kayaknya belum pernah ketemu saya tadi ya.
Oh iya.
Kepala. Mereka pilih, silakan mau apa?
Kita yang pilih.
Yeah, you choose. You want to present or defend? Defend. So let them present or let them present. Okay, you present. Okay. So we will listen to D5 presentation. Is it the slide? Already? Okay. The time is ready? You ready? Yeah.
Thank you. Here we again. I'm Sri and Cory from Team D05. We would like to present you our data about geospatial approach for hepatitis B virus in Indonesia. Next slide, please. So, hepatitis B virus infection is a major risk factor for liver cancer, and the HBV genotype itself has a different geographical distributions, and these genotypes has been associated with clinical implications and also treatment responses. Next, please. Therefore, we utilize geospatial approach to analyze the regional molecular epidemiology. We collect the isolates from 2 referral centers, one in Jakarta and one in South Sulawesi, and we perform the genotype profiling, and then further we continue to geospatial analysis. Next. Here are the results. As you can see here, from the 2 referral centers, there is no significant difference for sex and age. Meanwhile, for the subtype, we found ADW as the most prominent in subtype in Jakarta, and ADR is the prominent one in South Sulawesi. Meanwhile, for the genotype, HBV-B is the most frequent genotype in Jakarta, and HBV genotype C is the most frequent in South Sulawesi. And here is the genotype distribution. As you can see, blue is the B, red is the C. And from the hotspot analysis, we found that North Jakarta and Makassar are both are the hotspot cities for both genotype. Thank you.
D41. Ready for the question?
Okay.
Ready?
Ready.
Time, wait. Okay. Q&A starts in 3, 2, 1.
Okay. Is there any specific reason why you choose Indonesia and Jakarta versus South Sulawesi to compare for this geospatial approach?
Thank you for the question. So this is a follow-up study from our earlier study. So the novel things that we do is the geospatial analysis, and the reasons we use Jakarta and Makassar is like we want to see if there's any differences in between the West and East Indonesia, considering we have very fast expanse of the regions. And here we only compare the referral centers in Jakarta because it's also one of the lots of patients who can the patients are more accessibility is also in considerations, and in Makassar we chose that site as a place of our collaborators and also as the most referral centers for the eastern parts of Indonesia.
Okay, so what is actually the clinical relevance of your findings? Because you found genotype B and C. We understand that in type B, for example, it's more sensitive for interferon therapy. However, in the national guideline, interferon is not the drug of choice for hepatitis B. So what will change based on your finding?
So yeah, the reason we do this is sort of like provide baseline data for the overall national hepatitis B management program. So as far as we know, there's no we have the national prevalence for Hep B infections, but there's no specific genotype data in each region. So hopefully this approach will inform the policymakers to sort of like targeting more screening or more treatment efforts in that hotspot area. And also, yes, why we focus on genotype, not the subtype, is because it's more representative or more associated with the clinical manifestations of the infection.
So my question is, how did you include the patients? What is the inclusion criteria?
Yes, so this is all the samples are collected within 6 months range in 2017. So the inclusion criteria is age, because all the patients are adult, older than 18, and then they have confirmed hepatitis B infection for as measured by positive Hep B surface antigen for 6 months. So they are clinically considered as chronic hepatitis B. And for exclusion criteria, we exclude any patient that have any other source of comorbidities like diabetes or other viral infections like Hep C or HIV.
Interesting that I read another study, previous study by Sunbull. They actually mentioned the subtype B actually more prevalence in patients with acute hepatitis B instead of the chronic. So the prevalence of development to chronic is less likely in the subtype B. However, in this study, you show that the dominant, especially in Jakarta, it is mostly subtype B, but it is all of the subjects in the study actually chronic hepatitis B. Right.
Okay, so I think I have to clear up the nomenclature first. So genotype and subtypes are different parameters. So genotype, we analyze the sequence of the virus itself. Serotype is measured based on the antibody response of the host. So when you're, I'm not sure about your references, but from what we know from based on the literature, the genotype B, yes, respond better to treatment, especially for interferon, and has like less severe clinical manifestation.
Yeah, well, yeah, my reference also about genotype. So in that reference, actually, the genotype A is most likely to be the chronic development, and also the genotype D, it is more likely to develop chronic and also mutations inside the virus. So yeah, I'm just want to clarify.
You mentioned before you collect the studies is from the acute hepatitis B, right? What we mentioned here, we highlight the chronic hepatitis B itself, because this chronic hepatitis B lead more to liver cancer. So the prognosis is more severe when you have this chronic hepatitis B, because you have a chronic inflammations in your body and it's prone more to cancer. So that's what's the point of this study. And the genotype itself is like, it's vary between regions, and therefore we want to study here.
Yes. Okay. So do you also compare the viral load of the patients with the genotype results?
Yes, I think if I remember correctly, genotype C patients have slightly higher viral loads during the inclusion of the patients. Yeah.
And also for the hotspot region, I assume it is based on the address of the patients. So for the Jakarta, in the referral hospital in Jakarta, when you have the patients come from outside Jakarta, did you exclude it?
Yes, of course, we exclude it, because what we want to try is to make it homogeneous, comparable with Jakarta and also Makassar. So that's why what we put here is the regional on Jakarta, because what we don't want is too wide, because it's the referral centers stay in Jakarta, because if we put it too wide, we cannot specifically see the hotspot because it's too wide. And also because this is the first initial study, so what we want to do is to see this geospatial approach in these studies first.
So did you also put that in your exclusion?
Time is up. Okay. Thank you. And that's gonna be D41. Wait a moment. Yeah. Ready? Time?
Okay. We begin in 3, 2, 1.
Okay, thank you. Good afternoon. Let us represent our project entitled Sputum Microbiota Profiles in Hospitalized Adults with Community-Acquired Pneumonia. So dysbiosis or imbalanced bacterial composition in the respiratory tract can affecting the incidence of community-acquired pneumonia or CAP. So the study objective is to identify the etiologies and also to profiling the microbiota of hospitalized patients with CAP To confirm the etiology, we perform culture and real-time PCR, and then for microbiota profiling, we conducted 60S sequencing. We divided the subject into two groups, mild, moderate, and severe, based on the pneumonia severity index, and we also check several variables that contributing to the severe cap, and we found in the study that in patient more than 65 years old is the only risk factor that significantly statistically significant. Most of the patients are multiple pathogens followed by bacteria and virus. Then, in microbiota profiles, we found the higher alpha diversity in severe cases compared to non-severe. Even though it's not statistically significant. However, it is compared by the Spearman correlation that in the severe cases dominating by the pathogenic bacteria. So in conclusion, so in a CAP adults with mostly caused by multiple pathogens, and patient older than 65 years old are more likely to have severe CAP than younger patient, and in patient with severe CAP had higher alpha diversity but had more pathogens and fewer commensal bacteria. Thank you.
Okay. Still have time? You want to have more?
5 seconds.
5 seconds. Okay, that's it. Okay.
Okay, so for Q&A, we begin in 3, 2, 1.
Okay.
Thank you very much. It's a very interesting data that you have. Also, I'm quite curious because you select the sample of adults. Meanwhile, CIP is more prominent also in children, in infants, right? And from your background, you already mentioned that it's more positive in infants rather than children. And what you found finally is that the microbiome here in your sample is not significantly correlated with the CAP. I think what's the main, maybe the main problem is because the selected samples, because as you can see here, probably there is a host immune also plays in here. And for example, infants has the highest possibility for having CIP because they have this immune system is not well developed. I think it should be better if you have the age more than 65, because in this age also they have a lower immune systems. Then it's maybe you can have a significant microbiota profiles compared to children, compared to general adult.
Okay. Thank you for the question and the comments. And yeah, in this case, we actually include all the doubts because we also want to compare patients older than 65 and younger than 65. And also in this case, it is not statistically significant because we only took point in one point for the sputum samples, and it is on the beginning of the hospitalization. So in that point, maybe the alterations of the microbiota is not really already very high, but since the beginning, we already see the alterations. We can already see in the patients with severe pneumonia have higher diversity than the patients with the mild pneumonia. So since the beginning it is already altered, we can know that if we took another samples, the follow-up samples, it will also changing. And we know that for microbiota, it took a while to really change the whole microbiota. But already in the beginning, we can see more pathogens in patients with severe pneumonia.
Okay. I have also a question about the severe pneumonia cases, because in severe pneumonia, it's also related to probably there is a co-infection, right? How do you exclude this co-infection with the others bacteria besides the causes of pneumonia?
Co-infection, you mean in other system?
Co-infection diseases.
Yes, you mean co-infection as the respiratory infection or
also,
in
or outside the respiratory infection. How about if there is a co-infections in your sample?
So the definition of co-infection, actually, if you took the samples, so the source of infection is the same. So there is one or sorts of infection. However, for example, like in pneumonia cases, the source infection is in the lower respiratory, in the lung. However, you can also collect the sample from blood because it's already bacteremia happens, but the source of infection at one system.
Yes. What I'm trying, I need to clarify. For example, you have HIV patient with CAP. That's what I mean.
Oh,
chronic conditions.
So HIV usually we put as a comorbid, right? So for the lower immune system, so
But you don't exclude in your samples.
in this sample, no. Because if I remember, I have only one patient with HIV.
Yeah, because it's very important because in HIV you have a lower immune system. And of course, the growth of a bacteria can be different also.
Yes, of course.
What's your question?
No, it's important to have to exclude and consider the HIV and convictions.
We put it as
Yeah, so yeah, because in all the subjects we only have one sample that have a HIV. So in this condition we include all.
Do you test all the patients with the HIV?
No, so HIV actually, if it is what we call it routine examination. So every patient that come to the hospital and suspected with HIV, so all the patient will be tested by the HIV.
Okay, regarding your analysis, have you ever considered recruiting normal samples, normal adult without the infection as a control? And also, I noticed that you have multiple infections by different pathogens and bacteria. Would you consider stratifying your microbiome data based on that, the single versus multiple pathogens?
Thank you for the question. So actually, we in this study we only focus on the patients with severe pneumonia, and we can really see what happened between patients with mild, moderate, and the severe patients. And we collected samples, all the sputum samples. We try to make it like similar for all the patients, so it can be comparable. And in healthy patients, we cannot collect sputum in them, of course, because they don't have any. And in this case, we just focus on when the patients have severe pneumonia, they have more diverse, more pathogens inside them, beside compared to the mild, moderate.
So we also plan to stratify also the patient. It is still ongoing analysis. It's not only this.
Okay.
Judges, please finalize your scores.
Okay. What is your clinical application that you expect from your study? Jangan bantuin ya nanya. Lupa ya mau nanya.
We actually from this study we can see that the importance of maintaining the homeostasis of the microbiota in patients, all of the patients, but to really interfere with the microbiota interventions, we need more data about that.
Yeah, because it doesn't really give me an idea which kind of antibiotics that we want to use, right? If we have the data at this point, right?
Because that's not the focus.
Okay, to be fair, I will ask the question to them as well.
Sure.
Why don't you do the analysis or the test within the highest prevalence of hepatitis B in Indonesia? Well, judges scoring. Right?
Well, of course, time constraints and funding constraints. So that's the short answer, I guess.
But what do you think if you want to do that in the highest prevalence?
Yeah, I think there will be an interesting follow-up study if we can actually go to more endemic. So if we look at the prevalence of the Hep B positive, we have to go to the Eastern Islands, Papua and others, Moluccas and also Sumba Islands. But it's, of course, we have to make sure that what we collect is really real data on that samples, not like we have to be concerned about immigrants and also local things, I think. Local.
Are both of you clinician or not? You're a clinician.
Clinician, but no researcher.
Okay, do you think the child pool score will impact the result? The genotype?
I don't think the child postcard will impact because genotype is we it's about etiologies.
Okay.
Yeah.
So child pool is a fibrosis score.
Yeah.
Okay. Okay, that's it. Thank you very much.
So the winner is D05. Congratulations.
Okay.
Thank you, Mbak.
Masih ada nggak di sini?
Masih.
Masih ada?
Sampai selesai di sini.
Oh.
Jadi D05 still stays in this
Oh,
room.
di sini.
While everyone will move here.
Oh, oke.
Belum tahu soalnya match
All right. So this match. Oh, okay. Oh, okay.
Have all the judges received the link for the Arena 3 scoreboard?
Arena.
Is the team number correct? 16?
Yes. Group DO5 versus D16. DO5 over here, D16 over here. All right, to begin. All right, to begin, let's start with an introduction of your names and a short background, and then we will go to the coin toss to decide who presents first. All right. Please, DO5.
Okay, hello everyone. My name is Corey and this is my partner Sri. We're from the Eichmann Research Center for Molecular Biology, BRIN.
D16, please.
We are Utomo and Utomo. So Pak Chandra is in computation now going to bioinformatics hopefully, and I'm studying stunting and epigenetics.
All right. Thank you very much. Now for the coin toss, please call heads or tails. Whoever calls first. Heads. All right, heads. And it's numbers, so you get to decide whether you want to present first or let them present.
Ladies first.
Okay, then they will present first. Please prepare a presentation for DO5. The presentation will be in 2 minutes. Each judge will be able to give up to 3 marks for the presentation, so it will be a good jump start. And I will start the timer for the 2 minutes after you start speaking.
Hello everyone, here we are again. I hope you are not bored with us. I'm Sri and Cory from my team. We would like to explain to you our study about the special approach for hepatitis B virus in Indonesia. Next. So chronic HBV infection is a major risk factor for liver cancer, and the HBV genotype is vary across different regions, and it's also associated with clinical implications. Next slide, please. Therefore, we utilize a geospatial approach to analyze the regional molecular epidemiology of HBV genotypes. Here we took the samples from 2 reference centers, one in Jakarta and one at South Sulawesi, and we performed genotype profiling, and then we from these 2 reference centers, as you can see here, there is no significant differences between sex and age. Meanwhile, for the subtype, we have ADW as the prominent subtype in Jakarta, and in South Sulawesi we have the ADR1. Meanwhile, for the genotype, we have HBV-B as the most frequent genotype for Jakarta and C for South Sulawesi. Here you can see in the genotype distribution, the blue dots is B and the red is C. And from the hotspot analysis, we found that North Jakarta and Makassar are the both hotspot cities for both genotype. Thank you.
Thank you. Thank you very much. Now, to continue, to make it more intense, so the next part is the question and answer. Every question and answer is limited to 1 minute, which means that if it goes beyond that, I will stop you and you will get the deduction of 0.2. So it keeps it a little bit more lively. So because of that, D16 has the chance to ask the questions and DO5 will answer the questions. I will start the timer after your first question.
Thank you. Interesting data. Obviously, the obvious one is the HPV-C is more prevalent. But curious, when you do this geospatial analysis, any evidence from previous studies using this approach?
What do you mean by previous studies? I mean, performing genotyping or performing geospatial?
Geospatial methods. I mean, were there any benefits of doing this geospatial analysis using geospatial analysis to look at this HPV genotype?
Sure. Thank you very much. Yes. So the use of the urgency of this genotyping, geospatial approach for the genotyping is actually to see the wake of the burden of diseases. And mainly because this HBV is infectious diseases, it's very important for us to understand the hotspot itself, because it will lead to the prevention, treatments, and other public policy, health policy for preventing or to reduce the burden of HBV infection.
When you look, I mean, this is very interesting data to look at the location. But first of all, how do you define location? Either by the address of the KTP or because there are only two hospitals. So according to hospitals, only one spot, right? But how are you going to define all this? The distribution is so different. It's based on where they live or where they work.
So based on the ID, because it's the valid for all and it's the general address that you can find for.
Did you actually check they live in that address?
No, of course not. It can be biased for this study, of course, but we still rely on the factor that it can be a fake address, but for the moment we don't have a capability to measure if it's fake or not, because we cannot go to their address, knock their door and asking, are you live here or not?
Right, because it's going to change the hotspot, no?
Yes.
So if the hotspot is changing, how is reliable is this data?
Okay. So take the assumption that it will be, some of the data will be fake. I mean, they have fake address, which is limitation that we cannot avoid.
You mean fake, not
yes,
fact.
fake. Sorry, sorry, fake. So that's why we cannot avoid that one. But one thing that we can try to do here is the referral centers itself. So in general, they collect the data from majority. For example, in South Sulawesi, is from whole the various region from South Sulawesi. So it can be realistic data that we can have so far and actual data.
And also the mode of transmission. This is HPV is not by air. Generally, is by blood. And how knowing this spot is very important, because generally that means you have to know their lifestyle. I mean, when these two persons can meet each other, did you actually check also the history of this subject? Do they have certain possibility where they get the HPV from? Because to make sure this is really meaningful.
Thank you for your questions. And that's actually the question that we hope when we show this data. And this is the use of this initial study, because we want to raise the question more, why hotspot analysis happen here and there. But for example, I can give you a special explanation why this hotspot analysis will be important. For example, there is a cultural difference. For example, in Makassar, they have this cultural tattooing in South Sulawesi that could be different from Jakarta. And meanwhile, in Jakarta, you have a higher density of people, and also you have higher migrations here. So it can be it's very interesting to know.
Right, because how is it going to change the current practice of public health prevention in HPV, knowing this different hotspot, how is it going to change the policy? Because you still have to, make sure you don't get contaminated.
Thank you very much. We realize that this geospatial could be very useful. We are an archipelago countries with high problem of diseases. We cannot apply the same problem if you have a low cases. Why we want to apply the treatment more or push more money to that point when you don't have basic data for that? And that's what we want to. We want to have a targeted regions for every problem that we have. And I hope this geospatial approach can be very useful for that.
Could you entertain us what would be the example scenario you want to do as a public health organization? If, say, the Ministry of Health wants to ask your opinion using this data, what kind of specific measures you want to advise so they can control the HPV infection more effectively?
Okay, thank you for that. So by using this data, we're hoping that we can inform the policymakers to do more screenings for hepatitis B in that certain hotspot area, and hopefully with more screening, we can have the patients might have more options for treatment, and then if the patients are treated, they can have less probability to develop more severe liver diseases, because the problem with the clinical practice and the management of the patients now, they are low detection rate, low screening, which means they're low treatment proportions, and then but the disease will progress without any interventions of treatment.
Right. That could be very interesting if you actually have this good selection on the samples, because the way you select the samples are from patients. You don't select from the general populations. But if you want to do some screening, then you need to have the screening from the population first, don't you think?
This will be your last answer.
Yes, thank you for that. So for So we have the national data for Hep B prevalence, but we don't have any epidemiological data for Hep B genotypes. So for genotype, of course, we all without any we are concentrating more in patients because they have the higher probability of manifesting these infections. Of course, if you want to have more ideal data, you have to compare. We have to healthy populations and the patient populations. But I think this is also this is an initial study. So for the next project, it will be something that we can consider. Thank you.
Thank you. Thank you very much. And that is the end of the first round. Nine back and forth. Very, very, very intense. All right, now for the second part, with 2-minute presentation from Group D16. Remember that you can get grades up to 3 per judge. So, all right, I will start the time when you start speaking.
Bismillahirrahmanirrahim. This is our work in detection of DNA methylation from buccal swabs using next-gen sequencing to study stunting. As we know, in Indonesia, the prevalence of stunting national survey says 21%, but in Pandeglang it's about 30%. That's why we focus on Pandeglang. And at least from the mechanism, we know due to the chronic malnutrition, the mothers don't get enough nutrient. But how it's going to work, because in the nutrients there's a methyl donor. Methyl is required to reprogram the genome of the children or the fetus in order that they grow normally. So how we're going to prove that there's actually some problem with the epigenetic marker, we actually use this source, which is easy to get this from buccal DNA instead of from blood, because previous study has established blood as a good source, but this is not very practical in epidemiological studies. And then we also use the NGS that can detect the native DNA methylation without the need to chemical conversion, so we can preserve some amount of DNA. Next slide, please. This is actually our population. We take from 200 plus children, but we only take for this technical validation, we focus on the first 12 top high concentration DNA, which is consisted of 6 normal and 6 stunted children. And then we look at the differential methylation in the certain regions using this technology of nanopore. And the data is quite interesting, because if you look at the IGV data, next slide, please. So this is the top one is a normal, this one is stunting. This data belong to the aggregated sample of 6 samples, and 6 samples of stunting, 6 samples of normal. Apparently we can tell a difference of the methylation, because they lack less of methylation, but this is actually true because the DNA is here. This is actually to make sure that the DNA is available, but they are not methylated. And the IGF-2 has been published previously in the Dutch hunger studies, and also the Chinese hunger studies, where the mothers don't get enough nutrient, their children grow probably issue, and with the problem with epigenetics, deregulation in this locus. And we also look at different locus of the loci of other genes, a study from the Gambia study, and apparently they have a similar patterns of deregulated methylation.
Oh, very good. Perfect, thank you. Thank you very much. All right, now for the question and answer sessions, same rules. Each question, each answer will be 1 minute. I will start the time after your first question.
Thank you. It's very interesting result. And as the methylations, for the methylation, the main issue usually is the heterogeneity of the cells that you, especially when you come from buccal swab. And how you can tackle the issue of this heterogeneity in your samples?
A very good concern. And we were happy to learn from the literature that somebody else have done a correlation between different cells, from the neuronal cells, the buccal cells, fibroblasts, and also from the blood cell, from the leukocytes. Then they have a very high correlation, higher than 90%, which is quite good. Because if it's less than that, then we start wondering whether there's going to be some tissue heterogeneity. And we think that this dysregulation happens in a very early stage in the womb, when the baby is still in the mother's womb. So whatever changes in the epigenetics should be represented in all cell types. And based on these previous studies, looks like we are hoping, and we wish we're going to validate later on, that there are a similar methylation profile.
Following up this question, because, for example, if you took a sample from buccal swab, it's also beside the epithelial cells, of course, that you have, you will have also the immune cells there. But in your sample, did you check this, or?
Right. We don't check the differences, the proportion of the buccal cell, also from the DNA from the white blood cells. But based on the previous study that the correlation is quite tight, higher than 90%, then to us that could be less of an issue. The issue would be if it turns out we cannot see a difference in the established locus of IGF2, we see no difference. If it turns out to be no difference, then you're probably right. There could be an issue of the different heterogeneity. But when we aggregate the samples, we put together 6 samples in normal, 6 samples of stunting, we can still see some differences. So that could be an interesting study, preliminary study, to look for the higher sample numbers.
Okay. Thank you. We agree that stunting is actually multifactorial, right? And what you show us here is a monofactor so far. I remember that growth hormone is in one axis with thyroid stimulating hormone as well. So how can you tackle the issue? Because I got also that the TSH also can be affected, for example, by pesticide from environment. But how can you took it out, this confounding factor?
Right. Definitely, there are about 30,000 CpG methylation sites that potentially have differentially methylated. We only start with what we know from the literatures. But later on, we're going to scan the entire 30,000 sites. It has been actually studied also with a different method using Illumina because they have a microarray based. So yes, you're probably right. Maybe there's some other genes, probably the thyroid stimulating hormone. Whether this thyroid stimulating hormone also have differentially methylated, it could be tested in our data set. So right now, but unfortunately, we don't see a publication looking at the TSH methylation. Maybe they look at the hormone at the protein level, they don't look at the epigenetic side, but we have now a data set that we can analyze whatever genes that we think that actually may affect the growth of the children, maybe due to the epigenetic difference methylation.
So you're looking at methylations. We know that methylation is not a permanent modifications of a DNA. How do you know that what you're seeing this is real data that's really happening and could be the factor for the stunting itself?
Right. This is actually interesting that we think that epigenetic is changing, right? Okay. Now the question is it really changing? Because this part of the actually longitudinal studies, where children will be followed for 7 years until they go to school. And we already have data too. This actually taken from samples in 2022, and now we also taken 2023. We don't know the epigenetic methylation yet. Interestingly, the phenotype, those children who were actually initially normal, they become stunted. About up to 25% of children under 2 years old. Now the question is whether the epigenetic is really the same or they're actually changing. That could be a challenge to a current dogma, because if it turns out that they are really fixed, that tells us that epigenetic in the early stage, during the gestational period, is very important. You cannot change it anymore. It fits. So that's why it's very important for us to take all this the methylation across the different time.
Thank you. Okay, it's very interesting because you mentioned that if it's happening, if intrauterine as well, right? My question is, if we found this changing in this intrauterine, do you think we can have put it as a targeted treatment, or what's the purpose of your study?
So we wish to actually, the problem with nutritional intervention studies is that we cannot track using a method that is objective. Sometimes we just give the mothers some food. Whether the mother actually really eat the food or maybe they sell the food, we don't know. So the idea is if we can get actually from the children as early maybe as the newborn, then we can tell. If it turns out that the model is correct, then we should see as early as newborn babies, right? Then we can tell, look, the mother didn't consume the right food as we did, because we can actually use for the intervention studies. So currently intervention studies require a survey, a questionnaire. So it depends on the what the mothers remember. But having this objective marker, hopefully they can at least to make sure to clarify why the intervention didn't work. Maybe because they have problem.
Your last question.
Okay. Considering, because you mentioned it can be useful for screening tools, right? My next question is, as a screening tools, I consider it's very expensive. What do you
Right. So here, when it comes to screening, we have to really establish first how close this data to the outcome. If it turns out to be very, very near, we don't have to look at all kind of methylation sites. We probably have to choose the most important methylation that's actually predictable. But what this kind of samples can also tell us, whether we can use this to support the intervention, a randomized control study, to actually correlate with the outcomes that we see. But when it comes to the prevention, yes, obviously the prevention has to be very early before they start the pregnancy. But I think this is very important to look at the mechanism and also to look at the further down risk factor for to get obesity and hypertension. Because if you look at if this is to be fixed, then we can look for genes that are maybe increased susceptibility to chronic disease. So we can tell, look, you were stunted, you have a problem, then you have to be very watchful with your lifestyle.
Thank you, and that is it for our talk. All right, so while we wait for the judges, please everybody to come to the front and we can check it out and see whether there's some point changes. All right, so wait, is everybody in? Is all the judges in?
Wow. That was amazing. Oh.
Oh, in that case, wait.
That was a great match. That was a great match.
All right.
So our top 3 teams are D16, D49, and D45. We are moving to match 39 between
Langsung, Pak, langsung, Pak.
D49 and D16.
So this is match for third place, right?
This is the match for
This is match for Please. D49, D49, right. 5-minute break. And then 5-minute break, yeah? 5 minutes.
Man, that was awesome.
5 minutes, right?
That was awesome. So you guys No, but they're not. Wait a minute. I think there's 2 more. Hey, hurry up.
This is for the third place. Yeah, so the loser of the next match is the third place.
Right.
And the winner goes to final against D45.
So D45 definitely, okay.
Yeah, D45 is either first or second place.
Okay. All right. So let's get this one going.
Yeah. Supposedly 5-minute break.
Masih ada 2 kayaknya.
Is it done?
Tapi untuk kayak konsolnya terus kita on. Kita kolaborasi.
How about the judges? Do you want them to stay?
Let's go with that.
All right, let's go with that.
How about that? Minute break here. Coming up. So this is the third place system.
Yeah, this is our first place. All right.
So, those scores that came in later, where did those come from?
Yeah, where did that come from?
It was like it was like the Joe Biden Trump election.
It was the Joe Biden.
Malah seru Bang. Malah seru itu.
Sebenarnya kita bisa juga sih. Kita waktu belum kan udah terakhir.
Ya oke lah, oke. Start sama. It's exactly the same.
Tidak perlu internet kan?
Nggak perlu. Okay, exactly the same. Start. So, perfect. Alright. Okay. Ini third place kan berarti?
Ini mulai membuat hafal fisik yang saya tadi pas skip.
Iya benar. Just now? D16 won. Yeah. Huh?
Why Anurat Sensei said that it's not the winner?
Pak Ahmad is D16, right?
Yes.
Pak Ahmad
So 49 this side.
Enggak, yang menang siapa tadi?
D16, Pak Ahmad.
Yeah, okay.
Ahmad.
Right, okay, so D16
Because
won.
of the Joe Biden.
Oh, 5 lost.
Yeah,
Okay,
because
okay,
of
great,
the
great,
Joe,
okay, great.
5 lost. Yeah. Because of the Joe Biden judges change. Yeah.
Oke, bayangnya training dua.
Very intense discussions. I can see that a lot of you have improved your content and improved your way of perceiving research. So I just want to appreciate that for now. I also appreciate some of you are more confident on your English. It is important to first be confident and later on you can correct the pronunciation and everything. But as long as you're confident with your terminologies, that is much appreciated also for these sessions. All right, so now we have D16 versus D49, and I want everyone to know this is literally the battle for third place. So, the winner of this match will be the finalist against D45, while the loser is automatically in third place. So, I hope that everyone can give them the time for the presentation. So we a short introduction of who you are and a short of your background, where you're from, and then we continue to the coin toss. All right, please, D16.
Yes, we are Utomo and Utomo. Andra Utomo. He is actually a computational scientist. I'm actually a molecular biologist. Hopefully we can exchange ideas. Thank you.
Yeah, nice to meet you. And I'm Yeni, and this is my friend Fikria Hanum. We are from Research and Innovation Agency Indonesia, BRIN. Thank you.
Fantastic. Heads or tails? Head. All right. Heads. So you get to decide whether you want to present first or let them present first. You want to present first. All right. Here you go. Oh, okay. An orchestrated effort. All right. Okay, please prepare your presentation for D49. The presentation time will be 2 minutes. Each judge can grade this up to 3 points. So please, I will start the time as you start speaking.
Good day, my lovely audience. I want to tell you about our story, Journey to the East. Next. We went to Sulawesi, the eastern part of Indonesia, to study the most expensive wood in the world. Now, maybe some of you are asking, do we really have that, or why we have to go thousands of kilometers to study that?
Well, yes, we have ebony and endemic to Sulawesi. Ebony is one of the most expensive woods in the world because of its strength, black, and durability. However, based on IUCN, ebony is listed as a vulnerable species, and one of the factors is the plant disease.
So we came up with this idea. If we understand the microbial diversity, in this case bacteria, from the asymptomatic and symptomatic leaves, so we can formulate the natural biocontrol against this pathogen causing the disease.
Next. So at first we thought that the symptomatic one actually have more bacterial pathogen. However, we find a very interesting data where most of the symptomatic leaves actually have the potential bacterial group. As you can see here, that Spingomonas and Bacillus is actually higher in symptomatic leaves, and those bacteria are highly reported for its antipathogen activity for the plant. And how can it be happen? So this is one of the strategy of the plant to recruit, to survive from the pathogen or stress condition by recruiting the beneficial bacteria, so it can help them to combat the situation. So it is very interesting in the future that we can isolate those potential bacteria and formulate the biocontrol agent for that, and for the survival of the ebony. So we are Team D49.
Thank you, and we are open for discussion.
That was a good 1 minute 50 seconds, and now we will continue the question and answer. 1 minute for up to 1 minute for each question and each answer, and I will start the time after your first question, please.
Great works, very interesting. So the question would be, when you look at the bacterial diversity, how is going to actually affect the symptom? Is there any study previously that showed that they're actually causing the disease?
So actually, we don't know yet what is the cause of the disease in here. So we just want to know about what is the bacteria here and what is their role in the plant. So after the finding, we found that the bacterial genus and species, actually for references, we found that those bacteria is not a pathogen for the plant, but it highly reported for their good activity for the plant. For example, like biostimulation activity.
Yeah, because this is very important. If the bacteria has nothing to do with the disease, then what kind of biocontrol you want to do? Because you may just having the same diversity, but it's not.
Yeah, actually, we have a progress of the research in here. So beside the bacteria, actually, we also try to see the diversity of the fungal, because the symptoms of the leaves itself is similar to the fungal as well. So beside this, we also have the data, but we found some of the pathogen in fungal, but actually, we're still not sure that they are the cause of the disease, because we need to confirm that by isolating the fungal and do the postulate coach in the plant.
Okay, if you look at the previous data, the previous slide, if you look at the symptomatic ones, there's a degree, right? I guess the rightmost picture actually is the most severe, I suppose. Okay, do you actually differentiate between the least severe and also the most severe one? Do you see any differences?
Okay, so this is our preliminary data. So we just combine all the symptoms, even that it is like have a different characteristic, for example, like yellow, brown, or black one. But from this, maybe we can know about what is the structure of the bacteria in those symptomatic one generally. So we can found what kind of the bacteria, and then we can got this data and then isolate them to test it into the real field condition.
Do you think that in just a thought experiments, it turns out that you have to differentiate a really asymptomatic ones, less severe, a very severe, extreme severe. Should you expect there is actually a differential abundance in those different strata?
One more, sorry.
Will you see a correlation of, we call it dose dependency. So when you have much more bacteria, then you have more severe ones.
Okay, yeah. So actually, not only this, so we have also this metagenomic data of this pole and tree category. So for the pole category, actually it has more severe effect symptoms, and for the tree category, it has fair symptoms. And from that, we found that the severe symptoms has more potential bacteria compared to the fair symptoms one. So, but we don't include this in here. In our poster, we include it because we need to highlight the priority finding.
Yeah, because when going back to the Koch postulate that you mentioned, how are you going to do this? Because bacteria comes in communities. You cannot just isolate one or two bacteria and expecting they are actually dissolving. How are you going to design the studies to actually make use based on what you find here?
Okay, first of all, regarding the previous question, I would like to highlight that the golden novelty of our study, that there is a limitation of people that actually studying about this. So if we can publish this, is going to be the first report. And the secondly, second question, sorry?
Isolation of the, I mean, like the
for
test.
the Koch postulate, how you want to design the study?
So actually, we have isolate the bacteria and the fungal from this this leaf sample, and then we found some of them, like 30, I think 30 or 50 of the bacteria and fungal sample. And then after that, we for the bacteria, we try to characterize the biostimulant and also their biocontrol activity by in vitro. And then after that, we try to see the pathogenicity of the fungal in the ebony. And then yeah, we try to co-culture that in the field condition.
But you still have to establish that they are causing the disease.
Yeah, so that we isolate the fungal and the bacteria, and we want to know, especially for the fungal, we want to know if they are the cause of the disease, if they are have the same symptoms that we found in the natural forest. So for that, we already isolated the fungal, and then we found some of the pathogen from the references that we found, and then we will do the postulate coach in the ebony to see the pathogenicity of those fungal. So we will see that the symptoms, is it same or not for the one that we
I would see okay, I would see the samples. This looks like, from my understanding, you look at different trees, but trees, I'm sure there are thousands of leaves. Is it all the leaves have the same symptoms, or there's some leaves that actually have different symptoms? This way, there will be your internal control, because if you look at from different communities altogether.
Yes, the challenge is actually in the natural forest, we actually hard to find the ebony tree itself. It's very rare, and it is a very harsh condition. So we just take the sampling of the tree that we found, and in one tree, sometimes we find the symptomatic and asymptomatic one in one tree, and sometimes it's like a different tree. So because of that, yeah.
Still have 10 seconds if you have any other opinion.
Yeah, we do our best to take the samples actually, so based on the similar symptoms of the leaves.
Alright. And that is the end of the first round. Thank you very much. Alright, that was really good. Now please have at least 1 minute to discuss, unless you are confident with this overall. Alright, okay. It seems that he's okay with continuing on. Okay, so again, just to repeat, it's a 2-minute presentation. and you can be graded inside there. So I will start the time after you first speak.
Bismillahirrahmanirrahim. Thank you, the judge. We're going to express our study, detection of DNA methylation from buccal swabs using next-gen sequencing to study stunting. As we know, in Indonesia, the prevalence of stunting is about 21%, but in Pandeglang, you can reach up to 30%. And chronic malnutrition is the major risk of stunting. Why is that? Because the nutrition has we call the methyl donor, a chemical that can actually reprogram our genome. We call it epigenetic programming to make sure that the babies will actually grow up with the correct programming of their genome. So how we can detect that? Previous study using blood DNA, but now our innovation is using buccal DNA, which is actually much easier to collect. And we also use this innovative NGS nanopore because they can detect a native DNA methylation, so without any chemical conversion. So this is actually the population that we study. We for this particular training, for this particular protocol, we want to establish that the protocol is working. So that's why we choose the 12 highest DNA concentration, 6 normal and 6 stunted children, and then we run to the NGS sequences, and we see some interesting data. So next slide, please. So we focus on the previous genes that has been established from the Dutch hunger studies and also Chinese hunger studies. They look at the locus of H19 IGF2, which is actually encoding the protein for growth, insulin growth factor. But also from the Gambian study, there are also several genes that have been differentially methylated. So we aggregate together the 6 sample normal and 6 sample stunted. We see a difference. There's a lack of a methylation here, which is consistent with the idea that when you are malnutrition, then the baby don't get enough methyl. So there's a problem with the methylation. So the idea is, so this will be interesting to follow up with the rest of the 200 cases, so to understand how they work. And we also do the prospective study to track whether the children will have a consistent phenotype or they actually have a changing phenotype, to challenge whether epigenetic is really changing or they are fixed. Thank you.
Very good. Thank you. Yes, and now for the question and answer session, I will start the time after you start.
First question is, do you use a composite of sample and do it like in one run for like stunting sample and normal sample? Do you do the composite of the sample and run for the analysis of this, or separately?
Okay, we use actually, I'm not sure what, hopefully my answer will enlighten this. So the way we do this, we barcode the samples. So there are actually 12 samples in one run. And the way we can tell a difference, because we barcode each samples. So if anything that is not problem, but issue with the mechanistic of the NGS should be the same for all samples. So this actually, the example of the aggregate data of the 6 normal and 6 stunting together is not running in 6 different experiments. No, there is a single experiment running together at the same time.
But you took the sample in different time, right? So how can you, is it right? You took the sample in the different time?
No, same time.
Same
because
time?
The children got recruited from the single day. I didn't example. So in the previous one, in the method section, so if you look at here, now, okay, this actually, the children taken from one day, so there are 230 taken from the same day. And then from this 230, the top six of the normal children and the top 6 of the standard children with the highest concentration of DNA, because the NGS require certain numbers of DNA to run.
You mean like the highest concentration to extract the DNA? I think you use the automated extraction machine, right?
Right.
No? Yeah. So why don't you use any other like extraction method or something, so the quality of your DNA maybe will be better from the one that you use in the machine?
Right. That's a good one. The reason we start also using the automated one is because we foresee it's going to be a very long epidemiological studies. So when you're doing a long investigational study, then you require a very simple ways of DNA extraction. So that's why we look at the numbers, because we're talking about hundreds, probably later is thousands of DNA to be extracted. If you have to use these manual ones, then you may actually have a variation in a technical aspect, because each individual may have a different efficiency in getting the DNA.
But here that if you, I see that, but you only get 12 with this kind of methods. So why don't you optimize another kind of extraction methods?
Right. The reason we choose this 12 is because for the practical reason, we do have a range of DNA concentration across these 230 cases. And because we have to prioritize which one we have to run first. So that's why we choose this 12 top one. It doesn't mean that this is sometimes our specialty, but we want to make sure that the technology works, and that's why we try to validate it using the low side has been shown they have been associated with stunting. So if you look at the next one, this is a table, because if it turns out, look at the next slide, please. Okay, if it turns out that the normal and stunting, there's no different in methylation whatsoever, then it says our protocol basically is not working at all. So we can stop here. But the reason we are very excited because we can see a difference here, and this using automated extraction method.
Do you do any like pre-treatment for the patient that you take the sample before the sampling? Like
Pre-treatment?
I mean, like you need to, for example, fasting or something like that.
Okay. No, actually, that's an interesting part because there is no need for children to do some sort of precaution, even as long as they don't eat maybe 30 minutes before sampling, because we don't want to have a rise in the sampling, right? But this will be very easy to do because for epidemiological study, you want to make sure it's simple as possible.
But how can you sure that they didn't eat in 30 minutes? Do you like put any information for the participants?
We
or
collect
not?
all the subjects in one go. So there'll be lots of children, and there's some announcement. Kids, please don't eat. They're gonna eat after the swab, and they'll be very happy, but they'll be very compliant. As long as you tell them you're gonna get some food afterwards.
Do you have any? How can you be so sure? Do you have any the letter that saying that they are not eating something before the sampling?
They can eat, no problem. But see, they come to the posyandu, right? So we can tell, within the posyandu they go for maybe about half an hour, 2 hours. So we can say that look, they're not eating at all. It's very easy to observe.
How? I mean, is that easy to see that someone is not eating before the sampling?
Yeah.
I think that's quite hard.
30 minutes before sampling, not hours. So they can eat, they can have breakfast, no problem.
So they are like external. Yeah. So they. Okay. So. Okay. Yeah. So. What else? So I mean, like the variation of your sample is also quite high. I mean, like from the age is different, from the severe is different. Why you don't like just normalize it? I mean, like make a similar condition for the participants.
Right. So this is actually a very valid concern. But if that is the case, then you cannot see the difference at all. If it turns out that there's going to be some effect of the age and also effect of the size and all that. But again, yes, at least it could be just a fluke. These are very lucky differences that we see. But at least we can tell that this is not really a false negative. Because if you look at the coverage data, even in here, we don't see any methylation. It's not because the DNA is broken. The DNA is there. We can see these nucleotide signals here. So that means there's actually lots of important DNA is over here, but they are not methylated. So that's why when we look at this data, and this actually collected together is an aggregate. So if it turns out that the age may matter, sex may matter, then we should see all across this board shouldn't be no difference, right? But again, we want also to correlate with the quantitative analysis that we're going to do later, see whether actually the certain amount of age is going to increase a unit or a decrease of a unit of methylation.
All right, and that is the end of this round. So, learning from the previous experience, let us wait a few seconds while please all the participants come to the front and while I confirm with everyone that they have input their results. All right, okay, so passed by 0.6. Please, picture for
Totally off. That's crazy.
It's getting tighter and tighter. Everybody input that. All right, okay. means congratulations, the third place. U45. All right, so finals.
Yes.
All right, finals.
Yes.
5 minutes break and then finals.
5 minute break. You guys are in the finals. Congratulations. Awesome. Awesome. Fantastic. Good job.
Di mana? Di sini atau di sana? Sini. Alright, okay. Semua judge nih, judge. Kayaknya ratusan gini. Di sana. Oh, So, I believe are all the judges relevant to this here already? All right. Panraj, the judges are the same or? Yes. All right. Okay. The judges are of the same constellation. Is that okay with the link? Does it work that way? We can try. Okay. All right. Okay. Okay. All right. So that means the scores will be at the hundreds then, right? Yeah.
All the judges are gonna give their scores. This is the finals. Okay.
Okay. Very good.
Raise your hand. Raise your hand.
Judges, please raise your hand. All right. 1, 2, 3.
Are you gonna be judging? No.
1, 2, 3, 4, 5, 6. All right, 6. Yeah,
Great.
6 judges.
Judges. Excellent. Good idea. Great idea.
All right. So, in this finals, D45 is against D49. Now, here's what happens in double elimination. Usually, in a double elimination, in the worst case, in a case where the winner loses, the match is repeated. But knowing the format of our match, if the winner that has never lost loses in the final here, it will continue to overtime for a tiebreaker. And if during that is a tie, we continue to coin flip on who asks and who answers one question. So it's a sudden death situation. Yes, correct. So if D45 loses this match, we go to overtime and then a tiebreaker. Time is a tie. If D49 loses, then direct the first position, second position, and the day is, ended. Yeah, over. Woohoo! All right. Okay. Now, before we start, yeah.
It was. I just want to make sure it's that way, right? Because these teams met before.
Yes.
This is a rematch. Yeah. Okay. So that's why it's arranged like that. Nice work. Okay.
Yes, because it is a rematch, it's kind of difficult to keep asking similar questions and answering similar questions. And I'm sure that they are at the wits' end of their creativity, which I really appreciate. And I also appreciate everyone that remains here to watch, to learn, and to discuss together. And that is exactly the kind of spirit that in the Genomics Science Dojo wants to instill and to keep the fire on. So thank you very much. All right. So anyone who is familiar with this arena, me as the referee, we start with the introductions from each side with their names and their short background, and then we go to the coin toss. Please start with D45, please.
Hello, I'm Hera. I'm from Summit Institute for Development.
Hello, my name is Edward. I'm from Oxford University Clinical Research Unit.
Hello again. I'm Yeni from National Research and Innovation Agency Indonesia.
Hi, I'm Fikriya Hanum, and I came from the same institution with Mbak Yeni.
All right. Thank you very much. Hats or tails? Hats. All right. Hats over here. All right. Heads. Decide if you want to present first or let them present.
Let's go first.
Presenting first. Please prepare D49 presentation.
I hope you are not boring with our presentation.
Please give me a moment. I need to prepare timer. All right. So you have 2 minutes for your presentation. And the judges, which is 6 now, can give up to 3 points each. So that would be a big boost if you can get a total score. All right, I will start the timer as you start speaking.
Good day, my lovely audience. We want to tell you about our story, Journey to the East. Next, please. We went to We went to Sulawesi, the eastern part of Indonesia, to study the most expensive wood in the world. Now, maybe some of you are asking, do we really have that, or why do you have to go thousands of kilometers to study that?
Well, yes, we have ebony and endemic to Sulawesi. Ebony is one of the most expensive wood in the world because of its strength, black, and durability. However, based on IUCN, ebony is listed as a vulnerable species, and one of the factors is the plant disease.
So we came up with this idea. If we can understand the microbial diversity, in this case bacteria, from the asymptomatic and symptomatic of ebony leaves, we can formulate the natural biocontrol against this pathogen causing the disease.
Next. At first, we thought that the symptomatic leaves actually have more bacterial pathogen. However, we found a very interesting data where the symptomatic one actually have more potential bacterial. You can see here that Spingomonas and Bacillus is actually higher in the symptomatic leaves, and those kind of bacteria have been highly reported for this activity. So how can it be happen? So this is maybe one of the strategy for the plant to suppress or to survive with the pathogen or stress condition by recruiting the beneficial bacteria to cope with the situation. So it is very interesting to isolate those bacteria and formulate that for the biocontrol agent for survival of the ebony. So we are team D49.
Thank you, and we are open for discussion.
Alright, now to continue, we will have question and answer. Each question, each answer will be up to 1 minute maximum. And then it will continue as such until it is 6 minutes, or sometimes I give extra time if there's pit stops and everything. I will start the timer when D45 asks the first question, please.
Okay. Yeah, thank you and nice to meet you again. And from our team, we have concern in your result. You mentioned that the bacterial diversity in symptomatic leaves was higher than the asymptomatic. Yet, in your recommendation is to add the biocontrol agent, which is it will increases the diversity. How do you address this inconsistency?
I'm sorry, because I don't say that we want to formulate the natural biocontrol agent for increasing the diversity of the plant. So bacterial diversity on the plant. So actually we formulate natural biocontrol agent so we can do the conservation efforts for the ebony in the natural forest.
We have another questions that you mentioned the samples are 30 leaves, but there was no mention specific analysis regarding the hotspot of microbial diversity that may give the valuable insight to the dynamic of microbial communities.
So actually, the 30 sample is the composite from the 2 locations. So we take the sample from 2 locations, and then we take the symptomatic and asymptomatic one, and then we composite the sample into one sample and do the metagenomic analysis. And the total of the part that we caught is actually 30 from the random sample.
Yeah. So again, I think one of the major concerns is also about the number of samples that you took, because you're essentially taking one tree and like 30 leaves from that tree. In human, this is equivalent of instead of taking 30 hair sample, you're taking one human 30 hair sample, which is then how we'll be able to generalize this to the whole tree, even in that forest population.
Okay, first of all, I would like to highlight that this is the preliminary study. And the second, I want to tell you that the population of ebony is actually limited. In fact, in a plot of 25 to 25 meters, we can only find 1 to 2 trees because the ebony is very huge tree. We can harvest it in 40 years old of the tree.
I think the fact that it's primarily should not be enough reason to then justify certain limitation of the study. It still need to be robust. But more than that, though, I think one of the problem that we see with your study too is that in addition to bacteria, there are also common diseases that can attack plants, such as fungal diseases, virus. And this is a blind spot in your study. What about the other organism? You didn't analyze other microorganisms such as fungal or virus, and how does these three or microorganisms, for example, interact to one and another?
Yeah, so actually in the progress of our project, we also do the metagenomic analysis of the fungal, but it is still progressing, so we don't have the data here. And from the raw data, we can find that some of the pathogen of the fungal is actually maybe similar to the symptom of the leaves that we are sampling from the natural. However, we cannot say that it is the cause of the symptoms because we still need to do the confirmation by inoculating this fungal pathogen into the plant, the ebony plant.
And did you do the inoculation for the bacteria to the fungal?
Not yet, because but we already isolate. First of all, the bacteria that we already isolated, and we already know that they have a biostimulant activity and biocontrol agent against the fungal. For example, like the bacteria have the nitrogen fixation, IAA biosynthesis, as well as the fungal pathogen for biocontrol for the fusarium, for example, and many other pathogen, fungal pathogen.
Sure. So you suggest a potential role for certain bacteria as a biocontrol agent. How feasible and ethical this is to implement this? Because I don't think it will be practical for large-scale effort, especially as you mentioned, like ebony is very rare. Particularly also as we are unsure about unintended consequences of introducing a new bacteria to like this diverse ecosystem that we are not sure about.
Actually, this is not a new bacteria. This is we isolated from the ebony leaves itself. So this is indigenous bacteria that is already present in the nature. And yeah, for the application, maybe it is very a bit difficult, but we try to plan to make the formulation of the biofertilizer by using the so we can like distribute the fertilizer into the natural forest, especially for the ebony part itself.
Yeah, but
This is your last question.
yeah, but I also have a concern about the genetic variation. Well, you actually identify genomic variation for the bacteria. A blind spot of your study is also you did not examine the ebony genetic diversity, whether or not the plant's genetic diversity influenced the disease susceptibility and the ability to recruit beneficial bacteria too.
Yeah, actually, we have our team in the botany field, and they already do the experiment for the variation of this ebony, and this is belong to the Diospyros celebica bug, and this is a specific ebony in East Sulawesi. So when we do the sampling of the ebony, and we also invite those expert to take the sampling from the natural forest.
I'm sorry, but that's you have 5 seconds, which is gonna be over by the time I finish this. So thank you very much, and that's the wrap. All right, that's a very tight score. Okay. Okay, so you actually have 1 minute to prepare if you want. You don't want? All right. Okay, in that case, we can start by.
I need to take over the presentation.
All right. Okay, the presentation will be 2 minutes, and as I said before, each judges will have the chance to grade that up to 3 points each. I will start after you start speaking.
Hi everyone, I'm Edward, and here with my partner Hera, we'll be looking at side effect or reactogenicity of COVID-19 vaccinations. So COVID-19 vaccination has been established to create a poorer health outcome both for the pregnant mother and the children, and therefore vaccination remains a crucial public health tool. The problem is right now we do not know much about whether or not different vaccine type might cause different side effect too. Given that most studies currently right now comes from developed countries which primarily use vector-based or mRNA vaccine, we aim to fulfill a knowledge gap by conducting a real-world COVID-19 assessment among pregnant and non-pregnant women in Indonesia. So we enrolled a prospective cohort in Lombok from July 2022 to October 2023, where we has vaccinated women for their symptoms after vaccination for each day up to the 7 days post-vaccination through WhatsApp, by phone, and by home visit.
In our study, almost 85% of our participants has reported any symptoms of adverse event within 7 days after they got the vaccination, which is nearly 30% for inactivated vaccine and 62% for mRNA or vector-based vaccine for any local and systemic reactions. And in our study, we are found that the pregnant woman experienced a lower reactogenicity compared to non-pregnant women, suggesting the potentially better acceptability of inactivated vaccine compared to mRNA vector vaccine among pregnant individuals. I'm sorry, but further analysis still necessary to recommend the best vaccine for pregnancy, for use during the pregnancy. Thank you.
All right, thank you. Okay, and now we go to the question and answer, which we can start. Which I will start the timer after you give your friend.
Okay, I have one question. Actually, what is your expectation from the result of your study? Because the outbreak of the COVID-19 is already like over. So the COVID-19 is already past, and maybe there is an outbreak, maybe there is a mutation of the virus, and you need to develop a new vaccines, and then you need to do again this kind of study?
So two responses to this. First, when we did the study, the COVID-19 is actually still very paramount, right? Like we're not forecasters, so we're not able to foretell when it died out. But secondly, the important thing about this study finding is that certain part of it can be generalized to other vaccines moving forward too. So the fact that inactivated vaccine may cause less reactogenicity, those kind of findings most likely will be able to be generalized too.
I think that your experiment from 2022, right? So this is, I think the COVID-19 is start from the 2019, as I'm not wrong. So why you start a bit late?
Yes. So we do need to remember that the vaccine was not even here up until late 2020, early 2021. And even then, Indonesia did not get the vaccine early on. And even then, the pregnant women need to wait until the guideline from Indonesian government and in the Indonesian BOGI. But we set up the trial very quickly once the ethical clearance is done and the other stuff is done. So this is actually pretty natural timeline. Yeah.
So you said that you can generalize for example, like inactivated virus, and then there's like outbreak of the new mutation of the virus. So you said that it will be the same result like this, right? How can you say that?
Yeah, sure. So usually study builds on top of another study. If there are new vaccine, and for the we would still probably have a study for reactogenicity, but what our study have is that if the finding result is consistent, that means this creates a body of evidence that inactivated vaccine is less reactogenic compared to mRNA vaccine. So again, it adds to the body of evidence, even if it's not relevant anymore. And I would dispute COVID-19 is not relevant anymore.
And
okay, regarding that, I think this is very important because considering your reliability of your study and your eligibility criteria about this, because you said that the COVID-19 may be mutated because of course the human body also adapted to the virus.
I'm not sure I get the question though. So the question is how reliable?
Yes, please.
What is your eligibility criteria of your participants?
Oh, sure. So that has been shown in the poster. We didn't show it in the slide, but the eligibility criteria is pregnant women. Because this is a pragmatic trial, there's not a lot of inclusion or exclusion criteria, but they do need to be aged 12, above the 12 weeks of gestational age and below the 30 weeks of gestational age. These are again coming from the Indonesian MOH recommendation. So our eligibility criteria for this study is exactly the same of eligibility criteria for pregnant women to get vaccination.
I also see that your graph. Can you see the graph? Yes, actually the reactivity of the inactivated vaccine and mRNA factor is actually more higher in the non-pregnant women. So how can it be happen? It's not really, you can see here that this is more higher and this is like the response is actually less compared the non-pregnant women.
Yes.
So, but you said before that the pregnant woman is susceptible or, yeah, with the vaccine or something?
Yeah. So if you compare it between non-pregnant women and pregnant women, what happened is that non-pregnant women have more reactogenicity. They experience more side effects. And the reason for that is because the immune response from non-pregnant women is much more robust. Because their immune response is much more robust, when they get injected with a vaccination, their body trigger more immune response to that. That's why they have more side effects. So this is very consistent. Again, what we are showing here is that pregnant women have less reactogenicity and inactivated vaccine have less reactogenicity. Those are like the true big takeaway from our study.
So even your like the control non-pregnant woman has like a not homogeneous sample because you said that robust diversity or something. You said before.
This is divided into 4, right? Like pregnant women, non-pregnant women, inactivated, and factor-based vaccine. Yeah, they're pretty heterogeneous in terms of the population within, because we did have some control between matching of age, matching of location too.
Okay.
This is gonna be your last question.
I'm still going to discuss about this graph. That you said that you have you did the statistical analysis and you did the ANOVA, and it is significantly different. But you did not assess the difference, such as with Duncan or 2K, and why did you choose the chi-square instead of the other tool? And how do you sure that this tool is reliable?
So we choose, I thought Taki is for post hoc analysis for multiple comparison. I need to check, but that's what I understanding. But this isn't necessarily a lot of multiple comparison. We are just comparing between generally 2 different groups, and usually ANOVA and chi-square are adequate for that statistical analysis. We don't even need to go to Fisher exact test because our number of population is pretty large. It's like almost 2,000. So these are adequate for our statistical practice.
And I'm so sorry, but that is the end of the round. And thank you very much. All right. Learning from previous experiences again. Please, if everybody can just go forward and then we face everyone while we wait for the judges to input everything, because sometimes polling happens. Please. Let me just confirm all the judges have inputted everything. In that case, congratulations, Steve. And congratulations, D49, for being in the second place. Awesome. So thank you very much. Congratulations. All right, and a big hand.
Great, thanks for fantastic. See you in Lombok. Congratulations.
Please, picture. Picture. All right, thank you.
Amazing. That was an awesome patch.
Okay, thank you everyone. Congratulations for the top three, but especially for the first place. So I think, as Dr. Anwarsh mentioned before, we're moving back to the auditorium for several announcements for particular winners, I think. So you might be intrigued in finding out what that is. So I'd like to invite everyone over to move back to the auditorium. So see you there.
Thanks. Thanks. I think it's a good format.